Hey, hello everyone. Good evening. Hope all of you can see my screen and hope all of you can hear me. Can I have a quick confirmation, guys? Can someone please confirm? Are you able to hear me?
Yes,
Yeah,
sir.
it's good.
Hi, sir.
Hey,
It is
hi.
visible.
Perfect. Thanks for the confirmation, guys. All right. So I don't know how many of you are already part of something related to clinical or something related to pharmaceutical. I'm not sure of that, but I'll be under assumption that all of you are newbies to this particular technology. I cannot call it as a technology, I can call it as a domain. So I'm going to be your trainer for next, I can say 30 to 45 days of the time, and I'm going to deal complete clinical SAS with. All right. So just to add, clinical SAS is nothing but a basic SAS technology. I cannot call it as basic exactly. I can say that a SAS technology along with all the clinical standards, right? Here, we need to understand that you have already completed the SAS part. That is what my assumption is, right? I can see some of the people who have already learned SAS from me. I could see Tejashwini, Mohanty, who and all joined here. So I know these folks who have already completed the SAS training. I'm also under the assumption that others also have completed your basic SAS, I mean to say how SAS works, what are the manipulations we can do there, how you can do the ETL, how do you do the reporting, and what is the basic analysis, right? So I'll be under assumption that you have already completed that. And in clinical SAS, we don't discuss anything or we don't what to say, teach anything related to the SAS syntax or something related to SAS procedures. No, I don't deal any of that. Instead, I'll be concentrating most of my time on explaining the clinical standards, or I can say that all the clinical domain related stuff. There are so many domains, we're going to discuss one after the other. And while learning these different domains, we are going to use SAS skills. We are going to use SAS skills to implement few of the things. So how it is going to be? We are going to start learning about all the clinical domain knowledge. The main purpose is to acquire the clinical domain, right? Along with that, we are going to use our SAS technology skills to implement few of them. This is how we are going to proceed, right? So it is going to be most of the time we will be discussing about the clinical standards. Right. Today, I'm not going to touch any of this. I mean to say that what are the various domains we have, what are the other stuff. I don't touch any of that. Today, strictly I talk about the very basic or the core structure of this pharmaceutical thing, the core structure of this clinical thing. I'm going to talk very basic. I mean to say, I consider all of you are the beginners in this particular domain. So I'll start from the very basic. So without wasting much time, let's start discussing about our very first topic of the day, which is nothing but drug life cycle. Maybe even before that, we also need to understand about what is called clinical trials. Most of the time we use a term called clinical trials. So what is a clinical trial, how and why it is important, why there is so much of research around it, what are the outcomes of this particular clinical trials, to implement these clinical trials, what and all required, and finally we discuss about where SAS comes to the picture, right? Because we are talking about clinical SAS, right? Why SAS is required in the clinical trials. So all these things we are going to discuss in the today's session. All right. So with that background, I'm going to start my discussion. I'm not a very big fan of using the PPTs and all, but since it is going to be more theoretical in nature, today's session I'm going to use some of the PPTs. All right. So before I proceed with anything else, I just need a confirmation from you. Are you clear with what we are going to discuss or what is going to be the agenda for today? All of you are comfortable with that? Or any questions? Feel free to ask me. All right.
It is okay, sir.
Perfect. All right. So let's start with our very basic discussion about what is this clinical trial. We are going to use some of the real world scenarios to understand this particular piece. All right. Okay. First things first. So when we talk about the clinical trials, the very first thing we need to understand is, because without a medicine we cannot live. That is the current scenario, correct? Irrespective of how healthy you are, irrespective of how strong family background you have in terms of your medical history and all, though you are coming from a very good solid background where you don't have any of these hereditary diseases or and all, but still medicine is the most basic thing, basic need which is in the current scenario, right? It is very, very basic. I'm not talking about the medicines which is going to cure us. I'm also talking about the medicines which are going to prevent the diseases. I'm also talking about the different equipments which are using on our day-to-day basis. All right. So we talk about, before we take any medicine, there are some of the treatments which that particular drug will undergo, right? So any medical drug you take, anything, right? Or you consider any medical treatment, or you consider any preventive drug that you're talking about, or any medical devices that we are using for our safety, or which are we are using for the diagnosis, they will go through a specific cycle. They will go through a specific process, and those cycle or those processes we can call it as clinical trials. What is that? I'm not talking about just the medicine or just the drug that we are taking. I'm talking about a broader picture. The picture that I'm talking is that can be a medicine which will cure the disease, or that can be a medicine that is going to prevent the disease, or we are talking about some of the devices which we will use for diagnosing our all these together, irrespective of whatever it falls under, whatever the category it falls under, it will go through a specific cycle, and that cycle we can call it as a clinical trial, or even you can call it as a process, right? So to be specific, what exactly it deals with? Mostly I understand that it is going to be a clinical trial. Okay, fine. So they will go through certain procedures. What are them actually? On a very high level, I'm talking about at a very high level, right? Clinical trials is mostly about research studies, deals with either medical or health related. There is a minor or thin line between these two, right? So there are going to be medical research studies and there are going to be health related studies, research studies, because most of the things falls under your R, right? So if you carry out those trials on the human beings, you can call them as the clinical trials. Similar kind of a trials we can also do on the animal. And if you do that, we can call it as a veterinary study, right? But we are not interested to go with the second piece for now. We mostly concentrate, or not, I cannot use the word mostly here. I can say that 100% we will be concentrating on the first one, which is nothing but the research studies that we are concentrating on medical or health related stuff. We can call it as the clinical trials. Guys, I understand that some of you might have clinical knowledge, prior clinical knowledge. For you, it might be a bit boring. Today's class might be boring for you because you already know all this. If you are not aware of this, but still if you are part of the clinical process, well and good. But if you are already in the industry for some time, and if you already have a very good detailed understanding about what I'm talking about here, maybe you can just take it as a a breezy note, on a very light note you can consider. All right. Otherwise, you may drop also. Today's class is primarily concentrating on the clinical trials. Hope I'm clear with that. Because some of you may feel that, hey, I'm already aware of all this. I know all this already. Why you're going with this? Because you might aware of this, but the people who have joined along with you may not be aware of it. That's the reason we are going in. All right. So just to start, I started talking about what is called clinical trial. So a clinical trial is nothing but it is going to be a research or a medical research, or it is going to be a health related research that happens on the or that carry on the humans. The same research, if I do it on the animals, we can call it as a veterinary. Right? So that is the first part. Now, just go with why it is important. What happens if I don't have a clinical trials? Correct? What happens? If I don't use them, there are a lot of definitions around it, why it is important at all. But in a simple way, if I want to talk about it, only two factors which are important to me. The two factors which are important to me are how safe the medicine is. The first thing is going to be how safe the medicine is going to be, right? I can have hundreds of medicines that are available in the market, correct? But some of the medicine we will take, some of the medicine will take strictly under the physician guidance, the general physician guidance. Some of the medicines, especially in India and all, we can even just buy it from our local medical shop and we can have it. But few, or definitely we need to have under the some general physician supervision. Otherwise, we cannot have it. Why? Because safety. If you have a normal paracetamol, anyone can have it. There is no problem at all. But when it comes to the medicines, few of the medicines, until and unless you have a proper supervision, you cannot have it. So the main goal or the main objective of carrying the clinical research is to ensure the safety of the drug. Number two, along with the safety, I also need to talk about the efficacy, or you can simply call it as efficiency. Maybe we can go with the simple general terms. We also need to talk about the efficiency. All right. So why efficiency is important? Why efficiency is important? Um, there is a debate always going on, but on a lighter note, I'll take this. I'm not criticizing anyone or I'm not supporting anything here, but generally I'm talking. There is an Ayurvedic medicine and there is an English medicine, right? Both of them are going to cure the respective disease. Let's say that. Most of us will be more inclined toward the English medicine because the results are quick. I can simply say that those are more efficient compared to the other Ayurvedic medicine, correct? Because both of them are going to cure the disease, but we need highly efficient drug. The one which is going to provide the quicker results, the one which is going to provide the better result, at the same time, the one which is supposed to be safe. So the main objective of the clinical trial is to provide or to ensure these two factors. What are them? The drug must be safe enough. Number two, the drug must have the very high efficiency. Is that clear? Right? A very simple stuff. This and all, most of these things are well known to us, but still, we're just having the baby steps towards the clinical science journey. All right. So what is the scientific method? Because if you see this, the previous slide, the first one I haven't discussed about, or I just carried on the second one, because why it is important, I concentrate on that. But prior to that, in clinical trials, on a lighter note, the researchers, the R&D department, research and development department, they take all the results from the basic scientific research and then they translate them into the different ways to prevent or to treat or to diagnose the disease, right? Let's say if you take the vaccines like, the corona vaccine, COVID-19 vaccines, right? All these are vaccines. Why we are calling them vaccines? These were taken to prevent it, just to avoid fall under your COVID, just to avoid that particular one, correct? That is a preventive one. So let's say that you already have fever, so you're suffering from fever. So you are going to take some paracetamol. The drug here is going to cure, to treat the disease that you're already suffering with, right? And there are few which can be used for the diagnosis purpose, correct? So there are some eye drops which will be used to dilate your eye. The primary reason of dilating your eye is going to be to see how good your retina is, to see what are the issues that are having, various issues that are having with your retina. To go with this particular retina test, your eye need to be dilated. For that, you need to use the diagnosing basis, correct? The eye drops that I'm going to use, not to cure, not to prevent, but this is used to diagnose my eye. So there are three different types we have here. One is to prevent, second is to treat, third is to diagnose. But to produce all these ways, or to translate my scientific research into these three ways, I'm using the clinical trials. But that leads us to a question, and that question is going to be, what is called this basic scientific research? That one we are going to discuss here. So the right-hand side diagram is pretty much self-explanatory. So that is going to give you the answer for what exactly is the scientific method is, right? So clinical trials are nothing but the real-world applications of the scientific method. Let me talk about what exactly this is. Let's say consider any drug, whatever the drug it is, all right? Consider any medical equipment. Maybe you can consider your, the glucometer, which I can use it for testing my, glucose levels, or you can take your BP machine, or you can just use your, the device which I can use to check my temperature. All these are various devices, correct? So any of this drug or any of this supporting device, first it will get tested. The moment the test is complete, we raise a question. Okay, so I just use this particular one to check the temperature. The question that I'm going to ask is this the correct temperature or not? I need to cross verify that. So a question is going to be raised. Then followed by a hypothesis is made, mean to say that, hey, I'm getting this temperature under the normal room temperature, correct? When you are within the room, you are checking your body temperature by using your whatever the equipment, either it can be a sensor one or it can be a holding one, whatever it is. I'm using it just to test the temperature. What if I'm in a extremely different conditions? I mean to say, I just switch on my AC. I'm under 16 degrees temperature. My room temperature has come down to 16 degrees. What if I use the same device to test my temperature? Will I get the same result or not? Right? A hypothesis is going to be. And then again, we are going to conduct an experiment. Because I made a hypothesis, I have asked a question. To fulfill these two, I need to experiment again. I'm going to get the result, new result. That new result we are going to analyze again. We do this multiple times. So what we are doing, I have an equipment. I will give the equipment to you. You started experimenting that. So what you are going to do, every time I give a device to you, first thing is you are going to test it. Then you are going to raise a question. Then you are going to make a hypothesis. To fulfill your hypothesis and to answer your question, you are going to do a retest on that particular one to meet these two requirements. The results are going to be generated. Analyze the result and get a conclusion. This conclusion, you can get it in the second test, or you may get it in the third test, or you may get it in the fourth, or you may get it in the nth time. That nth can be any number, right? A person like me, maybe I'll go with 10 different questions, 10 different hypotheses I'll come to a conclusion. Someone else is not satisfied with that. They may go with the 20 different questions they may raise, 20 different hypotheses they may make, and finally they can come to the conclusion. Someone who is not interested, who cannot wait for such long time, they conduct just 2 or 3 tests and they can come up with the conclusion. That can be anything, but all these falls under your science. So we are talking about why clinical trials are important. There we understand that we are going to have a scientific research. The scientific research we are going to systematically convert or translate into 3 different ways. That can be either of a preventive drug, or it is going to be a treatment drug, or it can be a diagnosis device. Then we come up with a question called, what is the scientific method? Scientific method is all about whenever I give a drug or equipment to you, you are going to test it, you raise a question, you made a hypothesis, get the new test done, get your results, finally analyze it. This is what we are doing here. Any questions? What is called scientific method is? Guys, the reason why we are discussing about this is, this may not be straight away affecting your day-to-day work, but if you're appearing for the interviews, right? Someone who really want to test your basic domain skills, domain understanding, then you are going to face these questions. That's the reason I'm starting from the very basic. Clear? Any questions? All of you are clear with scientific method? Can I have a quick confirmation?
Yeah, it's clear.
Yes.
Yeah.
You can sure. You can also respond by using your chat window. So I'm okay with that. I'm happy if you respond orally, but still, if you want to use your chat window also, it is good. All right. Now, 3 different questions are asked. What are clinical trials? We have a basic clarity now. Number 2, why are clinical trials important? Yes, we also discussed that. Number 3, how are clinical trials like scientific method, like the scientific method? All these three we already covered. Now, let's move on to the next topic, a very important one. What are the types of clinical trials, right? When I say types, someone needs to define it, right? Everyone have their own definition of what is what, correct? So someone need to standardize it. Someone need to mention that, hey, this is what the definition is. Everyone need to follow it. And that someone is nothing but National Institute of Health. The National Institutes of Health define that there are 2 types of clinical trials. The number 1 is going to be, there are not 2 types, so sorry about it. There are few more. So first we start with the first one, which is nothing but treatment trials. The treatment trials is all about, you're going to either test a new treatment, you're going to test the combination of different drugs, or you're going to test a new approach to the surgery or a radiation. Let me give you an example. Okay, let's talk about a surgery, right? So appendicitis, one of the most common, I can say that 3 or 4 out of the 10 people generally get appendicitis, right? It is not going to be a disease, but they may undergo this particular procedure. At least 1, or I can say 2 out of 10 people will undergo this surgery, right? Initial days, I can say that around 10 years back, when someone have this problem of appendicitis, so on the abdomen, lower abdomen, they are going to make a cut, at least I can say that 10 to 12 centimeters long, right? And then they are going to take away that appendicitis part, right? If any other surgery required, they also do that. And then they are going to put all these stitches and all. We need to take a proper care of those stitches for at least for 10 days or 14 days. And then again, we need to visit the doctor. The doctor is going to check how that surgical area is going to be. And then finally, he or she is going to remove your stitches by cutting them. Correct? And then again, we need to take some more care to get healed with that particular wound. Maybe after 20 days or maybe after 30 days, you'll become normal. Correct? There is a procedure of approximately a month time from the day you undergo the surgery. It will take minimum of 1 month to become a normal guy. Correct? I mean to say that complete healing of the wound. So what's the next stage? Now we are talking about, this is the treatment. So what is the improvement to this particular treatment? This is a new approach to your surgery. So we slowly started moving from, instead of having these nylon stitches, right? So why can't we have these dissolvable stitches? Those will be automatically dissolved and they will be fallen down, correct? So what is the impact of it? The impact is going to be less number of stitches are more sufficient. Number two, you don't need to consult a doctor one more time for removing your stitches, right? And the third thing, the total time it will take to get healed is also going to be less. So there is a slight improvement. So to bring back the normal life, you don't need to wait for 30 days. Let's say that span has been reduced to 20 days. What's the next stage? So we tested one more way of approach, then we moved to the laparoscopy. So what we are doing there, there's no need to cut your abdomen, lower abdomen, at least 8 centimeters or 9 centimeters. Instead, we just make a small hole. Through that particular hole, we do the necessary thing, right? In this way, you don't even need to wait for 20 or 25 days to get this healed and everything. We just make sure that it is going to be more effective and it will take less amount of the time, right? That is nothing but post-operation, the post-surgery, the amount of time that you need to spend is going to be less. Can I say that this is going to be a new approach? Obviously, all right. So then coming to the treatment of drugs and all, like combination of drugs and all, this is always going to happen, right? So let's say that for someone who come to a doctor, let's say that, hey, I'm having some severe body pains, along with that I have some lower temperature. So before, doctors used to write, a different kind of medicine for the, body pains, and there is going to be a different kind of medicine for your fever, and there is going to be antibiotic just to make sure that all your things under control. Nowadays, instead of having 3 different drugs, doctor may prescribe a single drug which will be efficient enough, because which is having a medicine combination which is going to treat all these 3 problems, right? So treatment trials is all about either finding the new treatment or combine the existing drugs to make sure we are going to produce a most effective drug, or always find new approaches to treat your existing surgery or to improve your surgeries, correct? Number 2, prevention trials. There is always a better way to prevent a disease, right? There is always a scope for that, and we are continuously working on it. So one of the best thing is going to be COVID, right? So it literally shaken the entire world, but now we are somewhat confident enough that, right, at least we can prevent falling sick because of the COVID, because most of us, I can say that 100% of us are already got vaccinated. So vaccine is, like, I can say that one of the preventive measure. So we used to have the vaccine in the childhood itself, maybe that within 2 to 3 years of our age itself, we may have vaccine for polio, we may had a vaccine for pneumonia, we may had vaccine for malaria, correct? There are so many vaccines now, hepatitis, there are a lot of stuff, right? All these are preventive actions to make sure that, to prevent the given disease. In addition to that, I told you there are few more. So what are they? Diagnostic trials, right? Diagnosis is nothing but, let's say that you suddenly feeling even under 24 degrees centigrade of AC, still you are sweating, you are getting sweat, right? So what is the meaning? You may have BP, you may have high blood pressure. That is the reason, though you are under AC, still you are sweating, So you need to get diagnosed to see whether you have the blood pressure or not, high blood pressure or not. If not, what exactly the root cause? Someone need to diagnose it. That's where we have all these labs. Correct? Then screening trials. Always find the best way, always try to find the best way to detect or treat the diseases, right? So what is again the example? We can consider COVID itself. COVID can be screened or can be detected in 3 different ways. Number 1, you can simply go with the rapid test. Take a simple, yeah, salvia or anything, just take that, just use your rapid kit and find out whether you're COVID positive or not. There are high chances that there are going to be false positives, right? So what generally we do then, right? Second thing is we go with RT-PCR, correct? So under that, again, when you go with the RT-PCR, it will go to the number of different tests, the different types of screening to identify whether you have any COVID related stuff or not, right? Again, under the RT-PCR, there are different measures. Some labs use, I don't go deeper into that, but each lab will go with different ways of different ways of screening your RT-PCR. Even after RT-PCR, let's say that you got a negative there, but still you're feeling sick. Your doctor is not convinced. What's the next method? He asks you to go with CT scan. Hey, go to the CT scan, make sure your lungs got scanned. So there he will identify what level you have there, what percentage you have there. Based on that particular percentage, the doctor can come to the conclusion. Can I say all these are screening trials, right? So you have a rapid kit test, you have your RT-PCR, you also have your CT scan, right? So you have multiple ways to identify or to detect the best thing, correct? Last one, quality of life trials. In India, this concentration on this particular trials, I can say that little less. But if you see European countries compared to anyone else, European countries, they spend most of their trials on this particular. If you see any new drug that either comes from India or it will be somewhere from your Israel or it can be from USA. Most of the drugs I'm talking about, not like entirely, most of them. But if you see European countries, especially countries like Switzerland and all, Austria, Vienna, they concentrate more on quality of life trials. That is, try to explore more ways to improve the comfort and quality of life of the people, right, who have some chronic illness, So if you see that someone is bedridden, right, the way we treat or the way we live with a bedridden person in India is completely different from the person who bedridden in somewhere in Europe. The equipments, right, the different equipments we have to handle that will be way better compared to India. I'm not criticizing. There are a lot of factors, right? There are a lot of factors why we are not into that particular stage yet. But still, the amount of time, the amount of money that we are putting on this particular trials is less in India compared to the other countries. Clear? So what we discussed here, we discussed about what are the different clinical trials types. What are them? The first one is going to be the very basic one, which is nothing but your treatment trials, correct? So this is nothing but to improve or to have a better treatment. And the second one is going to be your what is the second one? Preventive vaccines. The third one is a diagnosis, and is that third one diagnosis only, right? Yeah, diagnosis. And the fourth one is going to be, sorry, the next one is going to be like, once we have treatment related stuff, then we have preventive one. Third one is going to be the diagnostic one. The fourth one is screening, right? And the last one is going to be how to improve the quality of the life, which is nothing but quality of life, right? So these are the various trials that we generally have in under the clinical trials, correct? Now, any questions on this types of clinical trials? All of you are clear with that? Right. One main thing we all need to understand is, I have expected this question, but still I haven't get it. I told you that we don't spend much of, or we don't do much of clinical trials compared to the other countries. We means India, I'm talking about. The primary reason is clinical trials is nothing but it's more under the research and development. And generally, this research and development, the success rate is always less, a very minimal success rate, right? You need to do a lot of stuff. You need to go with a lot of questions you need to ask, a lot of hypotheses that you need to make, a lot of results you need to analyze to make sure the given drug or the given equipment is working as expected or to meet the demands or not, right? So definitely it is a more time taking process. It won't give you the immediate returns. There are long-term returns to you all the time. So you need a sponsor because you need to invest a lot of money there. Whenever money is there, they require a sponsor. So all these clinical trials are generally funded or sponsored by all your pharmaceutical companies, right? So where these pharmaceutical companies are there, or where exactly these clinical trials will happen? These clinical trials can occur at sites, mean to say, all these trials can carry out at the regular hospital's research and development department, or there are medical universities. So these trials can happen under these medical universities, or some of the specialized doctors. So they won't go with the regular treatment and all. Their primary objective is only to concentrate on the research and development. So those are nothing but the doctor offices. Some of them may happen under the community clinics, right? Or there are few people, they are dedicatedly working as a clinical trial contractors. In those offices also it can happen. So what is more important to us here? The most important thing here is going to be where these clinical trials happens. The clinical trials happens at any place. Say it can be a regular hospital research and development department, or it can be under the medical university, or it can be under the supervisor or a specialized doctor office. It can be a community clinic, or it can be at the offices of your clinical trial contractors. All right. So before we go more detail into this, let's say that what are the various clinical trials we have? Clinical trials generally happens in the phases, right? So which is nothing but the very first step is nothing but preclinical trial. Preclinical trial. It is the first stage. What exactly the preclinical trial is? You're finding a new drug, or you're going to bring a new medical device, or you're coming up with a new procedure that we haven't done on any human being before. So before you do this particular trial or testing on the human beings, you generally do it on animals. Remember this. All the preclinical trials, the first trial always happens on the animals. Why do so? Why we need to do that? To understand the mechanisms of the action, to determine how well the treatment is working, and to see the main thing, is it safe to test on humans or not? Of course, we are not so generous. Of course, we are not doing the right thing towards the animals. That and all the second story. But the first thing that we need to understand is the initial stage for your clinical trials is preclinical trial. The preclinical trial must be carried only on the animals. Why we are doing that is primarily to understand how safe it is, whether it is working or not, and what exactly the mechanism of that particular action. All right, so that is the most important thing. Okay, let's take a small pause here and try to understand this. Who sponsors the clinical trials? We understand this. Why it is important to test the drugs, we understand this. And why it is important to test the drugs on the animals rather than the human beings in the first stage, we also understand. All of you are clear up to this? If I summarize what we discussed up to this, we understand what is called clinical trials, the first thing. And then we understand the types of the clinical trials. And then we started with the phases of the clinical trial. In that, we just started with the pre-clinical trial. It is generally carried out on the animals. Questions? Any questions up to this? All of you are clear? All right. Questions? Fine. This is the another important thing. So let's say the pre-clinical trial is done. Now we are entering into the phase one. The phase one is always happen. It always going to do only on a smaller group of people, and the range can be from 20 to 80. That is nothing but the number of people who will be part of this particular phase one or maybe minimal 20 to 80. If you remember, when we go with this COVID vaccine, right? So which one it is? I think Bharat Medicals, right? So only within the organization, whoever are part of this particular R&D, only those guys have taken that particular vaccine first, right? A very minimal number of people. Similarly, so always the phase one is only on a smaller group of people, and it should not exceed 80. Sometimes it can be even lower, under 20, even 10 people will be part of it. But always make sure that it is going to be a very small range, right? The main purpose of this phase one is, again, at every point of time, we will be talking about the safety. That is the main thing, safety. So first we are going to evaluate the safety. We know that it is safe enough on the animals. That is the reason we are taking this particular medicine or we are using this particular device. But again, we need to check the safety of the human being. We need to evaluate that. Number two, at the animals level, we don't look at the side effects much, because side effects are not going to be considered as a major aspect when it comes to the animals. But when it comes to the human beings, we start evaluating the side effects as well, right? What are going to be the side effects? And then main thing, when I say that, hey, I'm just going with the clinical trials on a smaller group, from where these people will come? How do you find them, right? Most of the time they are going to be the volunteers. Most of the time. Sometimes they will get paid as well, right? Would you volunteer to be a subject in a clinical trial? What if it mean to you receive a treatment for a disease that wasn't available to anyone else? Very, very important thing. Most of the people, there are some of the contractors, there are some of the people whose main thing is, whose major income is to be part of these clinical trials. Some of them will do generously, but most of them will get the money for it, and that's the reason they have these trials. So the phase one, smaller group, maximum ranging from 20 to 80. The main intention is going to evaluate the safety of the particular drug, and number two is to evaluate your side effects. Phase number two. Now we are going to expand the range, right? The drug is working fine, or at least it is giving the good results on the smaller group, right? So why can't we expand the group, right? But while expanding the group, we consider a lot of other factors as well. We go with the different age groups, we go with the different genders, we go with the different people from the different continents to make sure it is working for all of them. Again, safety is the most important thing here. Next to the safety, we are going to test here the effectiveness. The efficiency we are going to determine here. Because we know that it is safe for the animals, it is safe for the smaller group of the people who are human beings already, but still, I consider the evaluation now. I'm going to extend my evaluation now to the different group of people. Those groups can be based on the age. Those groups can be based on the gender. Those groups can be based on anything else. But again, we're expanding the range. That is nothing but yet phase 2. Now, the phase 3, right? So when it comes to phase 3, we are going to expand the range to a lot. I can say that almost it is going to be 10 to 15 times of, minimum 10 to 15 times of your phase 2, right? Which we expand our range from 300 to 3,000 people. Here, the major objective is to find the effectiveness, right? I'm just checking the effectiveness compared with standard or equivalent treatments. What do you mean by the standard or equivalent treatment? There is already an existing treatment available. There is already one drug available to cure this particular disease in the market already. People are using it, right? So when I compare with that particular drug, how efficient my new drug is. When I compare with the existing drug, do I have any side effects? When I compare with that, let's say when I use the existing drug, I'm having some dizziness. The dizziness is the side effect of having the particular drug. When I go with the new drug that I'm developing now, even here, do I have the dizziness? If the dizziness is not there, then we say that compared to the previous drug, our drug is good because we don't have side effects now. Let's say that dizziness is not there, but it is causing some swelling. It is making it is producing some rashes, right? Or some other treatments that I need to take care of. Something is not good with my drug. So this in phase 3, we extend the group to a larger number. Then we're talking about the effectiveness of this particular drug, and we are also talking about the side effects compared to the existing drug, which is already present in the market, right? So we already have a benchmark. We need to overcome that, or at least we need to match that. Clear? All of you are clear up to this? Right? There are a few more trials here, but I don't go into that particular one. I'll give a pass here, and I'll tell now where SAS is required and why SAS is required in this entire clinical trials. All right. Let's say that. So we have done a lot of research and development. Let's say this is company A. After a lot of research and development, we come up with a drug A. So this drug, I did it on the animals, that is preclinical, phase 1, small group of people, phase 2, extended group of people, phase 3, a larger group. All the 3 phases are done. And I'm sure that this drug is really doing the wonders. Can I release in the market directly? Yes or no? Guys, can I release it in the market directly? Okay. What needs to be done? What needs to be done? We need someone. Am I correct? All we need is an approval. To approve, someone needs to approve it, and the someone is nothing but your FDA. Right? We'll talk about all the acronyms later, not an issue. Right? Just to understand that there is some monitoring unit at the top in each country. That particular country's that monitoring unit has to approve this. Correct? Only after the approval, I can release this particular drug into the market. That I can be done. But before dropping this particular drug into the market, I need FDA's approval. I'm talking about the US market. I'm talking about FDA. Okay. Let's say that someone has sent this particular drug A to the FDA. Similarly, there is another company. Let's say consider COVID itself. So what do we have? We have Covaxin, we have Covishield, right? And we also have the, what is the other one? Sputnik, right? And the new drugs are keep on coming, new vaccines are keep on coming. So let's say that there is a company A and company B. So drug A and drug B, there are two drugs which are produced by two different companies. Both of them are going to cure the same disease. Let's say there's a third company as well. So how do, when I receive drug A, drug B, drug C, all the three drugs are received to the are came for the FDA's approval, right? So every day hundreds of drugs will come for the approval in the form of a report, correct? I'm going to generate one report, and that report I'm going to use it for the evaluation purpose. Let's say I sent it in an Excel. Means I belongs to the company A. I sent all the details related to the drug A in Excel sheet. There is a company B, they send it in some XML format. Company C, they send it in a HTML format. All of them are going to have the very good information. The information is really good, right? They have all the information which are related to that particular drug. But how easy for me or how difficult for me being under FDA to evaluate them? Because they're not following any same format. Each individual is sending in a different format. So can I evaluate them on regular basis? Difficult, right? I don't have a standard. Until and unless I have a standard, right, it becomes a headache for me to evaluate them. So there need to be a standard. There need to be a standard guidelines, right? Let's say that all of them are sending all these in a Excel format. Let's say that. I'm just telling you. Again, in the Excel, when being a drug A, all the drug A details, let's say that I'm saying that I'm using the term called gender to represent male and female, right? I'm saying that gender is equal to NALE, FEMALE. I'm writing in a full form. When I search for gender in the other company document, Excel sheet, I don't find gender there. Instead, they mention it as sex. So both of them are representing the same thing, but my representation is gender, other company's representation is sex. So there is difference in variable indication. There is a difference in how we are sharing the information. So I have standardized the format, but I also need to standardize the parameters. Each parameter must be in a standard way, correct? Let's say that we have given the headers. Hey, anyone want to represent male or female? You should be use the name called gender only. The parameter name must be gender. Okay, fine, done. Again, when I see that company A has given gender as M and F. Company B has given the details as male and female. Company C has given the details as 0 and 1. 0 means males, 1 means females, right? According to them, each of them are correct. But again, when it comes to the FDA, when they are evaluating, should they need to consider FEMALE or should they need to consider F or they should need to consider 1? All three are three different values for the same parameter. So again, we need to come to a conclusion that we also need to standardize the values. It's not just about the parameters. It's not just about the file format. It is also about the standardization of values. Gender means everyone has to follow either F or M. That's it. Dot. Clear? So someone has to give the standard implementation guide. Hey, anyone want to generate this particular report and give it to me? You need to give me in this particular standard format only. That format is nothing but your SDTM. So what they said that they have given some standard implementation guide. This is implementation guide. Everyone has to follow this. Only by using this particular guidelines you need to send the report to me. To produce that particular report, right, by having all the SDTM implementation guidelines, based on that, I want to produce a report. To produce that particular report, we are using SAS. Because to produce this, we need to work on so many data sets. There are lots and lots of data sets that are available. I need to do a lot of analysis. I need to do a lot of reporting. To do all this, I need a technology, and that technology is nothing but your SAS. So the role of SAS here is, by strictly following your implementation guide, generate a report which we will send it to the FDA. FDA is going to evaluate that particular one. Either they are going to approve it or they are going to. Do you understand now where we are and what we are doing? Can I have a quick confirmation from everyone? All right. So here I just have given the same details. You can see that SDTM stands for Standard Data Tabulation Model. We're also going to discuss about ADaM, which is nothing but Analysis Data Model, the next level of your SDTM. And then we also talk about TLF, which are nothing but tables or listing figures. All these three are going to be covered under your clinical trials. Again, you can see phase 1, phase 2, phase 3, all these things. I just told you drug A, drug B, drug C, 4 different companies, the same thing I just mentioned here. So now, to make sure I'm going to standardize it, I'm going to use the standard implementation guide, right? So who is going to do this? Who is going to monitor this? Who provide the standard implementation guide? CDISC, Clinical Data Interchange and Standard Consortium. These are the guys. CDISC org. Even if you Google it, you're going to get it. A simple thing, cdisc.org. This is the organization who are going to use or who are going to maintain these standards, right? These are the ones who is implementing, who is monitoring, who is doing all these SDTM things. Standard, what is this? Standard Data Tabulation Model. This implementation guide is actually maintaining, monitoring, producing, all these things will be taken care by your students. All right. Now, so we know where SAS fit into. What is called your SDTM is. We haven't deal too much about the SDTM because we are going to start discussing about that. Today's discussion is only to understand what is called clinical trials, where SAS fit into, and what is called your SDTM on a very basic or low level. From tomorrow onwards, what we are going to do is we start discussing about SDTM. The current version is going to be 3.2. That is the latest guidelines. Every time there is any changes in the guideline, we are going to get a new version. The latest implementation guide is 3.2. Based on the 3.2, we are going to have a discussion, right? Every day we are going to discuss about what is this SDTM, what are the various domains we have. For each domain, we are going to understand all the variables. For that particular one, we are going to start parallelly, we are going to working on the practicals. All right. Remember, guys, clinical SAS.
Before SDTM, you are not going to explain about raw data. Quick
Raw data
explanation
is
about
nothing.
raw data.
Raw data is all about your data, right? What data sets that we are using and all. Is that what you are talking about?
Yes.
Raw data is. See, the raw data I start introduce to you when I start giving the practicals. At least one domain I'll explain first. But that particular domain, what are the data sets that are required? That raw data I'm going to explain, and then I can start giving you the practical stuff on that. All right. So without knowing what is the standard implementation, why do we need to understand about the raw data? Raw data is all about the trials that are already conducted and the readings are recorded, right? So those we go for each of the different domain, we go separate. Questions. So there are certain questions in the chat box. What days we are going to have the class? Generally, it is Monday to Friday, 1.5 hours. That is how it is. Maybe I'll see to that and I'll confirm on it. But now just consider that it is going to be a daily class, 1 hour class. Maybe next week onwards, I'll tell you if either it is going to be an alternate day for 1.5 hours or it is going to be every day 1 hour. I need to confirm that. I'll check and I'll confirm. Okay. All right. Any other questions here? All right. If there are any questions, I'm happy to answer. If there are no questions, we can wind up the session for today. Vamsi Krishna, videos, you can just have a word with Bali sir. Bali sir can help you with the videos. The recording will be available after 2 hours from now, so he will upload into the respective drives. We can get the confirmation from Bali sir. Yes, class will be on the same time. It can be either 8 to 9 or 8 to 9:30. That alone we need to discuss. I'll give you the confirmation for this week. Tomorrow and day after tomorrow also, it is going to be 8 to 9, 8 p.m. to 9 p.m. IS. All right. Thank you, everyone. See you tomorrow. Take care. Bye. Hey, hello, everyone. Good evening. Welcome. Hope all of
Good
you are
evening.
able to see my screen. Good evening. Hope all of you are able to see my screen and hope all of you can hear me. Can I have a quick
Yes.
confirmation? Perfect. Thank you. All right. Just give me a moment. I don't see. Okay, all of them are from yesterday's session. Anyone, any new joiners today, or all from yesterday's class? Can I
Yeah,
have?
myself, Mani Maran. Yesterday I missed your this session.
Okay,
Today I have joined, but through the video I have seen your session. Up
perfect.
to
That's
50 minutes I
yeah.
have closed. Yeah, fine.
Okay, fine. Anyways, yesterday's session was just a very basic introduction to the clinical trials. We haven't entered into anything that need to be taken very seriously, right? We just started, a first baby step, I can say. All right. So today, what we do is I'll have a quick recap about what we have done yesterday, maybe 5 to 10 minutes, and then we go with elaborative clinical trial, and then we set up the base for what we are going to learn for the next 30 days. That is next 30 classes. All right. So let's start with what we have done yesterday. First of all, we all know that we are talking about clinical SAS, right? Again, when I say clinical SAS, it is more of domain. That is, we need to know how the clinical trials work. And then we need to understand where SAS fit into this particular one and how SAS is solving the complex scenarios and how SAS is providing the resolutions to the given problems or given problem scenarios. This is what we need to understand. This is what we are going to do for the next 30 classes, right? So it is a right mix and match of the domain plus the technology. So we started with the clinical trials. We understand that clinical trials is all about from the time we start developing the drug. We are going to develop a drug, correct? We means the pharmaceutical industries. So the moment we start developing drug to till it got approval from your FDA, until it reaches the market and reaches everyone. Till that particular one, what exactly happening in the background, we are going to discuss. Correct? Yesterday I have given few details. So what we have started, we said that the drug that we are talking here is nothing but it can be a medical drug which can be used to treat the people, right? It can be a preventive drug kind of a vaccine, or it can be a medical equipment. Any of these will fall under the new developments. Correct? What are them? I said the first thing is going to be the drug which can be used for the treatment, any disease treatment, or the drug that we are going to use as a preventive measure or it can be a medical equipment or medical device, simply I can say it. Any of this without approval or without going through a process, it will not come to the market. And that specific process we are calling as a clinical trial. And this clinical trial, if I put it in a proper way, it has certain steps. Let me tell you what are the steps are. So we haven't discussed this in detail yesterday, but we are going to start with this particular thing now. So the entire clinical trial starts with your sponsor. Remember, these terminologies are very, 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 very important. So that's the reason you guys need to concentrate more on understanding this particular terminology, right? So first we are going to have a sponsor. The sponsor is the one who is going to fund your project, or who is going to provide all the funding which is required to develop the drug, right? Generally, these sponsors are none other than your pharmaceutical companies. All, or not all, I can say the people who are into the research and development, all the pharma companies are nothing but your sponsors. So your drug development ideally starts with the pharma. So here is what the pharma guys comes to the picture. So these are the drug companies. These are nothing but the sponsors. So the sponsor is the guy who is going to provide the funding for you. Once the funds are available, then we are going to enter into the second phase, or we are going to enter into a facility, and that facility is none other than your research and development facility. Correct? So most of the organizations are going to have this particular one. We can call it as a research and development facility. This is the facility where your drug is actually going to develop, right? But there are few exclusions as well. It is need not to be your R&D department all the time. It can be some other things as well. When I say some other things, those some other things are nothing but it can be of your university, the medical university. It can be of your a normal community clinic, or it can be under the supervisor, or I can say that the doctor itself, the premium doctor itself, even under the doctor supervision also it can happen. But majority of the times, the drug development will happen under the R&D facility of the bigger company, I can say that, or a well-known company, I can say. All right. So let's say that the drug is developed. So they have developed some drug. So what's the next step? This, whatever the drug they have developed, they need to get tested. Without a proper testing, without ensuring the safety and efficacy of that particular drug, it cannot come to the market. So here the clinical trials are going to start. So up to this is only a smaller picture, correct? We haven't actually entered into the clinical trials yet. Here we got the sponsor who is ready to supply the funds. We have the R&D facility. The guys are really good in doing the research and development, and they are able to develop a drug now. But the drug need to be tested. There are hundreds of drugs will be developed on day-to-day basis, but all these drugs will not come to the market. If someone asks me what is going to be the general range, or what is going to be the span where this particular drug is developed, it is generally 2 to 8 years. In general, I'm talking about. There are drugs which will take more than that as well, right? If you consider COVID, for COVID, we were able to develop the drug within the span of 1 to 2 years, right? There are few drugs that say that if you consider AIDS, it is, we all aware of this particular thing from quite a long time, but still there is no proper drug for it. Correct? There is no proper treatment for it. There is continuous development. There is continuous research happening about that. Correct? So the minimum years we generally consider as 2. The maximum we can consider as 8. But there are some exclusions as well. Those exclusions are nothing but outliers. Right? Those are not fitting to our regular span. But in general, it will take 2 to 8 years. Okay. Now the drug is developed. So drug is ready. So now we will enter into the clinical trials. The very first clinical trial or the initial clinical trial will not happen on the human beings. Instead, it will happen only on the guys, can you hear me now? There was a power issue. The internet got reconnected. Guys, can you
Yes,
hear me now?
yes.
Yeah, yeah. Okay, thank you. So nothing I have done in the meanwhile. All I have done is we just have completed our all right. We just have developed the drug, and from the drug, we are entering into the very first step of the clinical trials. And the very first of clinical trials is nothing but yeah, preclinical trials. Generally, we can call it as a preclinical trials. The preclinical trials is generally carried out on the animals, right? It is purely going to conduct only on the animals, right? The reason why we do this is we want to understand the mechanism of the drug. We want to understand whether the drug is actually working on this or not. Number three, main thing, we want to see the safety. We want to see the safety. By considering these three factors, we generally carry out our clinical trials on the animals. The major outcome that we are expecting here is nothing but the major outcome is safety. Once we understand that, yes, whatever the drug that we have developed is delivering the results on the animals, at least it is getting responded. It is not giving much of the side effects. Then we will enter into the main phase, and that main phase is nothing but the clinical trials phase, and it has 4 different steps in that. Remember, so the clinical trials part has 4 different steps. First one, you can call it as phase 1. Once the phase 1 is successful, we will enter into phase 2. Even phase 2 is successful, then we will move ahead to the phase 3. Once we decide that phase 3 also successful, I'll tell you what and all various things we are going to discuss here, or various things we are going to check here. Once phase 3 also successful, finally we will enter into phase 4. So totally the cycle is going to be phase 1, phase 2, phase 3, and phase 4. Yesterday also we had a little discussion on this, but we haven't go in detail. Today we are going in detail discussion about what we are going to have in each phase, what are the various subjects that we are going to consider for each of this particular phase. Remember, in the phase 1, do you have any idea, guys, who participates under the phase 1? Since we discussed this yesterday, can someone please tell me?
Healthy volunteers.
Healthy volunteers. Perfect. So it generally carried only on a smaller group, We generally say that 30 to 80 people. And remember, we consider only healthy subjects. Remember, hereafter, we don't use the term called person, or we don't use the term called their patients. Instead, wherever we are going to refer someone who is part of this particular clinical trial, we call them as a subject. So we try to adopt the terminology. Remember, we are going to consider only the healthy people, nothing but healthy subjects, right? So the main interest here is going to be on a healthy people. You're going to test your drug and you're primarily look at the safety. You don't look at the efficiency, you don't look at any other thing. The only thing which I'm bothering about here is nothing but, is my drug safe enough or not? Once you get to a conclusion that the drug is 100% safe, then you will enter into the second phase, the phase 2. The phase 2 is, any idea who and all we are going to test under phase 2? Anyone?
Here it starts with patients.
Wonderful.
Contractors.
Contractors. Okay. Here also, these healthy subjects also can be contractors. Correct? There may not be volunteers all the time. They can be contractors as well. So what else? Only patients and contractors. So can I say that contractors, some of these contractors also patients? Can I consider that? Or how do you want to proceed with this?
You can consider that.
Perfect. Okay, let me tell you. So here we are going to take 3 different types of subjects. Number 1, of course, it is going to be patients, but patients of the same disease. What do you mean by same disease? Anyone? I have developed this particular drug to treat a specific disease, correct? The drug has been developed to treat a specific disease, right? Let's say the disease that I'm talking about here is going to be something called, maybe just consider as a fever. Fever is the disease. To treat the fever, we have developed a drug, right? So now my first set of patients are going to be the people or the patients who are already suffering from fever, who are already having high temperatures, right? The people are the patients with or patients suffering with the same disease, suffering with the disease. What disease it is? The disease to which we have developed this particular drug. That is the first set of people. Number 2, the second set of people are going to be again patients, but suffering with a different disease. They are not fall under this particular one, right? Suffering with a different disease. But you can ask me, Murli, can I go with the same? What do you mean by different disease? They may not be, suffering with a fever. Let's say that they have the body ache, right? They are having some allergy. They are having cold. They are having cough. A similar, I cannot say that exactly the same disease or exactly the opposite disease, but the diseases which I can consider as a subclassification are the diseases which have the same prawn as your current disease. All those patients also I'm going to consider here, right? Meaning this one, the second set of people have high chances of getting affected with this particular disease, or the second set of people are maybe because of the tolerance levels or maybe because of their immunity levels, they might not get the fever yet, but they have all the chances to get the fever, the related one. That also we are going to consider here. But 3, we also consider healthy subjects. So we have considered healthy subjects in the first one, right? So the same healthy subjects we are going to use here. We are going to test the drug on the healthy subjects, patients suffering with the same disease, patients suffering with a different disease. All these three, the cluster, we are going to consider for phase 2. And the phase 2, we are going to increase the number of people, that is the group size we are going to increase. It can be any number from 100 to 500 people. Some books will refer different number, but altogether it is fine. Either it can, some books refer as 100 to 300, some books refer as 100 to 500, but that is okay. We can consider that it is lesser than 1,000. Anything more than this 80, I can say 100. Anything lesser than 1,000 is a comfortable number. Now, what is the focus here then? The primary focus here is all about safety, right? But safety is not a major thing here. I can say that the focus is on efficacy. How efficient the given drug to treat the given disease. Is it efficient or not? Second, if I say efficiency, the efficiency I'm going to measure on two different things. Number one, how much time it is taking, that is the span it is taking to get treated. How many doses that I need to provide? Is it, can I say that I need to take this medicine every 6 hours, or I need to take this medicine 3 times a day, 2 times a day, or only at night time? How I supposed to give the dosage to get the better? So the efficacy is the primary thing. Along with that, I also check the side effects. Is this drug, because of this particular drug, because I'm looking at a different cluster here, there are group of people, different set of people there. So I also need to check about whether this particular drug having any side effects, right? While it is providing, while it is treating the given disease, is it causing anything else? Let's say that, is there any itching, or is it causing something called dryness in your eyes, or is it causing dizziness? If it is not giving that particular thing to the people who are suffering from fever, but when I test this particular drug, or when I give this particular drug to the people who are suffering from the body ache and all, so how they are responding? Is the particular drug is actually working on them or not? If it doesn't have any effect, we can say that we know we need to give this particular drug only to treat the fever, but not to anything else. I'll tell you one of the best example. We all know Dolo, right? The most, I can say that the tablet which is consumed by the most of the people in India during this particular COVID time, right? It has been used to treat your COVID. It has been used to treat the fever. It can be used to treat your body pains. It can be used to treat your whatnot. Correct? A simple paracetamol with 650 mg of the dosage, people are using for anything. And there are some memes saying that it is equal to your polo. Correct? So, is the drug that has been developed is targeted only to that particular disease, or it is having the capacity to also deal with a different set of diseases or not? That also will be tested under this. You can ask me now, only why we are testing this on healthy subjects? Why? What is the need? Anyone? Can someone answer this? Because we have already did on the phase 1 on healthy subjects, right? But here again, why do you want to do this on the healthy subjects again? Why? Anyone?
For detecting side effects.
Exactly. Even Mohanty, yes, you're right. Dosage adjustment. Both of them. How much dosage? Because if you see that you are in the early stage, let's say that the fever level, the temperatures are going to be very less. Let's say that it is somewhere around 100 to 102, right? What might be the dosage? That is again falls under the patients with the suffering with the same disease. There I can use this for adjusting the dosage. But when it comes to the healthy subjects, this primarily, let's say that this particular guy doesn't have a fever. He's not suffering from fever at all. But what happened if this guy takes this drug as a preventive measure, right? As a preventive measure, what happened to him? or what happened to her. That can be done under the healthy subjects, mainly to check what are the side effects that we are going to get if I go with the healthy subjects. All right, so that is about your phase 2. Then when I enter into the phase 3, here this is the most crucial thing. I can say that even though the marketing and everything will happen in the phase 4, but phase 3 is the most crucial in the clinical trials. Why it is crucial? Because we are expanding our base drastically, anywhere from 1,000 to 5,000 people. Here again we are considering a random set of people. Here we are considering the healthy subjects. The same 3 things are going to repeat, but when it comes to the healthy subjects, when it comes to the patients with suffering with the same disease or with a different disease, here we are going to consider a lot of other factors as well. Meaning we are going to consider the gender, because some drugs, even though the disease is going to be the same, for gender it is going to be different, right? So when we are going to have this particular drug, the doctor is going to treat differently for female, and the doctor treatment is going to be different for the male. Even though the disease is going to be the same, there are some specific diseases, correct? Because our body couldn't tolerate few of the drugs, right? So that is the reason we need to consider the various gender people as well. We need to take the mixed group of both male and female, right? Let's say that up to this, we are concentrating only on the male subjects or only on the female subjects, depending upon the type of the drug. But the moment you enter into the phase 3, you're going to test it on both the genders, just to see what happens if I give this particular drug to the other gender. What happens? Because side effects we need to see. Second, some drugs, the dosage we need to decide, because here my focus is on the safety. What happens if I give to the other gender? At the same time, I need to check the efficacy here. At the same time, I also need to check the side effects here. All 3 are major things. If you see the second one, I'm not going to bother much about the safety, because safety is already tested in the phase 1. But here, since I extended my base to the large people, because I'm talking about various genders, we are talking about various age groups. The age groups are also going to vary, because we're going to consider all the age groups, right from your, let's say, from the toddler to till your old age people. And we are going to consider the various continents as well, because based on the continent also these will be changed, right? What not? There are so many other factors we are going to consider. What are those factors? What are those parameters? Again, we are going to discuss in the later part. But on a whole, right now, all we are going to concentrate here is we primarily focus on the various parameters while selecting the people. We also use some of the techniques here called randomization and all, but it is too early to discuss about that. So we'll discuss about the randomization and all. How do we take random people and all later? All right, but the primary focus is on safety, efficacy, as well as the side effects. Once we are through with this particular one, we are ready to launch our particular drug to the market. So here, phase 4 will start. Phase 4 is everything. Diverse subjects, yes, true. Maybe the right technical word, if I say Maithili, yeah, I can say that it is going to be test on diverse subjects. Wonderful. Because we are going to consider everyone here, right? We are going to consider everyone here. All right. Now, the next thing that I'm going to talk is the phase 4 is one step before you enter into the market, just one step. So here we are going to extend our range from 10,000 to 50,000 people. A lot, right? But here the major thing that we need to understand here is a lot of factors come to the picture. A lot of factors, meaning you have given, the sponsor has done a lot of funding so far. They have given a lot of money to the R&D facility, They have provided a lot of money to get your contractors, get your volunteers. Sometimes if the volunteers are not there, they need to pick the people from the specific vendor group. He or she have done a lot of work here. They have put a lot of effort to bring this particular drug. So there is a lot of capital involved there. Correct? According to this particular capital, they need to fix the price. They need to fix the dosage. They need to fix the stock, how much volume, because if I'm ready to give this particular one, if you remember during the COVID time, even if you take Bharat Pharma or anything, before even we get the approval from the government, right, they have produced, let's say 1 million stock. I already have 1 million doses with me before even we go with the approval, because once the approval comes, we should be able to provide the minimum number of dosages to the cluster of people. So the capital, the price, the store, the dosage, the stock, whatnot, even the complete the marketing strategy, all my marketing strategies as well will be decided here. This is just one final step before we enter into the market. So here we don't talk about the safety, efficacy, side effects, because it's all done. Here only we talk about the external terms, which are nothing but what is my capital I have invested so far. So what might be the price for my given medicine? What might be the dosage for each of the age group, each of the gender, or each of the all the diverse subjects? Right? How much stock I should have before, at least if I enter into the market, what might be the minimum dosage? Because if a doctor, prescribe this particular medicine to a patient, let's say that for the set of patient, let's say each patient he suggests that you need to take this medicine 3 times a day for a month of time. So one patient itself is going to buy 90. That is nothing but approximately 100 tablets he is going to purchase, right? So let's say that if I'm giving this to a particular country or to a particular state, I need to release with minimum number of stock. So the stock need to be maintained here. Post that the marketing strategy, you need to approach the right facilities, you need to approach the right doctors to make sure your drug is part of their prescription. So all those marketing strategies also part of your piece number 4. So with this, with the stage number 4, ADRs in target population. True, but Maithili, I'm not going too much into it because I know that you sounds really good with your functional things, but I'm not going that deeper because I'm looking at the logic, right? Let's use all this terminology a little later. Perfect. Now, once we are done with phase 1, 2, 3, 4, we are ready for this particular drug. But if I want to release this particular drug to the market, I need an approval. Am I correct? Without an approval, I cannot do. So to whom I need to approve, or to whom I need to reach for the approvals? Anyone? Without an approval, I cannot do. So who are going to be the approvers? Can someone please tell me?
FDA.
FDA. Okay.
Drug regulatory authorities.
Drug regulatory.
Authorities.
Regulatory authorities. Okay.
TCG.
MHRA for Europe.
Someone said DC.
MHRA.
DCG.
Someone mentioned
That is
European countries. EMA.
DCGI. That is
DCG. Sorry. I'm just
CDISC.
typing what you are saying.
I.
Okay, there are a lot of things are happening. Okay, yeah. We'll go with the lighter note. Let's consider 3 different markets for now, because most of this started from the USA. So for USA, we have FDA. What is FDA? Food and Drug
Administration.
Administration. For Europe,
MHRA.
MHRA is there. There is another authority as well. We can simply call it as a EMA as well. Yeah. What is EMA? Anyone?
European Medicine.
A for authorities. You can also consider as authorities or agencies. Okay. For India,
Drug Control General of India. DCGI.
DCGI. correct? So we have drug control governance in India. We have for Europe, we have European medical agencies or medical authorities. For USA, we have Food and Drug Administration. So these are the regulatory authorities, right? So there are different authorities as well. Primarily, we consider these, right? Majorly, most of the times we follow FDA, right? Okay. So if I want to go to these approvals, what I need to do? To get the approvals, I need to prepare some reports, correct? I'll say that this is my report. These are the various subjects that I have considered. These are the dosages that I have provided. These subjects are suffering with these set of diseases, main disease, primary disease, as well as the child diseases of that. And after I use this particular drug, it got treated in these many number of days. That is nothing but the span. After recovery also, what are the various precautions that we need to take? And finally, if any side effects, we need to mention that. Correct? If you enter any drug, if you just, let's say, even if you put Dolo 650 in your Google, if you go to a 1mg site or any site, it is going to talk about what this drug is, what is going to be the composition of this particular drug, why we use this particular drug, what are the side effects. All these 4 parts will be there, correct? Even the dosage information will be there. So all these are supposed to be part of your report. I'm telling on a very lighter note, right? Generally, we don't follow that, but on a very lighter note, these are going to be the various reports we have. All these reports, we are going to do it in SAS. To prepare this particular report, we are going to use SAS. Where SAS is coming to the picture in the clinical trials means while we produce our reports to the FDA or to the regulating authorities to get the approvals, we need a report. That particular report, we are going to do it in the SAS. Fine. Murli, you're saying that SAS, but SAS is somewhere, you know, it got developed in 1960. We know all the things and all. Let's say before SAS, before using the SAS, are we doing on any other technologies? Yes. Before we standardize this particular process, before FDA standardized this particular process, everyone was using their own way of representing and their own way of producing the results to the FDA. No one is going to follow a specific format, right? For an example, right here I have the approvals part. So before I enter into this particular approvals part, everyone was following a different set of reports. For an example, there is a company one, right? So they have produced one drug. Let's say we can call it as a drug one, and this particular drug one is to treat a specific disease. So they have generated a report, and this report they have submitted to FDA. Similarly, to treat the same disease, there is another company, they also produce the drug. All right. So when they produce the drug, it is going to be drug 2, and they have generated their report on their way. Right. Similarly, drug 4. Now there are 4 different reports for 4 different drugs, but all of them are going to treat the same disease, and FDA has received all these 4 reports. Now, FDA need to go through each of the report, and they need to provide their approvals, or they need to say whether it is approved or rejected. Though, let's say that report 1 is prepared in an Excel, a report 2 is prepared in Word, a report 3 is part of PDF, a report 4 is in Excel. Because FDA have not asked for any specific format, so each company is producing their own report. So the first thing is, because it is difficult, if I want to compare an Excel with Word or Word with a PDF or PDF with XML, it is quite difficult, correct? So I need a specific format to compare the results as well, just to see which company is having the better track, correct? Because the parameters are going to be the same. So what they have done, they first standardized, because I need to have some standardization here. I need to place some standards. So to come up with the standards, because there is a need for coming up with the standards. The standards include the very basic stuff, which is nothing but the format. In which format you need to prepare a report. First thing. Number two, once the format is fixed, let's say that everyone is sending their report in an Excel, right? So one guy, when they say about the continent, he used a term called continent. The company too, they use it as a region. So continent here they mentioned as Asia, but when it comes to the second one, they use the same Asia, but they don't call it as a continent. Instead, they use it as they call it as a region, right? There is a third person, they simply call it as an area, but all of them are referring the same thing. Let's say all the three people are talking about the continent only, but the first company used the word called continent, that variable name. The second guy used region, the third guy used area. So there need to be some set of standardization when it comes to the parameters, correct? What parameters to be part of your report? Again here, report number 1 has 100 parameters, 100 different things he have considered, and he presented the report. Whereas my the report number 2 will have only 50. Whereas my report number 3 will have only 15 parameters, which are critical to that particular one, right? So FDA also has given the details about every report that you are going to produce to me. The report should have some mandatory parameters. This should be common for all of the reports. You may have some additional parameters that may not be mandatory to me, but I'm specifying that these are the various parameters you should follow. That is the number. At the same time, what are going to be the parameters? Those are critical parameters, or those are going to be the normal parameters. Both of them also I'm setting up here. I means someone is setting up this. Who is that someone? We are going to talk. Number 3, once the parameters also specified, now we are talking about the values. Let's say someone asked to mention country details. I'm typing as India. Someone saying IN. Someone saying IND. All three are valid. For me, country is India. For someone, the country, they are just using an acronym IN. Some are using simply IND. All three are possible. All three are correct only. But there need to be some set of standardization, right? If someone says that country should be of 3 letters, then you have to use IND everywhere. Someone has to mention that it is going to be 3 letters, then everywhere it is going to be 3 letters only. It should not be elaborative one. It should not be a short one. It's supposed to be 3 letters everywhere. Correct? So standardization should be happen even to the values. Like this, there are a lot of parameters which are required. Mainly those are required to do one thing, that is globalization, right? I need to globalize my standards so that whatever the report that I am getting, all the reports, I can quickly scan it, I can quickly do the approvals, or quickly I can do the rejections. All these 3 I can do because I know the format, I know how to validate. Even if I want to provide some information to the other person who want to validate it, it is going to be quite easy for me because it is under the standards that I have already set up. So to set up this global standards, FDA has come up with one organization, and that organization is nothing but CDISC. Any idea what is called CDISC? Can someone expand this particular acronym? Sorry? Anyone?
Clinical data interchange standards consortium.
Yes, exactly.
Clinical
So,
data interchange standards consortium.
exactly. Yes. You guys are correct. CDISC is the main standard organization. This organization is actually funded. It is actually started with the help of your FDA. FDA is the one who started this particular one. The acronym stands for CDISC is going to provide end-to-end global standards, right? It called as Clinical Data Interchange Standards Consortium, right? So this particular one is started somewhere in the 1998, right? So they are going to provide, it is a very open thing. You need to say that it is a open organization, and anyone who want to know the standards, they can simply visit cdisc.org website, and they can download an implementation guide. That is nothing but it will be called it as SDTM implementation guide. So maybe I need to elaborate few things here. This CDISC, after they finalize all the global standards for the clinical trials, they have given 3 sets, or I can say that 3 different standards. Number 1, SDTM. Number 2, ADaM. Number 3, TLF. Any idea about this? Anyone? Maybe basic idea about what is called SDTM and what is called your ADaM and what is your TLF? These are the
Study
different.
data tabulation module.
Yeah, you can also call it as, either you can consider it as a study data, or you can also call it as a standard data. Standard, or you can also call it as study. Some books refer standard, some book refers as a study. Standard data tabulation module. ADM stands for
Analysis data.
analytical or analysis data models. Right? Okay. TLF stands for tables, or
listings, figures.
listing and figures. So in our course, we are going to start with SDTM, and then we will enter into ADaM, and also we are going to cover TLF. All the 3 things we are going to cover in the course. All right. So these 3 can be considered as 3 different standards, or 3 different standards that we are going to follow to generate the report to send it to the FDA approval. Every report that is going to receive by your FDA has to follow this particular one and has to be in the same format. All right. For SDTM, currently we have 3.3 version. There are various versions as well, but most of, I can say that 90% of the pharma industry is still under 3.2. The implementation guide, which can be considered as a Bible. So the standard data tabulation model implementation guide, which is going to be 3.2. We are also going to use 3.2 version. And when it comes to the ADaM currently at 1.1 version. For TLF, there is no specific thing. There is no specific version and all, but we'll simply go with it. What are the various tables, listings, figures that are required? What are the supporting SAS procedures for that? We are going to discuss. Questions? Any questions on this? Anyone? Right. So the next thing I would like to talk here is, this is another thing, because I'm also going to provide all these notes to you guys. No worries about it. So today is only the initial thing. This particular one doesn't require much of the notes, but anyways, I'll save this particular Excel. And whatever I have discussed so far, the basic details I have already provided here. Maybe I can share this with you guys as well. And also, maybe I'll start with this particular one, a very small topic about how the clinical investigation start, what is going to be the protocol. Remember, irrespective of whatever the clinical trial you talk about, all these clinical trials, all these clinical investigations begins with the protocol. The protocol is nothing but it's a document describes about how the trial is supposed to be conducted. How the clinical trial is supposed to be conducted. You can see that these are various steps I have mentioned here, right? How these steps need to be started, how we are going to carry out this particular one, how to conduct this particular one, meaning the objective of this particular clinical trial, what is going to be the design, what is going to be the methodology that you want to follow, what are the statistical considerations that you want to do, and who is going to be the organization, which organization is going to start this. That is nothing but the sponsor, or it is going to be called it as, yeah, the person who is going to fund this. All I'm doing this just to ensure that the safety of the trial subjects, correct? Because you're doing this on the human beings, right? So when you're doing this on the human being, the first utmost important thing that I have to consider is the safety. How safe this particular clinical trial is going to be. Why you are doing this? What is going to be your design? What is the methodology that you are going to follow? What are the statistical considerations? That is nothing but your measures, right? All these I'm going to consider. Okay. What is that particular protocol document is going to describe? It is going to describe a lot of details. Can you see this? Title page, background, objective, study. There's a lot of stuff will be there. So our next topic into your clinical trials is going to be how your protocol document is going to be, why you are going with the protocol document, and what is the purpose of it. So before we start entering into your SDTM, that is going to be the entry phase for our clinical SAS. Before we go there, we need to understand few more things. That is nothing but we need to understand about what is the subject is all about, how we consider the subjects, and there are few terms and terminologies that we need to understand. It may take 1 or 2 days. I can say that minimum 2 days, 1 hour classes are required to enter into the SDTM. Once we enter into the SDTM, the story is going to be different. We are all going to talk about the raw data. Then we start discussing about the various data models and all, right? So before we enter into that particular one, we need to understand some clinical terminology. And the clinical terminology starts with your protocol. And this one we are going to discuss in the tomorrow session. So today's session you can consider as an elaborative session on the yesterday's one, because I can see that some of the new joiners as well. At the same time, I want to expand on each of the phase one. That's the reason I have done this here. So from tomorrow's session onwards, we are going to talk about the terminology. The main concentration is on the terminology. So we understand the terminology. Once we are good with the terminology, then we will enter into the SDTM phase. Clear, everyone? Can I have a quick confirmation? Any questions or doubts here? Please let me know. I'm happy to discuss. All right. If there are no other questions, we can wind up the session today here. And tomorrow we have a session, and in tomorrow's session we start discussing about the various terminologies that I can use it to before we step into the SDTM, ADaM, and TLM. Clear? These are the initial days, guys, before in a slower pace. Okay. Thanks, everyone. See you tomorrow. Take care.
Thank you.
Thanks.
Thank you, sir.
Can I have a quick confirmation, guys?
Yes.
Just a second. I have some issue with the headset. Just allow me a moment. Is it clear now, or still do you have any disturbance? Guys, is it audible now? Can someone please come?
Yes. Yes, it's audible.
Perfect. Okay.
Hi, sir.
All right. I don't know who is this. I don't have a full name here, but I can see T. First of all, so let's talk about the timing, guys. We haven't changed any timings. It is actually 8 p.m. IST. That is what we are doing, right? If anyone propose any changes, kindly let me know. Okay, first thing. Number two, coming to the how many days in a week we take the classes and all. This week I may take 3 classes. Maybe today and tomorrow I'll take. I'm not sure of Wednesday, Thursday, and Friday because I have some other commitments. I also informed the policy about it. But from next week onwards, it is going to be weekly 3-day sessions. That is Monday, Wednesday, and Friday. Every week 3 days, and each day 1 hour 30 minutes. So we generally start by 8 o'clock. The session timing is going to be 8 p.m. IST to 9:30 p.m. IST. This is from next week on. Is that okay, or do you want to have any changes? Can someone please comment on it? Yes, Monday, Wednesday, Friday. Yeah.
Okay.
This week, today and tomorrow, I'll take 1 hour of the class. That is 8 p.m. to 9 p.m. Today and tomorrow, I will take it. And the next class might be on the Friday, but I cannot give the confirmation because this week I have some commitment from Wednesday to Friday, right? But I'll give you the confirmation. But for sure, Wednesday and Thursday, I won't take the classes this week alone. All right? Yeah. Okay. Without wasting much time, let's start with our session. Last week, Monday, Tuesday, 1 hour this week. Yes. Probably we can have a class on Friday, but I cannot give the commitment right away. If I get a chance, I'll take the class, but I'll inform you on Thursday itself. And he also talked about the same time. And he also asked about the WhatsApp group. There are a bunch of WhatsApp groups because you guys are from the different batches. So that's the reason we are not sure is there a specific WhatsApp group or not, but you can have a word with Paulusar and Paulusar can help you with that. Clear? Any queries related to the WhatsApp group? Right? Just have a confirmation from Paulusar, ask him to add you into the same WhatsApp group so that we can discuss. All right. So let's start with what we have discussed in the previous class. Quickly, within 5 minutes, we can have a quick recap. So clinical trials is the one which we started with, because before we go with the clinical SAS programming, we want to understand the domain first. So we started with the clinical trials. So clinical trials, there are certain phases, right? So why we do clinical trials, irrespective of whether it is a treatment drug or it is going to be a vaccine, that is nothing but a preventive drug, or it is going to be a medical device, any medical device. Before it comes to the market, it needs to undergo some process. That process is nothing but a clinical trials. Why do we do this? We are doing this to ensure the safety and efficacy. These two are the two main reasons why we are going to the clinical trials. First and foremost thing is going to be, I need to ensure that whatever the device or the vaccine or the treatment, the drug that we are taking, that's supposed to be safe enough, that's supposed to be efficient. So to understand these two, primarily we go with the clinical trials. In addition to that, there are some other factors as well. Those factors are going to be the dosage information, how frequently we need to take this particular one, what is the mechanism, right? So what are the side effects? All these are going to be the sub factors, but the primary factors are going to be safety and efficacy. Clinical trials initially starts with preclinical trials. In a simple way, if I want to talk, preclinical trials is primarily done on or conduct on animals. Once we understand that this is risk-free, then we actually enter into the clinical trials. In the clinical trials, there are 4 phases. Phase number 1, it generally carry out on human beings only, and that too, we are going to conduct this only on the healthy people, right? Again, last week I haven't informed one thing. We consider healthy people not in all the cases. There are some exclusions here also. Consider there are cases like HIV, right? On a HIV, you cannot conduct your HIV trials on a healthy people. Here, I need to conduct this only on the normal people, only on the people who are already suffering with this particular disease. Guys, am I audible? Can someone please confirm?
Yeah,
Hey,
yes, sir.
Santosh, because I got a message from Santosh saying that he's not able to hear me. That's the reason I asked you. Okay, thank you for the confirmation. Just give me a second. Okay, done. Right. So consider the cases like HIV, cancer, and all. In these cases, I cannot conduct even the phase 1 trial on the healthy people. Instead, we will do it only on the people who are already suffering with the disease. It is generally carried out with a group of 10 to 30 people. Sometimes it can extend even to the 80 people. The primary reason why do we do this is going to be the safety. Safety is the primary reason, our primary outcome that we are expecting here. Then we have phase 2. What we are going to do under the phase 2, we will extend our group from 100 to 500 people. Here we are going to have the mix of the people. That is nothing but we go with the healthy people. We also take care of some patients. While considering the patients here, we consider the patients who are suffering with the same disease, and we also consider the patients who are suffering with the different diseases. Correct? So same disease as well as the other disease. Both the patients we are going to consider. At the same time, we are also going to consider the healthy subjects. Here, the primary reason, the primary thing that I am expecting is going to be efficacy. Then we enter into the phase 3. In the phase 3, the most important phase is always the phase 3. Here, we extend our base to 1,000 to 5,000 people. Here, we are going to consider all the ranges of the people, different groups, different genders, different age groups, different continents, healthy subjects, people suffering with the same disease, other diseases, whatnot. We are going to consider a lot of things here. And again, the primary reason of doing this is going to be to meet both of them, safety as well as efficacy. At the same time, along with these two, we also discuss about other factors which are nothing but the side effects, dosage, how frequently you need to take this. All these information we are going to do here. Then we go into the phase 4. Phase 4 is nothing but one step before launching this particular drug. Here we are going to test this on minimum of 10,000 people to the 50,000 people, and also at this particular stage we do the marketing, we decide the price of the particular drug, all the things, marketing strategies, stock, every piece of information we are going to maintain. That's going to be the end of your clinical trials. Once everything is approved, then we are going to prepare the reports, and these reports we are going to send it for the approval. Generally, we'll send this approval to the regulatory bodies. For USA, it is going to be FDA. For Europe, it is EMA. And when it comes to the India, it is going to be our drug control. Correct? So for the respective countries, we have the different regulatory bodies. To the regulatory bodies, we are going to send this. This is a simple piece of the clinical trials. And all this we have already discussed in the previous class. Correct? So what we do today, in every clinical trial, very, 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 very important. So where it will start, we are going to discuss today. Okay? Just give me a moment. Hope all of you are able to see my screen. So I'm just going ahead with a very simple introduction part we have already done. So I don't go with this particular one. Phase 1, phase 2, phase 3, all the things are there. I don't go with this particular one. So here, understand this particular piece, right? So what is the difference between a normal SAS programmer and a clinical SAS programmer? Can someone please differentiate this? Is there any difference between these two guys? What do you think? Anyone? Guys? Clinical SAS programmer apply their skills on the clinical research. Maybe we can extend the same to others as well. I can say that when it comes to banking professional, he or she will apply the clinical SAS skills to the SAS skill to the banking, SAS skill to the healthcare, SAS skills to the logistics. One day, what you said is absolutely correct, no doubt about it. No doubt about it. Maybe that we can fine tune a little. The simple difference is going to be, if you're a normal SAS programmer, let's say that I'm just working for one of the bank, let's say ABC Bank, right? Without knowing the banking terminology, let's say that I have something called secured loan. I don't know what is secured loan is. I don't know what is unsecured loan is. What do you mean by mortgage? I don't know, right? So what is called deposits? What are the various deposits are there, right? What is the difference between general insurance and life insurance? Or what is called Vickers, or what is called Silas, right? Or how your QR is going to work, or how your APIs are work, or how your PVDs works, right? How various marketplaces work. I don't know any of this. What is the usage of Adobe Analytics? Adobe Analytics. I don't know all this, but without knowing all these things, the moment I have the data set with me, or the moment I have the data with me, and when I have the problem statement, I can provide the meaningful solutions for that. I can do that. Because even without knowing this information, we can still work on banking industry. Of course, banking industry, again, if you go to something like AML, anti-money laundering, right? They definitely require the banking knowledge. Without knowing what the anti-money laundering schemas and all, you cannot work on it. Without knowing the KYC, you cannot work on the KYC. But other than these two, 90% of the cases, I can happily work on any of the bank product without having any domain knowledge. Right? Same goes to your healthcare also, right? As long as I have where the claims are coming from, what are the claims, and what is the business logic. If someone shares the business logic, I can work on that. I can provide the fraudulent claims to you, right? But when it comes to the clinical SAS programmer, without knowing the clinical standards, without knowing clinical domain, you cannot work on it. As simple as that, right? A clinical domain, the knowledge is very, very important. Without that, you cannot do, right? So you understand here, a clinical SAS programmer requires to understand the objectives of the clinical trial study. What is a clinical trial study? The objectives, the phases, the study design, all these things we must be aware of. Without this, we cannot work at all, right? So that is the reason, because most of you are, I can say that 100% of you are already completed your SAS programming. You know the ABCD of your SAS programming. But why you are here? Because you want to understand this piece, the second piece that I'm talking here. So what is the second piece here? The second piece that I'm talking here is going to be what is called here clinical trial studies. What are the clinical phases? What is the study design? Right? So that's the reason we need to understand the basics first. So these three are, along with these three, there are certain elements as well. These elements are very, very important and crucial to us. Like I said in the previous class, on our Friday class, I told you that, Thursday class, I told you that before we enter into the SDTM, ADaM, or PLF, we need to understand few of the basics. We need to understand some of the definitions. We understand some of the documents. Right? That work we have started now. Right? So the first step in initiating the clinical trial is writing a protocol. What is the first thing? We need to write a clinical trial protocol. Or I can say that any clinical research will start with this particular one. So it is the entry step for any clinical trials. Now the question is, what exactly it is? A clinical trial protocol is a simple document, but you can consider that it is a Bible for clinical trials. Every clinical trial is completely carried out only based on the details that are present in the clinical protocol. Whatever is mentioned as is, you need to conduct that. No violations. In certain cases, there will be few violations, but those violations also should be within the limits, right? Some people also write in the protocol, these might be the violations you can expect as well. You are going to take the amendment for that as well, right? But on a lighter note, I can say that every clinical trial will start with the protocol. Okay, I said that this is a document. So who will prepare this document? This document will ideally prepare by sponsor. So who is sponsor? The person who is giving us the funds. The person who is actually giving the fund to this entire clinical research. Sometimes a sponsor can be an individual person. Sometimes a sponsor can be an organization as well. If it is an organization, you call it as a CRO, contract research organization. So a clinical trial protocol which will have end-to-end details about how you can carry out your clinical trial will be prepared by a sponsor or contract research organization. These are none other than the people who will provide the funding to our research. Clear? The very first thing that all of us need to understand is going to be the first step in initiating the clinical trial is writing the protocol. But if someone asks you who will write this protocol will be written either by the sponsor or by the contract research organization. Then a sponsor can directly write this protocol. Let's say that I am the sponsor, I have enough money. All right, I have some billions of money with me and I want to initiate some research on, let's say that, increasing the longevity of the human being. So let's say that the people are now living for 80 to 90 years. I want to increase the longevity. For that, I want to provide the funds to some of the bunch of the scientists. But I cannot prepare the protocol, right? I can only provide the funding. I have an idea, right? But I don't have the details about how to write it, where I can get this particular protocol sample, right? What and all the fields that should be part of this particular protocol. All these details are not with me. So I need someone help to prepare this particular protocol. Correct? Sorry. Now, who just a minute, I will be on mute. All right. Now, who will prepare this protocol? Sponsor, but with the help of whom? Right? So here we can see an investigator, or we can also call it as clinician. A clinician is clinician, or even if you consider the investigator, both of them are referring the same person, which is nothing but a doctor. Nothing but a doctor. A clinician or an investigator, along with the biostatistician, will help or will prepare this protocol. Right? You can ask me why biostatistician and why clinician, or simply why a doctor. The doctor is the one who's supposed to go with this particular entire trial. He is the one who will conduct, because he is the only one who have the deeper understanding about the disease, or who is having the deeper understanding about the drugs or even the procedures. So a doctor must be there. A doctor, we can here call it as an investigator. Right? Again, or if you say that Murli, why you have two different names? You have something called clinician and you have something called investigator. Is there a difference between them? If you strictly ask that, again there is a difference. A clinician is the one who will take care of the patients, who will take care of the patients. Even he or she may directly or indirectly contact patients. Right? That is the deeper understanding. An investigator is the one who will be in contact with the patients at the same time. He or she are the ones who is going to conduct the entire clinical trial. Right? A clinician may not be part of your entire phase 1, phase 2, phase 3, phase 4. But the investigator is the one who will be throughout your disease. Okay. So the first thing that we need to understand is the protocol document will be prepared at the site of your CRO or the sponsor site. A sponsor alone cannot produce this particular one. He would expect some help from the either or both of your statisticians and clinicians. Once the investigator receives the protocol, first they will determine, let's say that I received a protocol. I cannot blindly start my clinical trials. I need to check the feasibility, right? Whether these clinical trials are possible or not. These are in realistic note or not, right? If they are realistic note, then only I can enter into the clinical trial, right? If someone asks you, how do you define the, whether the feasibility is there or not, right? So for an example, if I say that drastically I want to increase the height of a person, I need a drug to increase the height of a person by, let's say that 5 inches or 6 inches. How practically it is possible? May or may not. Check the feasibility. Check, are there any history where there is some drug which help us to increase the height of a person, right? So like that, I need to check the feasibility first. First of all, it is practically possible or not. Once we understand that, yes, on a lighter note or at a very high level, we understand that this particular clinical trial, carrying out this particular clinical trial is feasible. Right. Again, before we kickstart with our clinical study, again we ask few questions ourselves. What are they? Does the investigator have the necessary patient population? What do you mean by population here? What do you mean by population here? Number of subjects, that is nothing but number of patients, or number of volunteers, healthy volunteers. Because I want to carry out this particular trial, do I have enough number of people? Let's say that your sponsor has given only 2 people to you and asked you to carry on your clinical trials phase 1. Can we do it? Practically it is impossible just with 2 subjects. We already know the boundaries, minimum of 10, maximum of 80. Sorry, minimum of 30, maximum of 80. Those are the minimum numbers I'm expecting, right? The minimum population based on the previous values are going to be 30 to 80, but I doesn't have 30 to 80 people now. You have given me just 2 people and you asked me to carry out the analysis. I cannot do that, right? So the first thing I need to understand here is, do I have enough population or not? Who will decide the population? The population will be decided by the biostatistician. Not only for this, for all the phases, correct? So for the second phase, we have around 100 to 500. For the third phase, we have 1,000 to 5,000 people. And for the fourth phase, we have 10,000 to 50,000 people. All these phases, the randomization, the random number of people, all these algorithms and the numbers will be produced by your biostatistician. Okay. Second, is the protocol well designed and clearly understood by the study team at the trial site? What do you mean by trial site? I told you all these clinical trials will carry out either at a community clinic or at a doctor's site or at a research site in a hospital, right? These are the various places where the research will happen. These are the various places where the clinical trial will conduct. Now the question is going to be, whoever at the trial site, are they clearly understand the entire protocol or not? Because the protocol is going to give all the details. Whatever the details that are mentioned in the protocol need to be clearly understood by all the people who are working at that particular trial site, right? Even though the protocol is good, but if I don't have enough people who can support it, can I take care of that particular one? Not possible, right? Not possible, right? Because I need to have a clear and crisp understanding about the complete details present in the protocol. That is the reason we are also studying the protocol now. Okay? Then, are the study timelines are reasonable? Let's say COVID. If government asks us or asks all the researchers, hey, I want the vaccine, right? But I'm giving you a timeline, that the timeline is going to be 3 months. I'm giving you the protocol, what need to be tested, everything, how the procedures are going to be, everything I'm giving to you. The protocol is crystal clear. All the people who are working at the different sites also understand the protocol. Fine. The first 2 things are done. I have enough people who are volunteering this particular research. So the first 2 questions are answered already. Now the third question is the government saying that I want this particular stuff need to be completed in 3 months. But the timelines are not reasonable now. These are not realistic. For sure I cannot complete this particular within the 3 months of the time. So I definitely need additional time. So the timelines are not reasonable. I cannot quote the exact timelines, but I'm 200% sure that 3 months window for bringing up a new vaccine for protecting against COVID is highly impossible. So we need to understand. We need to have that proper population. We need to have the protocol need to be understood by all the people who are at the trial site, and also the final thing is going to be the timelines must be reasonable. Clear up to this? Why we are talking about the clinical trials protocol? Why it is important? Can I have a quick confirmation from everyone? Is this understandable? Yes or no? Any queries? All right. Next thing. So we only talk at a very high level. We said that there must be a protocol document. We said that who prepare it, with the help of whom it is getting prepared. Only that we have discussed. Exactly what it is, exactly what is called protocol document, and what are the various things that are present in this particular document we need to understand. Correct? For that, we see this particular one. So what exactly this clinical trial protocol document consists of? Number one, a clinical trial protocol document. It has the details about the first and foremost thing, very, very important, how the clinical trial will be conducted. Right? You have phase 1, phase 2, phase 3, phase 4, preclinical, all this, right? How it need to be conducted? What need to be tested? Number 2, what are the objectives of this particular clinical trial? Let's say COVID vaccine. What is the objective of the COVID vaccine? We need to ensure that we need to bring a vaccine. It should be protective against the COVID. As simple as that. Right? Let's consider polio. The polio vaccine objective is going to be, once the person has taken this particular vaccine, the person should not affect with the polio at all. Correct? What is the objective? There are a lot of stuff again. Primary objective, secondary objective, primary outcome, secondary outcome. Lot of stuff will be there. We will discuss that later. But on a lighter note, why it is required, what exactly it describes, how you are going to conduct your clinical trial, what are the objectives. Number 3, design. Number 6, when I say design, there are a lot of design procedures. Each design procedure we need to understand. Something like cross-functional study, or parallel study, or parallel design. There are a lot of designs available. So what design you are using for this particular protocol? There are n number of methodologies that are available to us. So what methodology you are using for this particular protocol? Statistical considerations. We need to say, what are the population limits? So when I go with the results, what are the percentages that I'm looking at? It should be 100% accurate, or 80% accuracy is fine. When I go with the efficacy, what is the level of efficacy I'm looking at? What are the standards that I'm maintaining? All these will be part of statistical considerations. Right? Then, organization of the trial to guarantee the safety of the participants, because you are also giving the healthy participant to them. Right? If you give the healthy participant to them, what is the level of guarantee that I'm getting that these patients will not fall sick? These patients are not going to be ceased. How I can give the safety consideration of this particular bill. And last one, this is very, 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 very important. Most of us will take this in a light manner, but very important. What is the integrity of the collected data? That is, you collected the data, right? I'll tell you a very simple example. Most of you might aware of a very rare blood group called Bombay blood. You might know this. Only 10% of the population have this particular one across the world. That is called Bombay blood, right? So we are carrying out some clinical research. For the clinical research, what we did is we looked for some of the healthy volunteers. Let's say that 2 volunteers has come up with this particular trial. So we know these 2 people. We carried out our entire analysis. The clinical trial is complete. We have sent these 2 people again outside. That is nothing but trial is completed. They are free enough to walk into the normal world. But your data is leaked, either intentionally or due to some reason. The data is leaked. So what do you mean by data leak here? You expose these 2 people to the external world, saying that there are 2 people who have Bombay blood group. Since it's a very rare group, so what will happen? Anyone who is in badly need of this particular blood group, right? They illegally also can contact these 2 people. If these 2 people are not willing to, donate their blood, they can go to the any extent. Correct or not? So the data leak is going to be the major problem. So you need to maintain the integrity of that particular data as well. You need to ensure that this data is highly confidential. We will not expose this data to the external world. All these measures you need to do. So for that, what are the various approaches that we are doing? All this information you need to keep in this particular document. First of all, you understand what it describes. So what it is describing, how the clinical trial conducted, what is the objective, what is the design, what is the methodology, what are the considerations, which organization is doing it, and how they are providing the safety to the participants, and finally, how they are maintaining the integrity of the data that we have collected. Is this clear? What a clinical trial protocol document consists of? Everyone, can I have a quick confirmation? Any questions here, guys? Wonderful. Next, this is extension of the first thing. What is this? Again, I'm talking about what is clinical trial protocol? Why is this clinical trial protocol needed? Maybe I can extend the same here. Why? The protocol is the key quality control tool for all the aspects of the clinical trial. Why? Because what do you mean by quality? How my clinical trial is happening? What is the outcome? Is that outcome is the correct outcome or not? Which is nothing but the quality control only, right? So since the protocol has all these parameters, if I follow these parameters as is, I can 200% ensure that our quality is going to be good, or the outcome is going to be good. Correct? Again, the same thing. So what this clinical trial protocol document is going to be prescribed, or what it is going to ensure? It ensures that the health and safety of the study participants. This is what I have also mentioned here. It provides the precise study plan. Here it is. Design. It defines and manages the trial. That is nothing but methodology and design. It should be strictly followed by all the study investigators. That is, and all the doctors who are helping to carry out this particular trial. They need to strictly follow this particular one. Again, it guarantees the integrity of the data. We mentioned this here, allowing the combination and comparison of data across all the investigators and the study sites. Sometimes you may not be able to carry on the clinical trials at a single site. There will be multiple sites across the globe, At multiple sites, we are carrying the same study. In that particular case, we need to collate the data. So for that particular one also, we are going with the clinical trials, right? So this is purely for the interview purposes. What is the clinical trial protocol? What it consists of? What are the elements of that? Each element we need to understand. Remember, each element we need to understand. All right. Let me go to the next one. So I just have given the information about protocol on a lighter note. Now, if someone asks you a good protocol, a good clinical trial protocol, what are the elements it should consist of? These are the elements. What is that? Title. This is very generic. Background information. What is the objective? Study design. Selection and exclusion of the subjects. Treatment of the subjects. How I am assessing the efficacy. How I am assessing the safety. What are the adverse events? Is there a discontinuation of the study? What are the statistics? What is the quality control? What are the ethics that I'm following? How I am handling the data? How I am keeping my records? Is there any publication policy? Right? What is the project timetable or flowchart? What are the references I'm considering? Finally, what are the supplements and analysis? Right? Why I am going with this is for the next 2 classes, we are going to discuss all this, everything. What is a study design? What are the types of study design we have? If someone says that what is selection or exclusion of subjects, on what basis you do the selection, on what basis you do the exclusion, right? Treatment of the subjects. What are the different types of treatments we have? How you go with the efficacy? What do you mean by events? What do you mean by adverse events? Right? Under what cases you go with the discontinuity? What are the statistics? Everything, whatever you can see it on the screen, each and every topic that is present here, elaborately we are going to discuss. Once we complete this particular one, I can say that that is the end of theoretical stuff, which is part of your clinical trials information. Then we can happily enter into the SDTM. So to do that, or to enter into the SDTM, we may need to wait for 2 months. Clear? This is just for your reference purpose. Now, let me show you one of your template. This template, you can download it from the website. I'll give you the website details also. All right. So this is one of the protocol template. So generally, we take this template, we are going to fill it. So what and all we have mentioned here, we said that it should have title, page, or general information. Can you see this? Well, who is the sponsor? Because the sponsor is the one who is going to prepare this particular one. Sponsor or the CRO, correct? That is Contract Research Organization. Now you can see protocol name. That is what I mentioned here. Title page is nothing but the protocol name. The protocol number, version date, we may have multiple versions. So what is the product, the IND number, development phase, who is the principal investigator, who is going to be the medical monitor, who is going to be the coordinating center. Remember, these also we are going to discuss. That is, who and all involved in the clinical study. What are the different, what and all the teams which will participate in the clinical study, right? So what is the work of each and every team? That also we need to discuss, right? So that is also part of your, I can say, definitions. Okay, now scroll down a little. Protocol agreement. I'll share this particular template with you guys. All of you kindly look at this. This is the one. This is a very, very important one. You can see there will be approximately 17 different topics here. All these 17 different topics are part of this. Okay, see this. One by one we go. I'll share this particular one later. You can look at it, but still, I'll just give you the few details. Background. Identify the product to be studied and describe it briefly. So, what is the product or what is the drug? Is it going to be a developed treatment drug, or it is going to be preventive drug or it is going to be a medical device, give the information of that, right? So if this particular product have some non-clinical information, what is the significance of it? Give the summary of it. So for clinical studies, give the clinical data because everyone requires the data, right? So give the data. So here you can see that the risk or benefit section, and you can also see study objectives. What is the primary objective? What is going to be the secondary objective? What is the study design? Can you see this particular one? This is a single center, double-blinded, placebo-controlled, randomized, incomplete block, 3-period crossover trial. Can you see this? This is the study design. We need to know all this information. That is what is called single center, what is called double-blinded, what is called placebo-controlled, how randomization is happening, what is called incomplete block, what is called 3-period crossover trial. So each one of this we need to understand. Here it is only double-blinded. Here it is going to be placebo-controlled. What is called placebo, right? What is called 3-period crossover trial. These are the definitions, because these are the only interview questions. When you say clinical trials, these are the clinical trials questions, right? So we need to understand this particular one. So what is primary efficacy endpoints, secondary efficacy endpoints, how you are actually defining your endpoints, right? So this is a template. Whatever you can see here is a template. There is another thing as well. Let me just give me a moment. Just allow me a second. I'm trying to. Here it is one. This is one of the protocol. This is defined by WHO. This is defined by WHO. Yeah, World Health Organization. Here also you are going to have the same thing. More or less you are going to have the same thing. Here the template is slightly different. Slightly different. But on a whole, more or less it is going to be the same. Right? Here you can see block randomization with randomly varying block size. Right? Here also blinding or masking is going to be, the blinding is going to be placebo. Right? And it is going to be a saline one and it is a oral one. So what kind of blinding it is going to be? How I am collecting the samples? Every piece of information must be entered. So there also we talked about the adverse events, right? Here you can see the adverse events. The adverse events is going to be health status of participants will be monitored and reported as a study outcomes, right? So at this particular point of time, you may feel that this looks little difficult to me, maybe at this particular point of time, right? But once we understand all these topics, that is nothing but what is called placebo, randomization, and what is called adverse events, what are the treatment methods. Once we understand all these parameters, and when you start reading this particular document, you don't find difficult at all. And even I can say that you feel comfortable with this, because you know how to read this now, right? Till that you don't know how to read. But once you have this particular one, you can clearly understand and you can go ahead with how to read this particular protocol. Just give me a second. I'm trying to find out one other protocol. I'm not able to get this. Just give me a second. Okay, I'll check and I'll share the protocol with you. So that protocol will be the original protocol that was used by one of the clinical trials, that we can use it for the reference purpose. All right. So what we need to discuss after this, we know what is protocol, we know who are going to prepare it, with the help of whom it will get prepared, what are the elements that are involved as part of your protocol, because it is the entry point for your clinical trials. So the next step is going to be, we need to understand each and every topic or each and every parameter which is part of your protocol document. So once we complete that, then we need to understand the cross-functional interaction. That is nothing but you have a sponsor, you have an investigator or clinician, or you have a statistician, you have a data manager, and you have a medical. we are going to work with all of them, being a clinical SAS programmer, right? So we need to understand the role of each of this particular individual. So what is the role of a sponsor? What is the role of an investigator or a clinician? What is going to be the role of your biostatistician? What is going to be the role of your clinical data management team? So what is going to be the role of your medical data? So when you go to the clinical data management team, you are going to understand something very interesting with this context, the CRF, case report form. So how to how to understand this particular case CRF form. What is called ECRF? What is called ACRF? All these terms we are going to understand and we are going to clear, right? So we may need 2 more classes to understand all these definitions. So from tomorrow's class onwards, I'm going to explain about the definitions. So the definitions, I'm going to start with protocol. So we are going to understand all the details which are present in all the elements which are present in the protocol, and then we understand what are the various teams that are involved in the clinical trials and what is the role of each of this particular roles and responsibilities of this particular team. And then we are happy to enter into the most important the interesting part of your clinical trials, or clinical SAS, which is nothing but the SDTM. Before we start this particular one, we need to understand this. So to get this particular base, we may take a little time, but it won't take too much of time. So ideally, 2 classes are more than sufficient. Is that clear, guys? Is that okay to everyone? Is that understandable? Anything else you would like to discuss here? All right. Okay. Now, so what are the things that are pending from my end? So to start with this particular one, or if you want to refer something, first thing I need to provide is I need to provide you the protocol template. And second thing, I need to send you one protocol example. That is, I need to send you the protocol template. I also need to send you the protocol example or sample. So these 2 I will send along with this class notes. So you don't need to worry about the class notes. I will share the entire class notes to you. Even the clinical trials, phase 1, phase 2, phase 3, phase 4 is there, right? That one also I'm going to prepare it and I'm going to send it to you because it is there here. Here you can see, but I will put it in a proper format and I'll send it across to you. Clear? Mean to say, send it across to you means I will give it to Balusa. He will upload it in the questions, anyone? All right. So I'm going to wrap up the session here, right? It's already 55 minutes we have started the session and it went well, I believe. So any questions, I'm happy to answer that. We can have a Q&A now. If you don't have any question, we can wrap up the session for today. So perfect, everyone. See you tomorrow. Take care. Perfect. All right. So where are we? So yesterday, let me share my screen. Here it is. What and all we discussed yesterday, I think if I'm not wrong, the last thing that we were discussed yesterday was about the clinical trial protocol. Am I correct? I told you that a clinical trial protocol is the one which I can call it as an entry point for your clinical trial. Correct? So this is the first document, or it is the first one which will define your clinical trial. So this document was prepared under supervision of your sponsor. Sponsor is nothing but this is the person who is going to provide the funding for your entire project. I mean to say that for the entire clinical trial, sponsor is the guy who is going to provide the funding. But we also discussed yesterday that sponsor alone cannot do this particular one. Sponsor will take the help from either clinician. We can also call him as an investigator. Along with the investigator, we also have a biostatistician, correct? This is what I have mentioned yesterday, correct? So we have a biostatistician and investigator. Investigator or clinician are nothing but the doctor, right? There is a minor difference. The minor difference is going to be a clinician is the one who personally take care of the patient or the subject. The investigator is the one who will carry out the entire, right? Sometimes you may also have another role. We can also call them as a principal investigator. A principal investigator is the one, you can simply call him as a senior doctor, right? That's a basic difference. Okay. Sometimes the sponsor may not be an individual. Instead, we have CRO. What is CRO, guys? Anyone from yesterday's class? If you remember anything, what is CRO?
Clinical Research Organization.
Clinical research, no.
It's contract research organization.
Contract?
Research organization.
Exactly. It is a contract research organization, right? So when the sponsor is not an individual, in that particular case, we may also go for the CRO. The CRO is nothing but the contract research organization. Either of these two guys is going to produce the protocol. Remember, being the SAS developers or being the SAS programmers, right? You don't need to have an in-depth knowledge about the protocol. Doesn't require. Why? If you are part of a CDM, clinical data management team, or if you are part of your clinical research team, in that particular case, it is mandatory to know each and every piece of your protocol, right? It is. I'm not restricting you to learn about the protocol. I will guide that, and I'll also suggest that learn about the protocol, but don't go deeper into the particular one, because the protocol will be having around 14 to 15 different elements there. All the elements may not be required for us being a SAS programmer, right? Then again, the second reason why I say that don't go in deep about this particular means each protocol is different from company to company. Mean to say that from sponsor to sponsor, the protocol will be different, but there is a standard guidelines, right? So that is nothing but how a good protocol can be. There are guidelines for that. Every company or every sponsor or CRO more or less will follow the particular guidelines. Sometimes there are violations as well, but we need to ensure that FDA will ensure that the guidelines are within the boundaries. All right. So I told you yesterday, or I have also shown you the protocol template. You can see that this is the protocol template. You can see there are approximately 17 different parameters here. 17. Right. Going through each one of this, understanding each one of this is really a difficult thing, right? Every one of us doesn't need to do that. But whatever the basic information, especially for the interview purpose, we need to understand. All right. So today we spend some time on this particular protocol, right? And again, parallelly, we also try to discuss about, that is first topic for today's class is going to be how the protocol looks like, what are the elements of the protocol. If you completely go with only the protocol elements, it looks like a very dry session, right? Completely a dry session. You may feel bored or you may feel exhausted. So parallelly, I'll also talk about crossover teams. What are the various teams we collaborate? We work with. Being a SAS programmer, as part of your clinical science, clinical SAS, you're going to collaborate with multiple teams. There are multiple roles will be there. Who and all? What are their responsibilities? That also we need to discuss. So parallelly, I'll also start this today only. In addition to that, I'll also talk about what are the various definitions which are involved in your clinical trial. That is, before we start with SDTM, this is our core. SDTM, ADaM, and TLF. These three are the ones we need to concentrate. But before we go there, we need to understand few definitions. We need to understand what are the various teams we work with, and this entire clinical research, the entry point is protocol, so we need to understand the elements. So from today onwards, for the next 2 classes, we discuss these 3 things parallelly. No worries, these 3 are individual. Once you complete your definitions, you will be able to understand your protocol properly. That is the overall objective. So we start with the protocol, slowly we discuss the elements, parallelly we go with the crossover teams, and we also discuss about the definitions. Clear? All right. All right. So a clinical trial protocol, the elements are here. So what are the elements? There are 17 elements, but I have given you like the crux of all the elements here. Ideally, the first element is supposed to be the title, but I haven't keep the title here because every one of us know how the title looks like. So I'm going to explain the title directly, right? But I haven't keep that here. So there is a title, there is a background, study rationale, objective, study design. This one we need to understand in detail. What is that study design? Subject selection, study treatments, what are the various procedures, adverse experience reporting. We can also call them as adverse events. Very, very, very important one. So discontinuation of the subjects, under what cases the discontinuation of subjects will happen. And then, just give me a second. Just give me a second, please. Yeah. And also we need to talk about, I told you about the violations, correct? There are some violations will happen in the protocol. And I also told you that those violations should be within the limits. So what are those violations? And statistical considerations, that is nothing but what will be the, there is going to be something called efficacy. Efficacy is nothing but the efficiency. So what might be the efficiency? For few people, efficiency is 100%. For few people, efficiency can be anything about 80% also. What are going to be the considerations? What is going to be the data collection and monitoring? Make sure all the data that you are collecting is going to be ethical and regulatory considerations you need to consider. We are going to discuss all of them starting with the title. All right. So let's start with the title. How a title looks like. So you can see that a title page is the one which can have the details about what is the protocol name, who is the sponsor, when this got started, that is started version date, what is the product that we are actually investigating, what is the development phase. You might be asking, Murli, what is this IND number? This IND number is also related to us, but we don't go in deeper about that. But the development phase is important. Meaning of the development phase is phase 1, phase 2, phase 3, phase 4 will be there, correct? At what level you are writing this? At what level you are writing it? Sometimes you can see here on the right hand side, that's the reason I have given you the example also. Can you see that on the right hand side? The phase development, here it is. The phase development. It is at the phase development is 3. So it is actually representing the third phase of the development. So on the right hand side, whatever the example that I'm trying to give here, this example, this particular protocol is the protocol that we are giving for the phase 3, right? You can ask me, Murali, you said that it is the entry point for the clinical research, correct? So what is the meaning then? The phase 1 development, phase 2 development were already completed. For those two, phase 1 and phase 2, there were different protocols already, correct? If I'm continuing with the phase 1 itself, let's say all the 4 phases I want to include in the one protocol, I can do that. If not, I want to go with the different phases. For each phase separately, I want to provide the this one. In that particular case also, we can do it. So it's up to us. Mean to say it's up to the sponsor. It's up to the CRO. Clear? Now, let's see that how the title looks like. Can you see this particular title? A phase 3, because we're starting with the phase 3 development. Double-blinded, placebo-controlled, randomized trial to determine the efficacy and safety of a low and high dose of, this is the drug name, NS03140 as add-on therapy in subjects with Alzheimer's disease. This is a Alzheimer's disease treated with a stable dosage of single, what is this? Dopamine, not able to, maybe I need to zoom it. Agonist. Okay, first I'll tell you what and all we need to understand from this. Phase 3 we know, right? This is nothing but the phase 3. You can see the double blinded. So there are 3 types of blindings. Blinding is nothing but masking. Anyways, I'm going to discuss that as part of these definitions. So just I'm going to give you a overview. So there is going to be a single blind, a double blind, as well as the triple blind. Totally 3 kinds of blinds will be there. All the 3 blinds we are going to discuss. A protocol or the treatment can be a single blind, or it can be double, or it can be triple. Then placebo control. Placebo is nothing but a kind of a drug only, right? Right. So what is the randomization? Like how many subjects we are taking? All these things we are going to discuss. Okay. So for now, we just understand that there is a title talking about some of the definitions and parameters. Those parameters were part of your definitions. Today itself, we are going to discuss about this blinding thing. Right. Background. Because if you see that the second element that we are going to talk here is going to be background. So what is the background of this particular one? Under the background, 4 things we are going to discuss, or 4 things we are going to concentrate. What is the drug composition? Right? Whatever the drug that we are going to take it, what is the composition of that? Mean to say that most of the drugs are not a single medicine, are not a single chemical composition. There are going to be more than one composition will be there. Best example is going to be if you consider your one of the most common irregular thing in the Indian community is diabetes. Correct? We cannot call diabetes as a disease. It is a irregularity. Correct? So because of the food habits or because of the your living methods or your lifestyle, you are going to get this irregularity. Correct? So to manage this, what we are doing, we are consuming the tablets. So what is the most common tablet that we are going to use? There are going to be a lot of vendors, but the common composition is going to be, you are going to have the metformin plus there will be another most of the times. But the common composition is going to be metformin plus something else. Correct? So what is that particular composition? Right. Second, mention the disease or condition for which the investigational drug product is being evaluated. That is, whatever the drug that you want to take or that you want to consider, for what disease you are actually doing this, because metformin has multiple things. It is going to cure or it is going to act like a supplement for the multiple diseases. But for what disease, to treat what disease you are actually investigating this. Okay. Provide a brief summary of non-clinical data that has clinical significance. We are going ahead with the clinical trials. There is some non-clinical data. Those are not falls under clinical, but those are important for our clinical trials. So if you have anything like that, brief them. Brief them. The last one is going to be provide a brief summary of the clinical things, clinical significance. That is nothing but what we need to test on all. That is called clinical. Other than the clinical, what is the other details which are having impact on our trial is going to be a non-clinical data. Both of them are required for what we are developing this particular drug. And finally, what is the competition? This is the basic background of your clinical trial. Am I correct or not? This is the basic thing. What is the medicine? What is the composition? To treat what disease you are doing this. What was the non-clinical data that you need to consider? What is going to be the clinical summary? Then, study rationale. Study rationale primarily talk about one thing: risk or benefit assessment. What do you mean by risk or benefit assessment? Every drug has a risk involved in that. Every drug we are taking to get some benefit. Am I correct or not? I'm taking metformin. Metformin we are taking to get the benefit of lowering your sugar levels. Very straight thing. The benefit is going to be, it is going to lower your sugar levels. But what is the risk involved there? I don't know how many of you are aware of that. Metformin is one of the primary reasons for cancer. It will activate the cancer cells, right? Though we all aware of that particular one, but still we consume the diabetic tablets because there is no other alternative. Either you need to control that by having a proper food, maintaining the lifestyle. In addition to that, since the sugar levels are very high, we must take this particular medicine. So the risk involved there is going to be, there is a high chance that it may activate the cancer cells, because every one of us will have the cancer cell. When it got activated, then the case is going to be different. Correct? So we need to understand how the specific risk of this particular product will be mitigated in the study. So we know that the risk is involved already. The risk is going to be activate the cancer cells. I told you in the beginning, we always take this metformin plus another chemical composition. The plus another chemical composition is to mitigate your risk. That is nothing but it tries to suppress those cancer cells. Metformin is going to activate it because it is having two things. The benefit is going to be it lower the sugar levels. The risk is going to it activate the cancer cells. On the other side, the other composition that we are providing along with your metformin is going to lower your sugar levels. Sorry, it's going to suppress your cancer activity cells. Correct? So we need to understand this particular one, because if you are not able to mitigate the risk, it is not safe enough. If you remember, the primary reason of carrying out the clinical trials is to understand and measure the safety of the individual or safety of the subject. When the safety features are not meeting, when the safety parameters are not able to meet, then there is no way, or then there is no need of doing the clinical trials, or there is no need of developing your drug. Am I correct? So what we need to do? Assess your risk, assess your benefit. When you assess the risk, understand how to mitigate the risk as well. Is that clear? Right? Because we all know that it is to describe why investigational drug will be safe and effective for the clinical indication intended. Meaning, for what we are targeting, for what purpose we are actually taking this particular drug, that drug need to be safe and need to be effective. Right? If there is any risk involved, analyze the risk, assess the risk, and mitigate the risk. Then only we can proceed further. That's the reason the study rationale is very, 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 very important. Can I have a quick confirmation from everyone? 3 parameters we have discussed today. The 3 parameters that we have discussed here are: the first one is going to be under the elements, the title. Basically, the title gives the complete information about the health. It has the sponsor information. It has the version details. It has what treatment we are actually doing. Correct? And it also has the information about the primary investigator and secondary investigator and any other teams involved there. The team information will be available. Number 2, background. The background is going to talk about 4 parameters. What are the 4 parameters it is going to talk? It will talk about the drug composition. Second, it will talk about if I'm taking this particular drug to treat what disease it is going to be, we are going to consume this particular drug. So it is also going to have the details about the disease. All right. Then what is the non-clinical data that have impact? What is the clinical data that we need to consider? That is about the background. Third thing, what is the third thing? The third thing is going to be, we are going to talk about the study rationale. So what is the study rationale you talk about? It primarily talk about 2 things: risk, benefit. Obviously, we look at the benefit. The benefit is nothing but it primarily talk about the why I'm taking this particular medicine. If I take this particular medicine, what is the benefit I'm going to get out of it? But along with the benefit, what is the risk? Analyze the risk, assess the risk, mitigate the risk. Yeah. So 3 parameters, all of you are comfortable. Can I have a quick confirmation? Every one of you? Any questions here? Wonderful. Right. So the next thing that I'm going to talk here is study objectives, because we also have the study objectives, right? So now to understand this better, parallelly, we also see this particular document. Let's say this is one of the protocol. I told you yesterday I'm going to give you some of the protocols, right? So this is one of the protocol. Let me also open this particular. So rational, maybe I can also share this particular one after this particular class. I'll share this. This is the protocol for you. Can you see this? This is the protocol. This is 2.0 version. This is primarily for the drug NS0314D, the same that I have also mentioned in your this one also. Can you see this? Here, NS0314D. This is the investigational drug. For this investigational drug, this is going to be the protocol. All right. This drug is primarily to treat Alzheimer's disease. Okay. How it looks like. Title, we already discussed the title there because the title is already there. Now we are talking about the objectives. Can you see that? We are actually at the objectives page. This is the objective. So there are 2 objectives. Primary objective, the second one is going to be secondary objective. For primary objective, what is the efficacy endpoint? Efficacy endpoint is nothing but this is the way we are going to measure the efficiency. You can understand in that particular way. This is the highest parameter for identifying at the same time for measuring your efficiency. Let's talk about the objective first, then we talk about the efficacy endpoints. Can you see the primary objective? The primary objective is nothing but what is the specific aim and objective of this particular trial, right? Why I am carrying out this particular trial, right? So to do that particular one, I need the data. So we are going to collect the data, right? We are going to collect the data to address a specific aim and objective of the data or of the clinical trial. Clear? Let's see how the objective is defined here. The objective of this trial are to evaluate the safety and efficacy of 2 doses of 0314D drug, NS0314D drug. One is 50 mg, another one is 100 mg, right? Compared to placebo as add-on therapy in the subjects with Alzheimer's disease, right? So what they are doing is they are currently receiving this one stable dose of single dopamine agonist. This is the single dose they are actually taking already. They are currently on this particular one. But what we have to do now, We want to test how safe it is and what is the efficiency of it. Sorry, we want to understand how safe and how efficient it is if I give two different types of drugs. One is going to be 50 mg and another one is going to be 100 mg. That is the primary objective, correct? So that is the objective. Here you can see primary as well as the secondary. So for the primary and for the secondary, what I need. So I need to evaluate the changes, right, from the baseline to W24 in a motor symptoms. So this is too deeper and this is too much related only to this particular Alzheimer's disease. So there is going to be a baseline. From the baseline to W24, till that we are going to evaluate. Clear? So second one, the secondary objective is going to be, this is UADRS section 3, this is another parameter. Don't go that deeper into that. Second one is, again I'm going to do the same thing, but here, instead of motor symptoms, I'm going to do the same in the daily living, cognition, changes in the global clinical status, all these other things. This is nothing but a regular life. This is at the motor symptoms, right? So each drug has a specific target. So each protocol is going to be different. When it comes to us, when it comes to that being a clinical SAS programmer, we don't worry too much about it, right? But if you are a clinical data management guy, a clinical research guy, then you need to understand what is W24, what is called motor symptoms, what is called UADRS section 3, all these. But for now, our objective is to understand the protocol. To understand the protocol, this information is more than sufficient. All of you are clear about it? Can I have a quick confirmation on primary and secondary objectives? It is all about objective is nothing but why I'm taking this, why I'm considering this particular drug, what I'm doing with this particular one, right? Can I have a quick confirmation? All right. So it's going a little rough. So what we can do now is we can slowly move from protocol to, let's go to the crossover. So till now we discussed about the protocol, right? So now let's stop here because we talked about four different parameters: title, background, study rationale, and also we also discussed about your objectives, right? So let's stop here. The remaining 5 or 6 or remaining 5 or 10 we're going to discuss in the tomorrow session. For now, we can make a switch from here to crossover teams because until unless we change the topic, we don't get the fresh air. So let's change the topic and continue with the crossover teams. Again, tomorrow we will discuss with the remaining of them along with the remaining crossover teams and the definitions, like the previous. Okay, right. Now, let's come back to our slides. So, what are the various cross-functional teams that we are going to interact? So, with what people we are going to collaborate? So generally we have a sponsor, clinician or investigator. These two we are already aware of. The person who will provide the funding, the person who will carry out your investigation. So these two we are pretty much okay and pretty much comfortable. That's the reason I'm not spending much time here. Instead, I'll be moving to the biostatistician. When it comes to the interviews or when it comes to the roles, there is always a confusion. The confusion is going to be, is statistician and clinical SAS programmer, are these two are the similar roles or the same roles, or are there any difference between them? Remember, biostatistician is entirely different from your clinical SAS programmer, right? A clinical SAS programmer over the years, by gaining enough knowledge with respect to the statistical terms, can turn as a biostatistician. There is a possibility. But ideally speaking, these two are two different roles. Right? Now you can see here clearly. The role of a biostatistician is to work with the clinician and clinical programmer. Remember this. Clinician is nothing but a doctor. A clinical programmer is nothing but you and me, right? What are the responsibilities? The first thing, help with the statistical design of the clinical trial study. So we need to understand what is called the statistical design. That will come under your definitions. Today only we are going to discuss that one as well. Then help with formulation of hypothesis. You guys remember where this hypothesis has come and where we discussed it? Anyone? If you remember the clinical trials procedure that I have explained on the day one, I told you three things. Number one, in each clinical trial, by end of each clinical trial, a question will be raised. A hypothesis will be developed. Third, analyze, assess, and repeat. Am I correct? First, a question will be raised. Second, a hypothesis will be created. Number three, analyze and assess the question and hypothesis, and then repeat the clinical trials if required. Remember that? So, simplest example. Let's go with a simple example. Let's say there is a person. Let's consider COVID itself. So there is a person who got COVID symptoms. He diagnosed with the COVID. Initially, let's say that during the drug development time or during the clinical trials, the investigator understands that. So let's say we have developed a drug. The drug has been given to this particular subject. The subject has taken the drug and he is recovered. Let's say that his body temperature becomes normal. He's not feeling any body pains. He almost feels normal. He doesn't have any COVID-related symptoms. So then investigator will say to the clinician, and investigator will update the information saying that our trials looks fine. By the time, the biostatistician along with the principal investigator raise a question. That is, what is the dosage that we have given? In how many days this guy has recovered? The subject has recovered. The question. The investigator can answer it. Then again, the principal investigator or biostatistician ask a question. What if, because we conducted this particular trial in a proper environment, controlled environment, meaning we have provided a proper oxygen to this particular guy, correct? We use the oxygen cylinder and we supplied the oxygen through the machine. Number two, we maintain the same temperature throughout the day because we are in an AC controlled room, correct? Number three, we ensure that there is no other bacteria or there is no other virus in this particular room because we're continuously sanitizing this particular clinical room. So under these circumstances, the drug has worked or the vaccine has worked. What if the person is not in a controlled environment? That is, when the person joined this particular clinical research, let's say he joined in the phase 3, by the time he already have COVID, and we also understand that when we take the CT scan of his particular lung, we can see that he's already having, let's say, 15% of the lung already affected with the COVID. We can clearly see that particular volume in the CT scan. So we started giving him, first we started giving him the oxygen. Then he becomes stable. Then we started giving this particular drug. The hypothesis that I'm making here is, what if we provide the drug without using this particular oxygen support? What if we are not giving this particular drug in a controlled environment? What if we are not continuously sanitizing this? Under those scenarios also, will this drug work or not? That is the hypothesis or question that we make. Right? Number 3, determine the optimal size required to guarantee the level of precision. That is nothing but, optimal sample size is nothing but how many number of people that I do or how many number of subjects that are required for this particular randomization. Give the sample. right? So give this particular sample to get the optimum precision. Calculate the appropriate sample size for the study to address the specific aims. Both of them are going to be randomizations. Give the proper samples to carry out your study properly, right? Determine the data variables to be measured and how they are related to the study objectives, right? Let's say that when it comes to your COVID again, because I'm taking this example more frequently, let's go with the COVID. When we are going with the COVID, right, what are the vitals that are important to me? What are going to be the various parameters I need to measure, which are related to this particular COVID? I need to concentrate on that. What is the primary parameter? We need to check the temperatures. Second thing, we need to check the oxygen levels. Exactly, Sachi, right? We need to check the temperatures first. Second thing is going to be need to let the blood oxygen level, which is nothing but here is SpO2. Correct. So these two are the primary things. Later on, we'll take about the your blood pressure. Later on, we consider about the all other things. Correct. Let's say we are talking about the dengue. When I talk about the dengue, SpO2 is not my primary parameter. My primary parameter is going to be the platelets count. Correct. What is the platelet counts of this particular patient? Are the platelet counts going down or are they stable? These are the parameters for me. Correct. For each of this particular drug or for each of the particular disease, the relative parameters only we need to measure first. We need to give the priority to the relative parameters. So what are those relative parameters? Biostatistician is the guy who's going to help with that. One. Develop the data collection process, provide the method for analyzing the data, and finally interpret the results. Meaning, so we have the data coming from the various sources. So collect the data and cleanse the data, because the data can be data is not going to be proper all the time. The raw data is going to have a lot of things. You need to properly cleanse the data. So the data cleansing, the data collection will be taken care by the biostatistician. Only the process he will develop, but the original data cleaning will be done by the clinical SAS programmer. Second, what are the methods you need to provide? What are the methods for analyzing the data? The methods need to be provided by the statistician. Finally, how to interpret the results? Whatever the results we got, how to interpret them? All these things can be done by a biostatistician. Remember, biostatistician is the most critical guy in the project. I'll tell you why. If I ask you in the SAS, even in the SAS also, maybe it is not that relevant, but at least I can tell you. If someone asks you, hey, this is my project, my end outcome is going to be, I'm looking for all the people who suffered with COVID and who got recovered. Just two things: who suffered, who got recovered. That's it. Being a developer, I follow one method. We have 15 more people in this particular group. Each of us may go with the different objectives, or may go with the different methods, or 2 or 3 of us may follow the same one. Maybe simple example. If I ask you, hey, can you give me how many number of observations in a data set? This is the question that I ask. 2 or 3 of us will simply go ahead and will say that, hey, I use the proc SQL and I calculate the distance count. Someone else will say that I don't use proc SQL. Instead, I'll go with the data step. In the data step, I'm using the N option and I'll calculate it. I feel it is more efficient. Someone will say that I don't even go with that particular one. I'll see what is the maximum value of underscore N underscore, and based on that, I'll calculate. Correct? Someone will say that I don't even go with all this. I simply go with the proc frequency. The proc frequency, whatever is going to be the cumulative number, that number I'll take it as my outcome. So 4 different methods, right? But the end is going to be the same. The end result is going to be the same. Each one of us is going with a different procedure. Same goes with the biostatistician as well. Biostatisticians doesn't have a standard approach for the given solution. Let's say you and me, both of us are 2 different biostatisticians. My approach is entirely different from your approach. So when the project started, I have provided one approach to you. So the investigator started the process and it is going on. I entered into the phase 4. When I entered into the phase 4, I decided that I don't want to work with this particular company. I left the company. So a new biostatistician has appointed. But the new biostatistician, there is a very minimal chance that he will continue with what is left by the previous biostatistician, because it is very difficult to understand what is the approach he has used. So what is going to happen? The moment I taken the charge of biostatistician, I will give my own approach. When I give my own approach, what is going to happen? Everything is going to change. So whatever the previous approach was there, that completely lost. I don't continue with that particular one. I will go with a new approach now. Right? This will increase, or this will make delay in the deadline, because whatever was done till now is gone. Is gone. I need to start freshly from here. I will change the random variables. I'll change the population. I'll change the deliverables. I'll also change the analysis methods, and I'll also change how I'm going to implement that. So the biostatistician is the one who is very crucial. Because of biostatistician, most of the time the deadlines will change. Clear? Right. The next, data management team. We can also call it as a clinical data management team, which will be led by clinical data manager. So a clinical data manager is the one, right, who is primarily responsible for case report form. I'm going to show you how it looks like, maybe, but not today. In the next class, I'll give you a simple CRF, case report form. He is the one who is responsible for your database design. He is responsible for your data entry, the validation and discrepancy management, medical coding, data extraction, and database logging. You can see that all these are almost related to us. You need to say that from here. Correct? Right? They will design the database, they do the data entry, they do the validations, they also do your medical coding, and they also complete your data extraction, and they log the database. The moment the database is logged, from there, clinical SAS programmer can take the charge. But on a whole, a data management guy is the one who will be primarily taking care of the CRF. Right? There is a normal CRF and eCRF. I'll give you the sample template of this and how a CRF looks like in the next class. All right. In addition to that, we also discuss about these and all common terms. You can pretty much understand this by this time. What is called database design, what is called data entry, what is going to be the data validation, discrepancy management, what is called your medical coding. Medical coding is not related to us, and how we are going to extract the data and database. All these are part of your data management team. Medical writer, a very crucial role, because this is the guy, whatever the outputs that are created by the SAS programmer, being a SAS programmer, we are going to create a table or a listing or a figures. Correct? We prepare this and we'll give it. But the person who is on the other side, that is the people who are going to review your data, the people who are in the FDA, they are not the SAS programmers. Correct? They don't know how to read it. Correct? So what they need to do now, since they are not aware of this particular one, how to utilize the SAS tables, listings, and figures, then this particular medical writer comes to the picture. This guy who is going to produce a scientific document, and that scientific document will be viewed and used for the entire research. Right? This is the guy who is going to utilize the tables, listings, figures created by us and prepare a meaningful scientific document. Right? So we need to understand to whom and all he will collaborate. A medical writer, he will work with doctors, he will work with the scientists, he will work with the SME, subject matter experts, and then he is going to create a clear research result, product use, and the medical information. All the things. This document is the final document, right? And also he need to ensure that it is not a very easy thing, because whatever the document he is preparing based on the tables, listings, and figures need to be in compliance with the regulatories. Either it can be EMA or it can be FDA or it is going to be a drug control, whatever it is, whatever the guidelines that they have provided, the document that is going to produce should be in compliance with this particular one, right? And also, while he preparing this particular one, he closely worked with your biostatistician. He closely worked with the SAS programmers, because SAS programmer is going to develop this, but the input for the SAS programmer will be coming from the biostatistician. So a medical writer who is going to consume our output will frequently in touch with the SAS programmers as well as with the biostatisticians for better understanding of the results. Can I have a quick confirmation? All of you are able to understand this? Is this clear? What are the cross-functional roles, or what, to whom and all we are going to collaborate? So what are the cross-functional teams that we are going to work on? Sponsor. Maybe we don't get direct interaction with the sponsor. It is very hypothetical, very vague scenario. You are going to get chance to work with the sponsor. Second one is going to be the investigator, or we can also call it as a clinician. That is the person who is going to take care of the patients or subjects, or who will carry out your clinical research. The third thing is going to be your biostatistician. A biostatistician is the one, primarily speaking, he is the one who is going to provide the logic to us. He is the one who is going to say, who is going to provide the algorithms to us. He is the one who is going to give the details about what is going, how the data looks like, and what are the various analyzing patterns that I need to follow. We can look from only from the SAS perspective. In that case, I can say these three. He will also help us to validate the data. Then there is going to be a clinical data management team. There is a manager who will be taking care of the preparing the case report form, right? And also he will be responsible for the database design, data entry, data cleansing, and also database. And then we have someone called medical writer. The medical writer is the one who is going to utilize the outputs that are produced by the SAS programmers. Those can be tables, those can be your listings, or those can be figures. He will utilize those particular things, and he will be in compliance with the regulatory guidelines, and he will prepare a scientific document. That scientific document is going to be the final document which will be produced to the regulatory authorities. While preparing this particular document, medical writer closely work with the SAS programmers and also with the biostatisticians. Is that clear, guys? All of you? Right. So can I continue with the definitions, or can I stop here? It's up to you. Can I go with some of the definitions, or we can pause it here and we can continue tomorrow? It's up to you guys. Anyone? Can we do it tomorrow? Subhasri? Okay, fine. Okay, let me set up the base for tomorrow. Tomorrow, guys. Tomorrow we have the class. I'll go with the class tomorrow also. So I had some plans to go to my native. That's the reason the day before yesterday I told you that Monday and Tuesday I'll go to the class, and I don't take the classes for the next 2 days. But tomorrow also we have the class same time, 8 p.m. Okay. So what are going to be the things that we are going to discuss tomorrow? Let's set up the base. We will continue with the remaining elements. At the same time, we will understand some of the definitions. The definitions include, let me write it here. First, we will go with the study design, the most important one. there are two study designs. Number one, parallel study. Number two, crossover study. So what is this parallel study and crossover study? We are going to discuss. Number two, once we complete this, we will go with the blinding. Blinding is nothing but masking. So there are three types of blindings: single blinding, double blinding, triple blinding. All three we are going to discuss with simple examples. And then we need to understand what is called site. That is very straightforward only, but we need to understand what is called site. In addition to that, there are few more. I can say that what is called multi-center study, what is called multinational study. We also talk about what is called unblinding. Sometimes we go with unblinding also, no blinding at all. We will discuss that. And then we talk about ICF screening and evaluation. So maximum how much time we can spend there, we can spend on the definitions. Definitions and protocol is done means we can happily enter into the testing. Right? So I expect or I believe that by tomorrow I can complete these definitions so that we can start with the testing. So this is the objective for the tomorrow's class. Clear, everyone?
Hello, hi sir.
Yeah.
Yes sir, can you share me this PDFs everything?
Which one? Yeah, I will share it with Bolusar today itself, right? So just have a word with Bolusar, and so I'll share the protocol, I'll share the template, and also I'll share the slides. This one.
Okay.
All these I'll share. Okay.
Yeah, thank you.
Thank you. Hey, hello everyone. Good evening. Welcome. Hope all of you are able to hear me and hope all of you can see my screen. Can I have a quick confirmation, guys? Wonderful. So I think after 4 days we are meeting, correct? Last class was on Wednesday, correct? Followed by today we are meeting. Sure, no issues. Hereafter there won't be these many number of breaks. So it will be continuous. At least we can expect 4 sessions in a week for sure. So it must be 5, but at least you can expect 4 from hereafter. Okay. All right. So before we start with today's session, let's have a quick recap about what we have discussed in the previous session. Okay. Before we go with that particular one, guys, all of you have received your what to say, the protocol, the sample protocol that I have shared, as well as the exam board that I have shared. All of you received that? Yeah. So any questions or any doubts in that, guys? Anyone? So all of you are clear, right? So can I proceed from here? Okay, perfect. All right. So let's proceed. Let's have a quick recap. Oh, Sanchi, you haven't received. So just ping Bolusar once and get it from him. Okay. Yeah. So even Mohanty is the same have with you as well. Just have a word with Bolusar and get your this PPT as well as your protocol. Please get it done. Okay. If not, what I will do is because anyways I have your Manimaran, I'm not sure. Guys, whoever have received, well and good. Whoever is not received yet, just ping Balisa and get it done. Clear? This is to everyone. Perfect. Cool. All right. So what we have understand here, last week we started discussing about the clinical trial protocol, some of the definitions as well along with that. So we all understand that clinical trial protocol is the very first thing which is prepared either by your sponsor or what is the other thing? We also have a research organization. You can also call it as a contract research organization. Any one of these two guys will prepare this with the help of a biostatistician as well as the clinician. This is what we discussed initially. And then the why the protocol is important is it is going to describe how you are going to conduct your trial, what are the objectives, study design, the methodologies, what are the various considerations which will fall under the statistical studies, and also who is going to provide the level of guarantee for your safety, and at last but not the least, the most important thing, who is going to maintain or how we are going to maintain the integrity of the data that we have collected from the subjects. Correct? So all these are going to be part of your clinical trials protocol. So typically a protocol will contain all this. We have seen this. Few of them we have already discussed, correct? The background, the study rationale, the study objectives, we have discussed about that. In addition to that, we also had a small discussion about who and all the various people with whom we work, maybe at a team level, I can say that. All right. So initially we thought like we go with sponsor. A sponsor is the person who is going to provide the funding to us. A clinician or investigator, or even the principal investigator or the senior investigator. All these are the guys who are involved in your clinical studies, who are responsible for investigation, like how the investigation is conducting and all. Correct. And also we discussed that a biostatistician is the one who is going to provide or who closely work with both investigator as well as the programmer. And some of the key responsibilities are going to be he or she are the one who is going to provide the statistical design, hypothesis, what is going to be the optimal size, the randomization, data collection process, and also how to interpret the results. But we also discussed about that biostatistician is the key person, right? Because they don't have any standard approaches, right? So what is going to happen? Each biostatistician has its own way of dealing with the study. Let's say in the middle of the study, if biostatistician decided to leave the trial, obviously the new biostatistician who we are going to recruit may or may not able to follow the previous biostatistician's methodologies. So what is going to happen? So the deadlines are going to be increased because we need to kickstart or we need to restart our entire methodology, because no two methodologies are going to be same. So that is the reason we can say that one of the reasons why the clinical trials deadlines are going to be extended is because of the biostatistician. One of the reasons, but main reason, correct? And we also talked about data management team who provides, I can say that everything. I can say that the raw data. Right, we are going to discuss that in the SDTM. The very first step is going to be the raw data. So the data, the clinical data management team, you can simply call it as a CDM. This is the team who is responsible for all your data related, raw data related stuff. Correct. And we also have a medical writer. The medical writer is the one who is going to interpret the details provided by the statistician, SAS programmer, and also he is going to convey this in a scientific way who performs the research. Correct. This is the he or she are the person who closely work with, I can say, all the entities who are involved in your clinical study. Correct. So the medical writer is the one who is going to prepare all the reports which should be in compliance with the regulators. That can be FDA, that can be EMA, that can be any regulatory authority which comes under the particular. Correct. So these are the cross-functional teams that we have that we have discussed in the previous class. So today we will continue with the remaining definitions. A few definitions. Today is going to be the last day for the definitions. By tomorrow we are going to start our SDTM. So we are going to start SDTM tomorrow. So today we are going to complete the entire base. Mean to say that what are the other definitions we would like to understand before we proceed with the proper SDTM, right? Before we go with the implementation, we just need to understand these. First, study design. One of the most important thing when it comes to your clinical trials is going to be a study design. There are so many study designs available. Means each study design you can consider as a one method. There are so many methods available, but we specifically deals with two. Before we understand what are the two different study designs, we need to understand why should we have a study design first, right? So again, we go back to the previous statement. What is the previous statement? We all know that any drug before it comes to the market, the sponsor who is actually funding this particular drug need to provide all the information about all the controlled clinical studies he or she performed so far to make sure the drug is effective, to make sure the drug is safe. Correct? So the main reason why we are going to the clinical trials is I need to make sure that whatever the drug that I'm bringing to the market, that has been developed under the well-controlled clinical studies and it is effective. Correct? To know that, to maintain that particular one, right? A study that is well controlled is designed to compare two or more groups of people with at least one of the group is taking investigational drug, or often we can call it as a study. So you might need to understand first what is called investigational drug, or you can also simply call it as a study drug. So there are two things first: study drug and normal drug. A normal drug is the one which we all take when we are sick. That is the drug which is readily available in the market. So what it is? It already gone through all the clinical studies. It got approval. That is nothing but the regulatory authorities are already approved this particular drug. It is safe and it is effective. So it is available in all your medical stores and hospitals. We can get it based on the doctor prescription and we can have it. That is called your regular drug. Then the study drug is nothing but the drug which is under study. What do you mean by that? Under the development process. It is still in the clinical trial. Correct? So what do you mean by that? The study drug is nothing but the drug which is actually undergoing through the clinical trials. Right. So that is the key difference. The investigational drug or a study drug is the one which is actually being developed under the clinical trial. So there's supposed to be a study. The study need to tell you how this drug is going to give to two or more groups. That is, we have subjects. Subjects are nothing but the patients, right? We are going to give this particular drug to the patients, right? So all these subjects, first we are going to make them as the groups. Let's say I have taken 100 subjects. Out of the 100 subjects, 50 people are going to be the healthy people, and the remaining 50 people are actually the patients. I hope you all remember this, correct? So in each study or in each phase of your clinical trials, we are going to pick the subjects randomly. While picking the subjects, we understand that we are giving this particular drug to the healthy people, nothing but the volunteers. At the same time, the people who are already suffering with the same disease are the people who are suffering with the other disease. Correct? All these three groups are part of our clinical studies. Hope you remember that particular one. We discussed this already. Correct? And again, out of these 100 subjects that we have taken, some of them are male and some of them are female. All these randomizations we are going to take. Now, after we picked up our volunteers, that is nothing but once we have finalized the subjects who are going to undergo this particular clinical trials, again we split them into two groups. Again we split them into two groups or two or more groups for better understanding of the drug efficiency. All right, so that grouping, how we are doing that particular grouping, again in each group what kind of drug you are providing, these are decided by your study design, right? The way the groups get compared varies depending upon the study design. So what study design you are choosing, based on that these groups will be compared. I told you right here we are segregating the groups, right? So these groups will be compared based on your study. So there are two studies available. One, mean to say there are more than two, but we are specifically go with the most commonly used studies are going to be parallel study design. The second one is going to be crossover study design. right? Again, out of these two, each one has its own advantages as well as the limitations. But which study design I need to create will be decided again by your biostatistician, your investigator. It is as part of your protocol. There are multiple elements which we are going to consider before we go with the design, right? Which design I will take, right? But in general, the most common study designs that we are going to use are either we follow your parallel study design and crossover study design. In a simple way, in a nutshell, if someone asks you, hey, what is called parallel study design? If I ask you that, a parallel study design is the one where, let's say there are two patients. I told you there are already two groups, correct? Let's say there is a group A, there are a bunch of patients in this particular group, and there is going to be group B. For an example, for our ease of understanding, let's say that group A have only one patient, 101. Group B has only one patient, which is nothing but 10. Throughout the clinical study, I mean to say that we have phase 1, phase 2, phase 3, phase 4, right? Throughout the design or throughout this particular treatment, this particular guy, 101, is going to get the same drug. Let's say he is giving birth a drug A, right? In phase 1, in phase 2, in phase 3, in phase 4, on all the phases, he is going to get the same drug. The same drug is going to carry out in throughout the entire design, throughout all the phases of your clinical trial. There is no change in the drug in all the phases, right? For one treatment, one drug throughout. Is that clear? Let's say there is another guy. Here I have patient 2. For him, we have given drug B. The same drug we are going to give it to him till the end of the treatment, till the end of your clinical trial. Is that understandable, guys? What is called parallel study design? One patient for one treatment, one drug throughout. Any questions here? Okay, let's see this, how it looks like. Here it is. Maybe for illustration purpose, you can also consider this. See, can you see this one? All we are doing is in parallel study, each patient is placed into one treatment arm. Treatment arm, don't worry about the arm. Arm is nothing but a treatment or a group. Okay, a study arm may consist of the study treatment, placebo, standard of care, comparative drug, different doses of the study treatment. This is bit in depth into the clinical research. Don't worry about it. In a simple way, you can say that they will remain in the treatment arm throughout the study. This, if patient A is giving a placebo throughout the study, we are going to give placebo only to him. If the patient B or the subject B, we are giving a normal drug, an investigational drug or a study drug. Throughout the study, we are going to give the drug B only, the study drug only, in a simple way. Okay, so let's have a look why and how it is effective or how we are going to use it. This is most common. Patients are randomly assigned to the treatment groups. Mean to say that I told you we are going to segregate them into different groups. Once the randomization done, let's say there are two groups. In group A, I'm going to put one guy. In group B, I'm going to put the other guy. Fine. Right? So each treatment arm include a particular dosage of the study drug. Let's say I'm giving 50 mg of the tablet to him. Means throughout the drug, throughout the study, I'm going to give 50 mg only. I don't increase the dosage. If I'm giving placebo, throughout it is going to be placebo only. Or if it is going to be, let's say that some oncology related stuff, throughout it is going to be the same stuff, right? You can see the patients then remains in the same treatment arm throughout the course of the study. If someone asks you, hey, can you explain in a very simple way in layman terms, I can say that subject will be exposed to only one treatment and the same treatment will be continued until the end phase. Is that clear? One treatment throughout. Now, what is the other way? What you can guess for the crossover study design? This is parallel study design. Now, can you guys tell me what you can expect from crossover study design? Anyone? Tejashwini, 1 second. I got your question. Can you explain again, please? Sure. Tejaswini, did you get it, or you want me to explain now? Because I have already done second time, but that doesn't stop me to explain one more. So did you understand, or you want me to go through one more time? Sure, again, fine, I'll do that. So parallel study design. So don't think about the groups or anything. In a simple way, let's say we are testing a simple drug. Maniraman, wonderful. Yes, I'll just hold you for a minute. Once I'll complete the parallel study design, again I'll come there. Okay. In a simple way, in a parallel study design, let's say I'm just thinking about a drug for treating headache. Headache is the one which I want to treat. For that, the sponsor has given all the funding and everything. So the clinician and the investigator has developed already, they have developed a drug. And the drug name, let's say that in a simple way, it's a dark tablet. Dark, and let's say that 200 mg. This is the tablet. So this will become your study drug, because this is the drug that we are actually developing now. So what we do is, and there is something called placebo. That is, the second drug is placebo. Now, so we have two different groups now, or two patients now. Let's say patient number 1, Murali. There is another patient, Krishna. So we decided that we want to go with a parallel study design. So what we do is, let's say we have 4 phases. We have phase 1. Let's say that pre-clinical trials is done. So it's phase 1, followed by phase 2, followed by phase 3, followed by phase 4. We all know that there are 4 phases. So what we do is, in phase 1, we'll give drug to him, or to Murali, which is of 200 mg. So where we are giving this particular drug? In the phase 1. Phase 1, what we generally do, we test the safety of this particular drug. We understand that this drug is safe enough. So we'll move to the phase 2. In the phase 2, generally what happened, we are going to increase the number of people who are involved in this particular one. In phase 1, we have 30 to 80 people, but in phase 2, we have 100 to 500 people. But out of the 500 people, Murali is still there. For him, even in the phase 2, we still giving the same drug with the same dosage. No changes in that. Still the same. Again, here we understand the efficacy of this particular drug. So we understand that the drug is working efficiently. Then we move to the phase 3. In the phase 3 also, we increase the group capacity from 1,000 to 5,000 people. That is, number of people who are participating in the phase 3 of the clinical trial increased from 1,000 to 5,000. But still, Murali is part of that particular one. And here also, I'm giving the same drug with the same dosage. Here we are testing both the efficacy as well as the safety of the particular drug, both of them. So phase number 4. What is phase number 4? Here it will go to the 10,000 to 50,000 of the people. Here all the parameters like how to do the marketing, pricing, everything will come to the picture. But here, in this case also, I'm giving the same drug. My treatment is done. My clinical trials, all the phases are done. Phase 1, 2, 3, 4 are done. But throughout these 4 phases, I'm giving the same drug, same dosage to this particular guy. Similarly, there is another guy, Krishna, who is in another group. For him also, I'm giving the placebo to him. So in the phase 1, I'm giving the placebo. Phase 2 also placebo only. Phase 3 also placebo only. Phase 4 also placebo. No change in that particular treatment. No change in the dosage. Tejaswini, is that understandable now? Let me know if you're not able to follow. I'll give you the different answer to this. Got it? Perfect. Cool. So if someone asks you how to put it in a data set, I'll tell you how we are going to get the data. Now, let's move to the crossover study design. A crossover study design is the one, as Manimaran already mentioned in this chart. Same subject received 2 treatments with an adequate washout period. Wonderful. So you have brought up 2 things. 2 different treatments. It can be 2 or more. It can be 2 or more treatments At the same time, you're also talking about the washout period. I'll explain both of them. So let me go to the PPT. So here in the crossover study design, this diagram will explain you what it is going to be. So here I have patient one. Patient one has given arm A initially, then arm B. Again, arm B, that is the patient two who is part of the arm B, that is in the drug B, again provided to the drug A. That is, in crossover study, a patient will start with one treatment initially and can switch to any other treatments during the study. Say, I'll take the same example that I have given here and explain that. Yeah. Let's say that in crossover study design, Murli we have given DART 200 mg tablet to him during the phase 1. When it comes to the phase 2, instead of giving the DART, they have simply given placebo to him. And phase 3, again, we continue with, let's say, placebo. Let's say that again here also. But again, when it comes to the phase 4, we started giving the DART. Similarly, let's say for Krishna, we started with placebo. Then we have given DART, then again we have given placebo, then again we went to DART. Meaning what is happening is the same patient for the same treatment, he or she will undergo different drugs. It is not that the patient will have the same drug, same dosage throughout the course. No, the same patient will get different drugs during the study, during the different phases of the study. There may be a chance that we can also increase or decrease the dosage as well. Is that clear? Each patient can receive more than one treatment or more than one drug throughout the course. All of you are clear? First of all, what is called crossover study design? Then I'll tell you about what is called washout period. Because washout period is the other thing, because even in this particular diagram you can see that there is something called washout period, right? So before I explain what is called washout period, all of you understand what is called crossover study design? Can I have a quick confirmation?
Murli,
Yeah.
I have understand the crossover, but I have one question. So in the phase 1, we are looking for the safety, right? In the placebo, it's like no drug in the placebo. So how they can check the safety in the second person in the phase 1?
Okay. First thing. So for that we need to understand what is called placebo first and how it is different from your investigational lab. Correct? Second, see, we are going with the two different people first, two different groups. Correct? So here I have taken only one person in each group, but in real time, in the normal design, as I have given you the other example, I have picked up 100 people. Out of the 100 people, 50 of them are healthy, 50 of them are already suffering with this particular disease. Again, in the group A, let's say consider this, Murli is under group A. So Krishna is under group B. Along with Murali in the group A, there are 49 more people. Correct? Similarly, along with Krishna in group B, there are 49 more people. It doesn't mean that for all these 50 people, I'm giving placebo in the phase 1. No. For few, it is going to be DART 200 mg. Let's say here is someone called Anu. Anu is taking DART 200 mg tablet in the phase 1, whereas Krishna is taking placebo in the phase 1. So, but both of them are falls under group B. I'm taking the randomization, I'm taking the entire group into consideration before I take any step. It is not on single person. You understand that placebo is nothing but it is not a original drug. It is not the actual drug. It is a fake pill, correct? So based on the fake pill, as you mentioned, you cannot determine or you cannot assess the safety. But you are not assessing that just based on Krishna. You are assessing that based on 50 people who are randomly getting this particular. That is the reason we go with the random distribution. Shubha, have I answered your question?
Yeah, got it, Murali. Thank you.
Perfect. All right. Now, guys, you might feeling that Murli, what is called placebo? You are saying it is a fake pill, because the people who are not coming from the clinical background will not have an idea about what is called placebo. Don't worry, I'll explain this in the later slides. Okay. All of you understand what is called crossover study? A crossover study design is the one where each patient or each subject undergo more than one treatment or more than one drug. Sorry, more than one drug. All right. Now, any questions? Let me know. I'll give a pass for a minute. All right. Now, what is called washout period? Let's say here I told you each patient, maybe I can say that, let's consider this. So I have DART 200 mg, which I have given in the phase 1. But when it comes to the phase 2, I increase the dosage to 300. Right? I want to check the efficacy, means how efficient it is. Let's say if my body already have this, let's say that in phase 1 I have taken this particular drug. Let's say the phase 1 went for 10 days. So for 10 days, every day night, I had this particular tablet of DART 200 mg every night. So my body is already adapted to this particular drug, correct? So this drug particles are still in my body. So every drug has some effect in our human body, correct? Few drugs have, that effect maybe for 1 hour, maybe for 2 hours, maybe for 4 hours. Few drugs may have that effect for 1 day or 2 days, correct? How long the drug have effect in our body, right, is nothing but your effective period, or how long the drug is actually working on your body in a simple terms. Mean to say, generally, let's say the same, let's say you're suffering from fever. Doctors generally prescribe that, here in India, most of us used to take this particular drug, Dolo 650 mg. Correct? Dolo 650. Doctor will say that for the first 3 days, since you have high temperatures, take this particular drug 4 hours a day. Sorry, 4 times a day. Mean to say, for every 4 hours, take this particular one. So right from morning, again 4 hours, again 4 hours, again 4 hours. So that before you sleep, you take 4 times this particular drug. What is the meaning of this? So generally 8 hours is going to be the sleep time. The remaining 16 hours. Mean to say, this drug is effective for 4 hours. Can I say that? It can fight with all your, the virus or the bacteria. Because of that, you got the fever. It can fight with that particular one effectively for 4 hours. After 4 hours, this effectiveness will go down. So to fight with your virus, again you need to take this particular drug. Again, doctor will say that after 3 days, if your temperatures are down, then take this particular tablet just 2 times a day. Mean to say, the drug is effective for 8 hours. Correct? Morning once you are going to take, or let's say 12 hours. Morning once you are going to take, again before sleep you are going to take the other one. So we know that for each drug, how effective it is and how much time it is effective. Correct? So if I want to test 2 different drugs, or if I want to test 2 different dosages, I need to ensure that the previous drug does not have any impact remaining in our human body. Mean to say, let's say after 4 hours, this drug has no effectiveness. So if I want to test another tablet, let's say I had Dolo 650, my temperature is not going down. Immediately I cannot take another paracetamol. If I want to take another paracetamol, at least I need to take for the 4 hours. This 4 hours is nothing but our washout period. Mean to say, how long I need to wait before I can take the other drug, or before I can take the same drug, how much time I need to wait. Is that clear? If I want to talk technically about it, I can see that. Oh, here it is. See, the washout period is nothing but it's a predetermined amount of time during which patient receive no treatment. This during that particular time, because my drug already there, so I won't take another drug. This period reduces the carryover effects from the previous treatments. So I already had a drug. So if I want to test a new drug, I need to wait. Otherwise, what will happen? My previous drug is already working on my body. I'm going to take another drug. I don't know how my previous drug is going to react with the new drug that I'm going to take it. Or my new drug may not be effective because my initial drug that I have taken already, which is still working, right? So I cannot estimate the efficacy of the new drug as well. Am I correct? So what is called your washout period? This is the period during which I don't take any medicine. I won't get any new treatment. I need to wait till my body is not responsive for the previous treatment. Is that clear, everyone? All of you understand what is called parallel study design as well as your crossover study design? Can I have a quick confirmation, guys? Any questions? Anyone? Right. In simple, this is your data set. Let's say in parallel study design, I have subject ID, which is nothing but the patient ID. Arm is nothing but your treatment. ACTAM is nothing but what exactly the treatment that is happening there. Actual treatment, actual drug. So 101, so drug A, drug A. So 102, placebo. Oh, this is wrong. 102 is wrong. Throughout, it is going to be the same one. So 102, 103, 104, all throughout the drug, throughout the treatment, the patient is getting the same drug. Crossover study design, what is going to happen? Let's say drug A I have taken, I wait for the washout period, then I take drug B, again I'll wait for the washout period, then I'll take placebo. So what is happening here? Here simply we are waiting for, we'll take one drug, we'll wait, there is no impact on our body, that is nothing but washout period, we'll wait till that, and again we take it. Clear? Simple illustration how you are going to get it in your data set. Everyone? All right. Now, a very few terms we need to understand, a few definitions, not very in detail or anything, few definitions. We already know sponsor, we already know the subject, so I don't go with that. Principal investigator also we know. Study coordinator. This one we haven't discussed. The study coordinator is the one, this guy closely work with your investigator. That is nothing but, the investigator is nothing but the doctor. The doctor mainly taking care of the drugs, how the body, how the patients are responding, all this. So there is going to be an assistant to this particular senior doctor or senior investigator who will help to proceed or who will help to conduct your clinical trials efficiently. Remember, not all the clinical studies can have this study coordinator. You can have it, you cannot have it. That's up to the budget, that's up to the sponsor and CRO. Site. Because this terms we need to understand, because hereafter we are going to use them. That's the reason I'm going with it. Wherever your clinical trial is happening, that can be a clinic, that can be a hospital, that can be any medical institution or academic institution, or it can be a doctor's office. The doctor is nothing but a research doctor, even in your home. Home is hypothetical, but still there is a possibility. Wherever the clinical trial is happening, that place you can simply call it as a site. Next, a multi-center study. A multi-center study is nothing but, let's say, you have a single protocol. There is no change in the protocol, only one protocol, only one sponsor, right? But this particular clinical trial is happening at more than one place by more than one investigator. Let me go with an example. We're just going with, let's say we have a sponsor. For an example, the sponsor is in Mumbai, right? Just say that the sponsor is in Hyderabad, the Reddy's laboratory. The Reddy's laboratory decided that we want to build a new drug. But let's say they have their offices in Chennai, they have their offices in Mumbai, they have their offices in Delhi as well. So how many offices? 4 different offices. All the 4 different offices has the research facility. So what they have decided is, we take 50 people from Chennai, 50 people from Mumbai, 50 people from Delhi, 50 people from Hyderabad. The protocol mean to say what clinical trial, what design we need to go with, what treatment we need to give it. All of this is fixed. So in 4 places, they have 4 different doctors, nothing but 4 investigators. Each investigator is going to follow the same protocol strictly, and they are going to continue with their clinical. If this happens, you can call that as a multi-center study. Is that clear? But one thing you need to remember, a multi-center study can happen within a single nation. You can say within the same country. If you're doing this in India, means all the centers must be in India only. If you are carrying this particular one somewhere in your other country, all the states that you are referring, all the centers you are referring should be falls under the same country. Can I have a quick confirmation? All of you understand what is called your multi-center study? Yes or no? Perfect. The next one, multinational study. The name itself is telling you multinational. Just the extension of your multi-center study. That is, here also we are carrying out our clinical trials at more than one place, but the place is not restricted to a single nation. That can be anywhere in the world. Mean to say, you can carry your clinical trials in India. You can carry parallelly the same protocol you can implement with a different investigator in USA. That is, we are testing this globally. If that happens, you can say that it is a multinational study. Right? Then, placebo. So we have been talking about this placebo multiple times. Remember, placebo is not the original drug. Placebo is not at all a original drug. A placebo does not have any active properties. Mean to say, let's say let's say generally, actually the placebo is the concept which has been, actively used by who is this? Psychologists. Psychologist is the one who most frequently use this particular one. Let's say that one patient have visited a psychologist and say that, hey, I'm not getting sleep properly, right? I have a difficulty in getting the sleep. So the psychologists understand that this guy has some psychological related stuff. There is nothing, or there is no proper drug for it, or there is no need of a drug for it. So what he will do is, he will say that, hey, everything is all right with you, but you require some drug. I'll write some drug, some prescription to you. Carry on that particular one, right? Let's say that every night before you sleep, take this particular drug. Means one tablet I'm prescribing to you, take that particular one. So it will help you. It definitely help you to get sleep quickly. So you can say that he will give you a commitment that within 5 minutes after you have this particular drug, you'll get sleep. But the actual drug is, it may not be a drug at all. It can be a normal sugar pill. Mean to say that it's just a sweetener. It can be a normal, you can say that a polo, equivalent to polo. It is all dealing with the psychological things of a patient. The patient will be in an assumption that our patient will strongly believe that doctor has prescribed a very nice drug. If I take this particular one, I definitely get a sleep. With that intention, with that belief, the patient is going to have that particular tablet, and obviously 80 to 90% of the cases, he or she may feel sleep as well, right? But actually, it is not a original drug. It is just a fake pill. It is just a fake pill. Is that clear? Why we do this particular one is, let's say we have a group of people, right? We have healthy subjects as well as we have the patients who are already suffering with particular disease, right? Every time the investigator is not interested to give the drug to the particular guy, they want to make sure that we'll give a drug for few of the people. We also give this placebo for a few of them. They will be an assumption that they are actually having a drug, but they are not having it, right? This is generally used for efficacy purpose as well, right? It is a random thing. Is that clear? You can also, I just also mentioned this here. If I believe the pill will help, it will help. Often patient's cycle, physical condition does not change, but it feels like it has. It doesn't have any effect on the physical things, but patients believe that it actually has an effect. Clear? Right? Because maybe you might heard of this particular one, right? A good doctor is the one who will give enough confidence to the patient so that the doctor words itself is going to be work like a miracle, right? For few of things, it really doesn't require a drug. A good doctor can treat the people with the warm words. Clear? So placebo will fall under that particular. All of you are clear up to this? Any questions? Because I have been using this particular word placebo again and again. So all of you must know what is called placebo. Placebo is not the original, or it is not. It is neither a study drug nor a investigational drug. It is simply a fake thing, a fake pill. Questions? Any questions from anyone? Wonderful. Let's go with the another most important thing, which is nothing but blinding. We can also call it as a masking, because your protocol will mention it is a single blinded or a double blinded or a triple blinded one. Sometimes your protocol will say that it is no blinding as well. What is called blinding? In a simple way, masking. Mean to say, I'm giving a drug, right? I'm just in the clinical trials, we are developing a drug. So what exactly this drug is about? How much dosage I should give? What is the composition of this particular drug? What are the side effects of this particular drug? Can I say all this together is nothing but a drug information, right? So now, who and all involves in your clinical trials? I can say the patient. So first one is going to be patient, that is nothing but subject. The second one is going to be an investigator, doctor. Now, who is the third one? Here you can see that the team, that is the team who is collecting your data. If all of them knows about what drug we are giving, what is the composition, everything, if all of them knows about it, yes, this guy knows about it. Investigators also know about what is called the drug composition, side effects, and everything. And even also the data preparation team is also know that what drug and all. If these three are yes, all the people who involved in this particular clinical trials are well aware of the drug and its information, it is called unblinded. I haven't masked anything. It is open to everyone. You understand what is the first thing? What is the first thing here? If blinding, first one is unblinded. Whoever is participating in this particular clinical trials, all of them aware of what drug we are taking, what is the side effect, everything, every piece of information about the drug, they are well aware of. Clear? Second one, single blinded. What do you mean by single blinded? A single blinded is the one where only the patient is unaware of the treatment. This guy don't know what treatment he is getting. He is volunteered for this particular treatment, or he is the patient who is undergoing this particular treatment, but he is not aware of what exactly it is. A simple example I'll tell you. Let's say you're not well. Doctor identified that there is some cyst is part of your stomach and it need to be removed. The doctor has diagnosed something, he is going to proceed with some procedure. Only these two you know. But what procedure the doctor is doing, right? What are the various vital reports, what are the various vitals that he is taking as part of it? That is nothing but vital measurements and all. All these things you are not aware of. Let's say you are undergoing surgery, you just got an anesthesia dose, you are simply just lying on your operation room, correct? The doctor is the one who knows about what treatment he is doing there, what procedure he is actually doing there, and also how, let's say that you want to make a cut of 9 centimeters. You know precisely 9 centimeters exactly at what place you want to make a cut. You are not aware of it, and you don't need to know about that as well. All you need to worry about is the cyst need to be removed and you need to be proper. That's it, correct? But what exactly, how what is the length of the cut that is going to make, or the stitches are going to be like it is auto stitches, or it is going to be again you need to remove the stitches again, or is it a laparoscopic, what exactly it is, you really don't need to be, you really don't need to know about that particular one. So if you're not aware of that, mean to say that the subject is not aware of what treatment he is actually undergoing, then it is called as single blinded. Only one person is not aware of it out of three. Clear? Then double blinded. Here itself we can understand the participant and the clinicians or the data collectors are not aware of the treatment. Mean to say patient is 100% not aware of the treatment that is undergoing with. Clear? We don't know that. Now, either investigator doesn't know about it or the data collection team is not aware of it. Sometimes the investigator is not aware of it, but the team who is collecting the information who is aware of this particular one, right? So in this case, oh sorry, this guy doesn't know, but here this guy knows about it. So these two cases are the cases which falls under your double. Both of them, right? Patient is not aware of this, that is no doubt about it. Investigator and team, any one of them also might not aware of this particular one. That is called your double blinded. Triple blinded, by this time you might know about it. What is the answer for it? None of them are aware of it. You have a protocol, whatever is there in the protocol, strictly you're going to follow as per that. That's it. One of the funny thing is you can compare this one with your Coca-Cola secret recipe, KFC secret recipe, all this. Everyone says that, let's say KFC secret recipe, because it is actually preparing in your local store, the store or the restaurant near to you, correct? But whoever working in that particular restaurant, they have given a guidance that this particular one, this spice has to be put into this particular one. This need to be, you know, fried at this particular temperature for this much amount of the time. Each individual team know their instructions, but on a whole, no one knows what are going to be the different things that are there. They are part of this particular recipe. The same thing, but the end product is going to be good. Definitely, it is going to be finger-licking product, right? But individual persons doesn't know about it. They're simply following one recipe. So your protocol is going to be like that. A triple blinded protocol is the one. None of them knows that what is going to be the drug information, but end product at the end you are safe and the treatment is going to be effective. Is that clear? Can I have a quick confirmation? All of you are able to understand what is called your single blinded, double blinded, triple blinded, and unblinded. Any questions? I have also given you the what and all falls under it, the detailed information, just elaborate I have given to you so that you can refer later, right? Double blinded as well as the triple blinded, right? And few more terms here. ICF, this is nothing but informed consent form. Because let's say you're a volunteer for one of the treatment. Let's say that COVID trials are happening. Someone, let's say Bharat Biotech is actually developing a drug. You want to volunteer that. Let's say today you volunteered that, you went there, you taken the drug. They said that you have to be in this hospital, you have to be in this particular site for, let's say, 2 weeks. But after a week of time, you feel that you got bored. You don't want to stay there. You want to leave the site. They will not allow. But if you force to do that, someone has to stop you legally, right? Or in other words, you go there, you started getting the treatment. After 1 week, your sponsor said that, hey, I want to stop this particular one. So all of you who are under this particular site, you can leave. But you are already taken the treatment for 1 week. You volunteer this treatment for 1 week. It cannot be stopped. So there's supposed to be a legal terms. There's supposed to be a legal consent you need to provide to your sponsor. That legal document, which is signed by the subject, that is nothing but the patient who is actually part of this particular treatment, will be called it as ICF. You're giving your consent there. I'm okay with all these terms and conditions. Most importantly, the date will be captured there. Here, number 2, screening. This is again another good thing. Screening is nothing but you want to volunteer, right? But do you have eligibility or not? Do you have eligibility or not? Mean to say, you are going to get, let's say you're a diabetic. For an example, you're a diabetic. For few people, for few of the treatments, the people who are already suffering with the diabetic, we don't want to test this particular drug on these people because it may have less effect. It may have less effect, or it may have side effect. So can I say that there are going to be some inclusive criteria? There is going to be some exclusive criteria, correct? Let's say for this particular drug, I want to exclude all the diabetic patients. For other thing, let's say that there's another drug that we are developing. We want to do this particular drug. We want to have the volunteers who are actually having this disorder of diabetes. I only want diabetic people here. So there is an inclusion criteria, there is an exclusion criteria. Let's say that inclusion criteria, I'm going to have, let's say 10 things. These 10 conditions need to be met. Exclusion criteria, the subject does not have any of this. Only the persons, only the volunteers who are able to clear both exclusion as well as the inclusion list, only then I'm going to consider for my clinical trials. Can I say all these procedures together will be called as a screening? A very simple thing, correct? Even in a simple way, if I want to say, let's say you're having some allergy, you visited the hospital. Doctor has prescribed a simple injection, a simple one. Let's say penicillin itself, right? Few people, they are very reactive to this penicillin, correct? It may go with a severe allergy, you're going to get the rashes, all this kind of stuff. So what generally the technician will do? A technician will pick your arm or somewhere. He or she will simply inject a piece, a little, I can say that less than 1 ml. They are going to simply prick at your hand and they're going to give that particular penicillin. They will wait for 2 to 5 minutes. If you are not getting any rashes around that particular area, they will confirm that, yes, to this particular patient, we can actually give the penicillin, correct? So what they're doing there, they're screening you, correct? They're checking whether you're fit for this particular drug or not. The same thing is going to happen here. So there is going to be an inclusion and exclusion list. You need to get clear with this particular list, then only they are going to give this particular, they are going to allow you to come to enter into this particular clinical trials. Am I clear what is called screening? Yes or no? Simple terms, but we need to be very clear with these terms. Then only, once we start with the SDTM, we don't discuss about suddenly if we say what is called ICF, it's difficult to explain again. That's the reason we are going with all these things. Okay, next, randomization. Again, the same thing. Randomization is going to be at the beginning of today's class, I told you we are going to prepare some groups, correct? Though I have 100 patients with me or 100 subjects with me, all those 100 subjects are not suffering from this particular disease. Few of them are healthy volunteers, few of them are going to be the people who are suffering with the disease. So while we creating the groups, for few people we give placebo, for few people we are giving the drug that we are testing, correct? Now, based on these two, we are going to create two different groups. One group you can call it as intervention group. The other group you can also call it as control group. So there are two groups, right? So I'll tell you what this each group is basically for. All these 100 people, let's say I have 100 people with me, I cannot keep all the 100 out of 150 people always in the intervention, and I cannot keep all this remaining 50 people continuously in the control group. No, they need to be randomly interchanged, right? So that interchanging can be done based on their randomization. Random allocation is a technique that chooses individuals for treatment groups and control groups entirely by chance, with no regard of the will of researchers or patient's condition or preference. Meaning, in a simple way, a patient is part of the clinical trials. Though he is a volunteer, he is getting paid for it. He don't want to be in the control group. He want to be continuously in the intervention group only. I'll tell you what is this intervention. But patient want to have, he willingly want to have only in the intervention. That is called biased, right? He's biased. Let's say investigator, he don't like one of the person, one of the subject. So he want to keep the subject continuously only in the control group. He don't want that particular guy to be part of the intervention group at all. So the biasing can happen at the investigator side, or it can be happen at the subject side. But a good clinical trial is the one, it need to ensure that all my people who are volunteer for this particular clinical trial need to be part of both the groups. There is no biasing at all. There should not be any biasing. I need to ensure that they're randomly getting a chance, or they're fairly getting a chance, right? I can simply say that it's like a head and tail, right? All the patients need to go with both intervention group. They should be part of intervention group as well as the control group. That thing can be done with the help of randomization. You can see here. This allows researchers to control all known and unknown factors that may affect the results in treatment groups and the control group. Treatment group, you can also call it as a intervention group, right? So if someone want to say simply what is called randomization, each participant has the same chance of being assigned to either intervention or control. What do you mean by that? Each patient have equal chances of being part of intervention group or under the control group. That thing I'm doing with the help of my randomization. Let me explain you with this. So AKV, which is going to be a headache drug. So intervention group, control group, what it is? A control group is the one where they are only going to get a placebo. They don't get the original drug. They don't get the original drug. Intervention group or a treatment group or the group where they actually get the original drug, right? So generally what we need to do, if let's say that 50 people are continuously only in the placebo group, they're only in the placebo group, what is the meaning? You're simply wasting 50 people because you are not giving any drug to them. You are giving a sugar pill to them and they are part of the clinical trial. Will it have any effect? No, right? Otherwise, these 50 people are continuously getting the headache tablet every day. There is nothing but only 50 people are effective. How to get the efficacy then? So for that, what you are doing is you need to ensure that one patient, let's say morally, he should be part of control group for some time. He should be part of intervention group for some time. That randomization I need to provide. The control group should have mix of both patients as well as healthy volunteers. The intervention group also should have both healthy patients as well as the volunteers as well as the patient. So the group should be random. The group should be having equal preferences. So now do you understand what is called control group, what is called intervention group? Control group doesn't get the original drug. Intervention group will get only the original. Original drug is nothing but strategy. Can I have a quick confirmation from everyone? All of you are able to follow this? Yes or no? Any questions? Last but not the least, very simple ones. Treatment. We're going to use the acronym TRT, which is nothing but treatment we are giving to the subject. Follow up. After treatment, how many days? Again, after how many days you need to visit the doctor? Right? Let's say if it is going to be a fever, right? Let's say typhoid kind of a stuff. You need to visit your doctor again after 14 days. Again, you need to retest. That's the best option, best thing, COVID, right? So you tested positive based on RT-PCR. You need to wait at least for a week time again before you need to retest, correct? So when I have to do the follow-up. So in general, what is the procedure? First, the patient will sign your ICF, that is consent form. Screening will be done based on the exclusion and inclusion criteria. Randomization is done. So we are going to place the people into the control group as well as the intervention group. Then we start the treatment. Then finally we do the follow-up. End. Done. Clear? Element. So we have phase 1, phase 2, phase 3, phase 4 clinical trials. Right? Each of this phase will be called it as element. So phase is the generic term. Clinical research will call it as an element. If I have more than one element together, let's say clinical trials and preclinical trials, phase 1, phase 2. So I have 3 phases now. These 3 phases together will be called it as approach. Approach is the one which I can say that group of elements together, group of phases together. You know, scheduled visit, unscheduled visit, I don't need to explain. That's the reason I haven't written anything there. Scheduled visit is nothing but you take your appointment, you go there. Let's say you have a follow-up by after 2 weeks, you have the proper date, you go there. Unscheduled visit, you don't have a prior appointment is there. At the same time, your follow-up is actually due by next Monday, but you're not well. You're not well. Your symptoms are not good. So you want to meet your physician or you want to meet your clinician once. That is called your unscheduled visit. So these are all the terms that you would like to know or that you need to know as part of your clinical trials. This is the basic. I think we have done with all your definitions. The next class, that is by tomorrow, we are going to start discussing about your SDTM. So we are starting with SDTM tomorrow. Questions? I'm going to send this particular notes to Balu sir, and he's going to upload it into the drive today, so you can get this. All right. All I'll do is, because each Jamboard can have only 20, so that's it. That's the reason we have only 7 slides here. I'll ensure that I'll take these as well. You can say that from study design, from here, I'll take all these slides. I'll ensure I'll keep it in one. So I just call it all of them together as a proto definitions, and I can share it across with you. All right. Any questions, anything, please feel free to ask me.
Murli.
Yep, Shiva, please.
Yeah, I cannot attend the classes from tomorrow because my break time changes. So my break is at 11:30 CST, but it will be 10 o'clock in the India timing. So I cannot attend, but I can go with the recordings.
Anything related to the recording, kindly be in touch with Melissa.
Sure.
Anything, because generally the recording will take at least an hour, because it need to be converted, because it's a transcript now, right? So GoToMeeting itself will take 1 hour to convert the entire thing as a MP4 and make it as a transcript, right? So now the time is, let's say, around by 9:10. Let's say if I finish the class by 9:15, it will take an hour. By 10:15, the transcript will be ready. But by 10:15, is it possible for Balisa to share it or not? I'm not sure. So you can just place a request. Based on that, we can get it.
Oh, okay. Yeah, I don't get full recording. That's what you are saying, right?
Immediately you won't get it. That's what I'm saying. You can get the complete recording only after an hour.
After an hour. I don't need immediately. Anyway, I'm working in that time. So in a day, anytime if I can get recordings, that would be great.
Sure. I can. Yeah.
But I could not make it. So, and I have one more question not related to the classes. Do you also take Python classes?
I do take it.
Oh, yeah. Some of our friends they're asking about Python classes and business analyst. If you know anything,
give me a call. Maybe it's already. You can give me a call by 9:30 today. In another 15 minutes, you can give me a call and we can discuss.
Okay, Murli. Thank you. Thank you.
Yeah. Take care. So everyone, any other questions from anyone? I have time, so I can explain now. Guys, one slide question from my end. Are you able to follow? Are you able to understand? That is the main thing that I want to know this, because people who are already coming from clinical background, I can understand they can easily go with this. The people who are not having clinical background and they want to get into touch with this clinical stuff, are you able to understand this? I try to simplify as much as possible, and that's the reason I'm just going. If I go with the regular base SAS classes and all, I don't prepare any Jamboard or anything. But this is more of domain in nature. That's the reason I'm trying to prepare all this. So I just want to understand, will that be okay, or if you need some more, a different way of explaining or anything, please feel free to tell me. Don't hesitate, because it is one of the domain which is critical in nature and difficult in understanding. So feel free to ask me as many times as possible if you're not able to follow. All right, guys. Thank you. Thanks, everyone. See you tomorrow. Take care. Hey, hello, everyone. Good evening. Welcome. Hope all of you are able to see my screen and hope all of you can hear me. Can I have a quick confirmation, guys? Perfect. Thanks for the confirmation. Guys, please remember this. I have already informed this in the previous week. So we have classes on alternate days. That is, we have classes on Monday, Wednesday, and Friday, right? And every day it is going to be 90 minutes. So since we are in the beginning of the classes, I'm not taking continuous 90 minutes. Instead, I'm taking it for 60 minutes duration only. Okay, because these are going to be like most of the theory that we are discussing. It would be difficult to continue for 90 minutes of the time. That's the reason approximately between 60 and 75 minutes, I'm stopping the session, right? And if required, sometimes let's say that we need some continuity. In that particular time, I'll inform you one day prior. Let's say that today is Wednesday, and tomorrow also I would like to continue because I want to finish few of the stuff which is really important. In that particular case, today itself I'll inform that, hey, tomorrow we have a class and we can continue, right? So this is how we are going to proceed. Make sure it is going to be Monday, Wednesday, Friday, regular classes. Sometimes we may take additional classes on Tuesday and Thursday. If that is the case, I'll inform in prior. Am I clear with that? Cool. Now, so last week we have completed all the basic information that we required to kickstart with our SDTM, right? So we already informed you. First, initial things are going to be, we understand what is called clinical trials. That's the first thing. We understand all the phases of clinical trial, and then so in the clinical trial, the most important thing is going to be how to kickstart this and what are the various definitions. As part of this, initially we started with the protocol document, and we have elements of that particular protocol. We have discussed them as well. In addition to that, we have also discussed about few of the definitions. Correct? So these are the things we have discussed, and then on I think Monday, I have informed that we are going to kickstart SDTM in the next class, that is today's class. So first we continue with SDTM, and then we have ADaM, and finally we go with the tables, listings, and figures, which is nothing but the TLF. This is going to be the flow. All right. So let's not waste any time. So I don't repeat this one now because all of you are pretty clear with this particular one. Your clinical trials. I also believe that all of you are pretty comfortable with protocol and its elements. The definitions was the one which we discussed in the previous, this one. In the previous one, let's discuss that once, not discuss, just quickly we go with this particular one. So initial thing we discussed was blinding. So anyone tell me what is called blinding now? Blinding or masking is nothing but what information we are not disclosing, what information we are actually not disclosing to the people. So if you are disclosing everything to everyone, because we have three different entities involved, what are they? Subject, it is nothing but the patient. Then we have investigator. Finally, we have the team who is actually preparing all your data, analyzing the data. If we are disclosing all the information to all the three people, we simply call it as a unblinded. Correct? Yes, exactly. If I'm disclosing if I'm not disclosing information to the patient, but investigator and team is aware of that particular one, in that particular case, I'll call that as a single blinded. Correct? And what is called double blinded? Anyone? Anyone? What is? Someone responded no. Okay, it's okay. So and single blinded is the one where either the patient sorry, single blinded we already done. Double blinded. A double blinded is the one where both the patient, right, and also the investigator not aware of anything, right? Sometimes investigator might aware of this, but we can also have a case where patient and the team is not aware of what is the drug that they are taking or the drug disclosure. Correct? So both the cases will fall under the double blinded. For sure, patient does not aware of what the treatment that he's undergoing, but either of the investigator or the the team is not aware of the drug information or treatment information. That will called as a, just give me a second, this is something important. Hey, sorry guys, some important call. That's the reason I supposed to attend it, and it's done. Okay, I switched off mobile, so no issues here after. All right. So double blinded is also done, and the third and the last one is going to be the triple blinded. The triple blinded is the one where none of the people who are part of this particular approach are not aware of what treatment or what drug which is actually happening there. So they're strictly going to follow the protocol. As per the protocol, they're going to proceed. Correct? So this is what the first thing, that is blinding. So I have given all these details here. You can refer later. The second thing is going to be ICF. So ICF is nothing but anyone, what is called ICF? Can someone please mention about what is called ICF, the acronym? Wonderful, Ankita. Informed consent form. This is the form which is going to be a, I can say that a legal document which we actually get it from the volunteer or the subject, which is having it, which is a signed form, and the most important thing is going to be we are going to capture the date there. Correct? So this is a legal form which has all the inclusions, exclusions, all the details will be available there. Correct? So why we need this particular one, the informed consent form? To make sure the subject is well aware of what is happening there and to maintain the integrity. Now, what is the next thing? Screening. Screening is nothing but, so let's say we are volunteering and before we go with this before we pick this particular person, right, we need to ensure that the subject is actually fit for this particular clinical trials or not. So we have set of inclusion criteria as well as the exclusion criteria. So we need to check all these inclusion or exclusion criteria are meeting or not, whether we can consider him or her as a subject or not. So that can be called it as a screening. The next thing is going to be randomization. What do you mean by randomization? Randomization is the process where, so we have a lot of volunteers, or we are going to have a lot of subjects, so many subjects. So we are going to have the right mix of both the people. We need to say, let's say I have 100 volunteers. Out of them, we are going to pick 50 healthy people, or 60 healthy people, or 40 people who are actually the patients who are suffering with the other diseases, or the same diseases. So how to randomly pick the people? So we have a population, we have a sample. How to get the right sample is nothing but randomization. Randomization plays a very important role while selecting the subjects. And then, so we also talked about two things, that is nothing but control group, because we are going to do the grouping. So control group is the one, and another thing is going to be, what is the other one, guys? Anyone?
Treatment group.
What is the other name for it? Anyone? Exactly. Either you can call it as a intervention, not the investigation, but few books also refer that particular one, because they are using the investigation drug. We are actually providing, we are actually giving the investigation drug to that particular people, or the study drug to that particular people. So the people who are actually taking the drug that we are testing is nothing but investigation intervention group, or we can also call it as a study group. Whereas the control group is the one where the people is not taking the study drug at all. Actually, they are taking the placebo. Placebo is nothing but a fake pill. Correct? It doesn't have any active elements. It is just a fake pill. Correct? So we also talked about treatment, follow-up, and procedure. What is the procedure? The procedure is going to be, first we get the consent, then we do the screening, randomly we create the groups, then we provide the treatment, and we do the follow-up. Correct? And then in each clinical trial, there are phases. Each phase is nothing but an element. Group of elements together can be called it as the approach. Scheduled visits and unscheduled visits. What is scheduled visit? As per the plan, as per the prescription, we are taking it. Correct? Intervention, active, or treatment group. Perfect. All the three are, we can call it. Even investigation also. Investigation group also is the right name. Thank you. Thanks for sharing the details. So these are the various definitions that we would like to understand before we start with the SDTM. So hope all of you are clear with this. Now, guys, did you ever get a chance to read the protocol now? Because you know all the elements, you know all the things now. So if you go now and if you read any of your protocol, let's say this is one of the protocol. This is Alzheimer's disease protocol, correct? Not completely, at least most of the information you might aware of that now. So phase 3, so that the current element is going to be phase 3 element. Double blinded. What do you mean by double blinded now? The double blinded is nothing but patient is not aware of it, and investigator or the analyzing team is not aware of what is the treatment that is happening. Placebo controlled. What do you mean by placebo controlled? So there is also a fake pill which is supplying along with this particular NS0314D drug. Both of them we are giving them. Correct? Right. So what is the name of the investigational drug now? NS0314D. So since already we mentioned the phase 3, here you can see the phase 3. So now you can see objective, the primary objective, secondary objective, what is called a safety, endpoints, lot of design. You can see the trial design. Under the trial design, you know it is going to be a parallel group. It is going to be a parallel group. And it is randomized. Randomly we are picking the people. Multi-center within the same nation, but it is going to be at the, it is multi-center as well as the multinational, meaning across the globe, the trial is happening as per the protocol. Within each country, at multiple centers, it is happening. Clear? So what exactly it is happening now here? Now you can see almost what is going to be the trial. All of you are able to understand now. So before, we may or may not aware of all these terms, but now when you look at this particular trial design, when you look at this particular document, we can understand what exactly is happening. Am I correct? Can I have a quick confirmation? Wonderful. So all of you are pretty clear with this particular one. So I'll share this particular one, guys. Just have a look into this particular one so that you can understand what is going to happen there. Now you can see the screening, double-blinded treatment, when the visits are there, randomization when it is happening, when is the end of the treatment, all these details you can see. Correct? And I also told you the inclusion as well as the exclusion criteria. You can read them. Right, it is not same. Why we haven't discussed inclusion and exclusion criteria in detail means because it is different from each drug to other. At the same time, this criteria is different from one treatment to the other treatment. Correct. So that is the reason we are not discussing about inclusion as well as the exclusion criteria in detail, because it is completely random and it is vary from. Okay. All right. Now, this is the, I think I have already shared this particular one with you. If not, I'll share it today with Melissa so you can read this particular one. Now, come back to the next one. So protocol definitions, we are clear. So today we are going to start with the SDTM introduction. So before we go with the SDTM introduction, may I know like who actually, can I say, who might have actually created this particular one, SDTM? Anyone? Okay, why SDTM has come to the picture? Why do you need it? Initial classes for standardization. Wonderful. Okay. Okay. Let me just reiterate that particular fact and we can proceed there. So what we understand, every drug that we are actually producing, that can be a treatment drug, that can be a vaccination, or that can be a medical device or equipment, need to be tested for two things. What are them? Safety and efficacy. Correct? So to do that particular, what this governing or the authorities are going to do, the authorities are the one who need to approve them. They need to give a go ahead, then only it will come to the market. So let's say one of the regulatory authority is FDA. Correct? So now, which is as part of your United States of America. Now, so we know that each company can produce their report to get the approval to the FDA. If the reports are not standardized, if the reports are not following the same format or same structure, it is difficult for FDA to approve them, to at least to analyze them. Correct? To review that, if all of them are following the same format, it is easy, quick to review, and we can also do the automation there. I mean to say that we can also implement the machine learning models there. Right? So FDA is looking for the standardization. So what FDA did is, since they want to look for that, since they are looking for the standardization, so they have come up with an organization. They have created one organization, right? And that organization, any idea what is that organization is? Anyone? That organization is nothing but the CDISC, right? So we all know how our CDISC is nothing but global. It is universal. It is everyone can use this particular one. I can simply say that CDISC is one of the organization. Here you can see. Correct? So we have all the details. You have standards. If you want to go for the, even CDISC is going to provide all the education as well, but it is a bit expensive, and I can say that At a very high level, it is going to be. It is not going to deal in detail, right? So this is what your CDISC is going to be. This organization, you can also call it as this organization main purpose is to set and maintain the standards. Correct? I need to set and maintain the standards. At the same time, the standards need to be easy to understand, and also we need to provide the guidance for these particular standards. That is the whole objective of bringing your CDISC. Correct? So CDISC, where it is, just a second. Here it is. Here is what your CDISC is. Now the CDISC has come up with 3 different approaches. There are a lot of approaches, but we are interested in 3. Out of that, the very first one is going to be SDTM. Right? There are many approaches, but what we are very much interested in is going to be a SDTM. Correct? So I hope all of you know the acronyms for SDTM as well. Anyone? Study design, or I can say that standard design, tabulation. Anyone want to finish this? Exactly. Study design tabulation model, or you can also call it as a standard design tabulation model. And we also have ADaM that we can discuss later, and we also have your ALF. So today we are going to start our discussion with SDTM. So today we understand what is going to be the end-to-end workflow. So remember guys, all of you will not get a chance to work on all these 3 steps or all these 3 different modules, right? Some of you, once you join an organization, you may stick only to the SDTM. Some of you might be working only on ADaM. Some of you might be working on your TLF, right? Some of you might get chance on working on more than one model as well, or more than one design as well, right? Anyways, we are going to understand the complete flow, and we are going to work on the complete flow as part of the project. But today we will be specifically starting with the SDTM. All right. So here we also need to understand few things. I think whatever I have done so far, I have explained this, because all the clinical trials are usually straightforward. All we are going to check is safety and efficacy. All these things we are going to record. All these things we are going to collect. Indirectly, I can say that there is going to be data driven. So the entire thing, the entire approvals, everything is completely based on data. The data need to be recorded, the data need to be collected, it need to be compiled and analyzed. So how properly you're handling this particular data will ensure how nicely or how perfectly you are actually producing your reports. So to maintain the data properly, FDA has come up with the CDISC, correct? So as part of the CDISC, you can see the CDISC standards here. So there are 3 people involved. Who are the people involved there? Researchers, pharma companies, technology vendors like us. Okay, how these CDISC standards are helpful to us? I can say that, let's say if you're a researcher, simply I can say that how the data is. We are going to have a clear, that is the cleansed data and simple data management at all stages of your clinical research, correct? So what is going to happen? The results will be quick, the outcomes can be easy, and it will be, I can say that, simple to derive, simple to understand, simple to produce. Because researcher is the one who is going to give you the breakthrough in your clinical trials. So if the data is clear and simple, if I have effective data management, my outcomes are going to be quick. So from researcher's point of view, it is more about my data. From pharma companies never bother about how the research is happening, all those. Mean to say that in detail. All they need is, I have a report, how quick my report is going to be get approved. So that if the report is approved, I can send this particular drug to the market and I'll get benefit out of it. Correct? So all they need is how simple and fast I can submit my report, and how quick my CDISC is going to approve it, and how quick I'm going to deliver this particular thing to the market. So technology vendors like us, right? So I already have the data sets. I already have all the data sets. how quickly I can create the solutions, how quickly I can produce the analysis on top of it, right? At the end of the day, all we need is we have millions and billions of the data. All these millions and billions of the data that we have extracted based on the subject's information that need to be properly processed, that need to be collected, that need to be analyzed. That's the whole point of bringing the CDISC into the picture. Correct? Now, in the CDISC, before I go with CRF and all, let's draw a simple workflow. What is going to be happening there? Right. So the first and foremost thing. So first, we are going to start with a very basic thing. We first start with a clinical study. Let's say that I can call it as a study itself. Correct? I have my study. I want to start this particular study. Okay. So we have started this. What's next? Once I have started this particular study, that is nothing but study is the one where I have all the phase 1 preclinical trials, phase 1, phase 2, phase 3, phase 4, all the things are here, all of them. So to do this particular one, or once I have this particular one, to proceed with the next step in this one will be, I need review documents. Most important thing, guys, try to understand, ask me as many questions as possible. Okay, all I'm looking is my second step after my study, or after I kick off my study, is going to be, I need to have the documents. If someone asks me, Murli, what are the documents that you are looking at? I can say that the documents that I'm looking at here is nothing but, we generally call it as review documents. So what are those review documents are? The review documents are, number one, I need protocol. We discussed this last week, right? Protocol is going to be the first document which is going to kick off your clinical trials because it has everything, right? Okay, I have protocol. What's next? I need to have CRF. CRF stands for case report form. Let's discuss about what is this case report form is and how it looks like. So this is your case report form template. This is a blank template, guys, right? You can fill it up. So a case report form is the most important thing because it has all the information of an individual study, right? Based on the protocol, what and all we are doing, all those information will be available here. Why it is important? First thing, to ensure the data collection in accordance with the study protocol. So what do you mean by study protocol? Yes, protocol say that, hey, you need to collect this particular data like this. So I'm going with the parallel study design, and with the parallel study design, this is the dosage that I would like to provide. This is going to be the period that I want to provide. So these many random samples I am taking, and also this is going to be, let's say, the double-blinded one. According to that, I need to get the data. So we need to collect the data as per this particular one. Again, what and all the vitals that I want to collect. Those information also I need to do. Second, whatever I'm filling there, right? So I need to ensure that the data collection should be in compliance with the regulatory authorities. That can be FDA, EMA, or our drug control, whatever it is. It needs to be as per the regulatory authorities, right? And most important thing to be, I need to make sure that it should be effective, the data collection should be effective, and I need to ensure that the reporting is proper, and main thing is going to be, I need to promote the safety of data sharing between the various teams and departments. All these things need to be included in this particular CRF form. In general, what exactly this CRF form is actually including? Study title and the number, who is going to be the investigator, what is going to be the patient or subject ID, inclusion or exclusion criteria, demographic data, detailed description of dosage, how much dosage and what is going to be the and how frequently I need to give that particular one. And also you can see the remaining things, the investigator signature, date and everything. You can simply see this one, it is easy to understand. I can have the study title, you can see this, and then we are going to have the where the site is nothing but the hospital or clinic or doctor's home, whatever it is, who is going to be the principal investigator, the subject initials, subject randomization number, that's nothing here, ID, investigator signature and date signature. You can see here inclusion criteria, what and all I'm including. See this, if the subject is healthy male aged between 18 to 60 years, yes or no, right? So exclusion criteria, and you can see that the ICF, the consent, the demographic data is nothing but age, sex, and your height, weight, BMI, all those. You can see here all the demographic information, right? So screening, I told you the screening is very important before we get this particular person is a fit for my clinical trials or not. See the previous medical history. This is going to be around 15 pages of the document, and this 15 pages of the document will have all the information that a subject is actually screening and undergoing, right? So this is, now you can see that, now you can understand why it is important. This is important because it has each and every information about the subject, individual subject. Remember, this is not at the whole clinical trial. It is not about the drug, it is about the subject. This is about the patient. You understand, guys? How do I differentiate it? Clinical trials is a whole. So to start this particular clinical trial, I need a subject. The subject is nothing but the patient. I'm collecting all the patient information. Yeah. Is that clear? Can I have a quick confirmation? So once I have this particular information, now after CRF, I need something else that is nothing but a little advanced of this. We can call it as ACRF, Annotated Case Reported Form. That is, we just have annotations there. So what is that? I'm going to show you anyways. In addition to that, we also need SOP, Standard Operating Procedures. Right? In addition to that, we also need SAP. This is nothing but statistical analysis procedures. That is nothing but statistically what and all we want to conclude all those information. But we mostly require these four. What are them? Once before we want to start with the next process, I need to have the details about what is the protocol. That is the first thing. It has information about the complete clinical trials that we want to execute. CRF, a case report form, which is very specific to the individual subject. Annotated CRF, I'm going to explain what exactly it is. And then standard operating procedures, it varies from subject to subject and clinical trial to clinical trial. Statistical analysis procedures, these are also going to be varies from subject to the subject. But on a whole, I need all these documents. Let's discuss about how this ACRF looks like. ACRF, which plays a most important role because it is a blank CRF. First thing is it is a blank, right? And it also have annotations about the document, the location of the data with respect to the data sets. I forgot to add one more. I talked about all this, but I haven't talked about the most important thing, which is nothing but raw data sets. All your clinical data sets need to be provided to you, right? It should be looks like this. Let's say someone has given you the data like this. These are going to be all your SAS data sets. Can you see this? AE, CM, TA, DM. I have renamed each of the SAS data sets based on the domain. Guys, these are all the domains. Demographic data, you have AE, CM, CO, DA, PR, PQS, all these are going to be the domains. We'll discuss all the domains, don't worry. Right? Now, each of these raw data sets are going to be the input to me. Along with that, I also have my CR. I also have my CRF case report form. I also have metadata. That is nothing but three things I'm taking here. What are they? Number one, or simply we can take two. I have CRF, which has information about the individual subject. Number two, I have my what is the other one? Raw data set. Let's say a simple way. Here I have subject demographic information. Here, let's say age, sex is there. I want to know where this particular field is, right? Because I have a lot of data sets here, right? I have data set called AE, CM, DD, DM. Where exactly this particular field I can see, or where exactly I can identify, I can find this particular field. Even if I identify that particular field, with what name it is going to be, because I have here body mass index. SAS variables does not have any spaces. We are going to maintain our SAS variable names in compliance with the SAS variable regulations. Correct? Your variable name should not have more than 32 characters long. Your variable should not have any other special characters other than the underscore. Your variable name should not start with a number. It can be either alphabet or underscore. Correct? We have a lot of rules for defining your variable name. Correct? So now the biggest task is going to be, if this is going to be the label, I can call it as a label or a parameter, where this particular parameter is exists, in which data set it is exists. If it is exists in this particular database or in this particular table, with what name it exists, I need that information. That information will be part of your ACRF. This is called your annotated CRF. Can you see this? Right? Now, can you see this particular one here? Let's say, so here I have few things. What is the first thing? The first thing is going to be prior or post-study therapy. One more is their standard implementation guide. Sure. That is also required. I'll go one after the other, because that is the guide which is provided by the CDISC. Correct? So I'll also give that information. Currently 3.3 is going on, but most of us are still following SDTM implementation guide 3.2 version. I'll also give that. So now you can see that if I want to know the information about prior or post-study therapy, if I need this particular information, this information is part of AC table, that is AC data set. Here you can see the data set, right? One of the data set is AC. I don't have all here, but don't worry. In the AC data set, there is a name, there is a variable name called ACPRIPOS. This is going to be the variable name. This is going to be the table name, and that has information about prior or post-study therapy. So if you go with CRF have only the name. But when it comes to the ACRF, it also have the details about which location, that is nothing but where exactly you have this particular one, right? But one problem with ACRF is ACRF has every part of every piece of information that may be submitted, may not be submitted. In this particular case, it is not required. So we have not submitted this. Right? Now you can see the therapy type. What therapy type it is? The therapy type can be, again, it is part of AC. You can call it as ACTYP. So this is going to be CM's SCAT. That is a value of it. Correct? Can you see this? All the information, whatever the information is going to be. So here I have adverse events, AE. So we have the AE data set here. You can see the very first data set itself is the adverse event data set. So this adverse event data set has the information about this. Can you see? What is the adverse event? What is the name of it? Now you can see adverse event is part of the AE dataset. The name is going to be AE term. Is it ongoing or not? What is going to be the start date? What is going to be the end date? Right? What is the severity? You can see the information. Right? Let's see one more table. Let's see demographic table. Where it is. It is also AE. What's SDV again? This is the label. I'll give this information later. This is nothing but the label, right? So you are going to have this throughout, not here, throughout you are going to have it, right? So this is nothing but this particular study details name. So that's the reason it is going to have throughout, completely you are going to have the same name, right? So this is varying from one to other, so we don't need to worry much about it. All we need to worry about it, we need three things. The first one, what is the parameter you are looking at? Number two, what is the column name, where it exists? These two are the most important thing. There is another thing we can call it as in your protocol, in which page it is, or in protocol or in your CRF where it is. When it comes to the in protocol where it is, in CRF where it is. That is the orientation. I can show you that. I can show you that. So first of all, can you see what is called CRF, ACRF, and how it is different from CRF? Can I have a quick confirmation? So I have two things now: CRF, case report form, annotated case report form. How these two are different? All of you are able to understand? This is very important, and also this whole ACRF will be reviewed by the team leader. So in every organization, we are going to have a team lead. The main use or the main thing that is going to happen here is, maybe I need to tweak this a little. I'll remove this. Based on this particular information, we are going to create a ACRF. This ACRF, whatever we are talking, okay, there is a question to me. Are we covering this? How to create this ACRF? I can say that it is purely into the clinical data management team. They are the one who do this, because whatever we are doing here, I'll tell you where the SAS programmer will role will come. So generally this ACRF and all we will not create. I mean to say that we, in the sense SAS programmer, will not create this. That's the reason I told you here this ACRF is there, and this particular ACRF will also be validated by the team lead. But I'll tell you the process. Maybe that can be helpful. Sanji, you are the one who asked, right? How it will be examined. I'll give you the process. Right? Generally we don't do, but still I can help you with the process. So let me help you with the process. All right. So the process is going to be, I have included that here. You can see the approach. Always make sure that this particular annotated CRF, it is always to be in an Acrobat PDF format. Only Adobe Acrobat format only will be accepted, because there is standard as well. And always it need to be included. The name should be always like blank crf.pdf. This is the standard. There are 3 steps in this particular one. There are 3 steps. Number 1, I need to determine which annotations are to be available in which pages and store them into an Excel file. Meaning, I'm going to refer this. This is my CRF, correct? I need to understand this particular CRF first. From this particular CRF, I'm going to create 3 different columns, and I'm going to put it in an Excel sheet. Let me show you that. Here it is. Maybe I can zoom it a little. Yeah. Can you see this? The first one is going to be the CRF page. That is in CRF, where in which page this information is available. Next, what information you actually want to annotate, right? What information you actually want to annotate. Number 3, the orientation. Orientation is nothing but ranking. It has 4 values, 1, 2, 3, 4. That depending upon the what type of study that you are doing, based on that we are going to create this, right? So what is the first thing that we are going to do to go with this particular step? I need the CRF. This is the first thing. Number 2, I need to identify these variables. Variables are nothing but the columns, right? All these variable details are part of your SDTM. should be somewhere. Just give me a second. There is an implementation guide. Let me pick this up. Just a second, guys. I have a very bad habit of keeping the things. This is one of these SDTM. Can I say that this is the version 3.2? In this, we have the details about the variables. I'll tell you where it is. Can I say this? Identifier, variables, these variable information I need to put it properly. So we have data sets and domains. These are the ones we are going to discuss. But can you see that the most important thing is going to be we need to know how to create the variables, what variables are there. Correct? So the first and foremost thing is going to be I need the information about your CRF. I need information about your variables, all the SDTM variables. Once these two are available, I can create them like this. This is the Excel that we need to create manually, right? So once you have this particular one, this Excel sheet, we are going to supply to the SAS tool. SDTM specification development means, where do you find it? Raja, you meant to say here, or I'm not sure what exactly it is. Okay, the specification development means where exactly the variable is going to be, how to bring it, all those information. Raja, if you want, you can unmute, you can talk about it. Else we can do this later also, not an issue. All right, let's discuss this and we can come back on it. So based on this particular one, because I want to create ACRF has three topics or three steps in that. The first step is going to be we are going to create an Excel. The Excel has three fields. In which page of my CRF I have the details. In which page? That is the first thing. Number two, what demographic information I'm looking at. That I can achieve from my SDTM. Then the third thing is going to be the ranking, the orientation. This orientation is nothing but this, you can also call it as your priority, or you can also call it as a ranking. So there are 4 rankings has been given for each of this particular value based on each PDF that we are going with. The values are going to be 1, 2, 3, 4. There are 4 rankings, but how exactly the ranking is going to do and for what variables and for what domains, what ranking we are going to give, that we are not very keen into. We don't involve much there because just we are not the one who are going to produce it. Is that understandable? Why we are not going in deep means generally this we will not do. Being a SAS programmer, we will not do that. This can be done by your biostatistician along with your TL. We don't do that. Got it? That's the reason we are not interested to go into this one in detail, because the role of a SAS developer has not come yet. These are all not the roles of your SAS developer. We are not into that yet. Okay. So once we have this Excel we are going to supply to the SAS program. The SAS program is going to convert this Excel into XFDF file. The XFDF file is the one which is going to have all these boxes. Can you see this? These boxes, right? Annotation boxes are there, right? These boxes are for part of your file, and that file is called XFDF file. So then this XFDF file, there is going to be a process, nothing but what is going to be the color format, what is going to be the priority of this. The orientation will be decided by here. And also we are going to talk about which domain and all these stuff will be part of your XFDF file. there is going to be a separate program for this particular one, the SAS program. Few samples are with me. I'm going to provide those samples. But believe me, it is different from each CRF to other, and it may not be useful. But still, if you want to have a look at all these three, how I'm giving this orientation details, right? And where exactly I need to place these particular annotation boxes, right? With what color I need to fill that. All those information we are actually programming by using the SAS itself. The SAS details, the SAS program details, I'm going to provide it to you. Just have a look at this. Is that clear? Everyone? This is not our piece, but still, if you are interested, I can provide the programming for you. Honal, Sanchi and T, both of you, is that okay? Or do you have any follow-up questions on that? Anyone, any questions? Right. Remember, guys, the other part that I would like to talk here is there is a difference between the clinical SAS programmer and clinical research person. You can also call it as a clinical research associate. So this role, the clinical research associate, is the one, this particular guy, along with CDM team, clinical data management team, these two are responsible for producing this data, this ACF and all, ACRF and all. Being a SAS programmer, you're still not going to utilize this particular one. You still not go with this particular one. Or once you have ACRF ready, based on ACRF, your protocol, SDTM, and raw data sets, then only you're going to proceed with your programming. I mean to say, maybe this is too early, but let's talk about it. There are 3 steps, guys. First thing is ACRF. Number 2, there is something called SDTM mapping specification. Number 3, programming. This is called your SAS programming, purely SAS programming. So now we are here, that is nothing but we are at this particular box, ACRF. So let's say that your ACRF is good and it is properly validated. Okay, there is a question. ACRF annotation means how to write a specification file, how to write the programming. No. ACRF will have, the ACRF is going to be one of the input file to develop this SDTM. Only when you have SDTM mapping specification, then only you can start your SAS programming. Okay, let's start from here. Let's say I want to write a SAS program by utilizing your raw data sets, right? So let's say you're a programmer. So what are all the inputs you have? You have inputs about your SAS tool. Tool is there. We all SAS programmers, we know how to use this. Number 2, I have raw data sets. That is, whoever the one who is providing this particular project to you, the project manager or the team who has given all these SAS data sets to you. So these two are well aware. We know how the data set looks like, what contents we have there, and we have the SAS tool. But the thing is, how systematically I can use this particular data? I mean to say, what variables I need to use, correct? And how to produce my TLF, the tables or listings or figures, which are part of your SDTM implementation, right? So currently 3.2 version is there. This is also with you. SDTM also with you. So SDTM has information about your variables. The next thing is going to be, let's say SDTM says that, hey, there is a variable called sex. Now, you need to understand where this particular variable will be. Because I have a lot of these data sets, right? In which variable it is, what format it is, right? What label we have, what is the length of this particular one, what is the source information. Only when you have this, it is easy for you to pick the right table and right information. Am I correct? Raja and everyone. Am I correct? You're a SAS programmer. I have given you the data sets. I have given you 100 data sets. Correct? But out of those 100 data set, from which data set you need to pick this particular variable? Correct? That information is part of this one. So this has the information about your SDTM, CRF plus your SDTM implementation guide plus CRF. These two information is part of this. Correct? Protocol, SDTM, CRF information. If I combine these three together, I'm going to get ACRF. But ACRF has information about table and a variable. Correct? But it doesn't have information about the label. It doesn't have information about your format, the length, all those things. Correct? To get those details, I need those details as well. Those details are part of your SDTM specifications. Is that? If I want all these information is part of SDTM specification Excel. And that Excel looks like this. Now this is metadata, just a second. Can you see this? Let's say I have demographic information. So the domain is DM. These are the variables. Study ID is the variable. The study ID variable information, I can get it from SDTM implementation guide. The label is called study identifier. You can see that it is a character. The length is 8. Code, is it required or not? It is required. And you can also see here CRF. It is actually the origin is a CRF. Right? Domain is not part of your CRF. You are assigning this domain equal to DM, demographic domain. And you can also see that U subject ID, that is unique subject identifier. This is also unique. This is also going to be a derived variable. You are combining your study ID with subject number, right? Study identifier and your subject number, both of them you are actually combining, then only you are getting this. So are you deriving it? Are you getting this particular directly from the CRF? What exactly it is, right? So this mapping Excel, you can see that. What is the origin? Is it assigned one protocol or what exactly it is going to be? This information is actually part of your. This information is actually part of your. What is that? ACRF. Correct? You can also see there is another domain. I have variable name, study ID, what type it is, what length it is, what identifier, is it origin or not, how it looks like. Can you see that all this information? Right? This particular thing will be call it as your SDTM specification Excel. This SDTM specification Excel or this SDTM mapping specification Excel will be prepared based on your ACRF. So I am a SAS programmer. I have a tool, I have raw data set, I have implementation guide. But I need the mapping based on ACRF. So I need to provide it with these 5 things. SAS tool. Okay. How would we be preparing this SDTM specification or Excel? I mean, what will be the base to create this Excel? Apart from ACRF, will it be okay? Generally, you will not create this SDTM specification Excel from the base. You generally don't create, right? So who will we do this particular one? So to implement this particular SDTM specification Excel, you require ACRF, right? This will be provided by your TL, followed by you have your protocol based on the protocol document. You also going to have your SDTM implementation guide. And raw data sets. Based on this, we generally created. Let's say you are the one who started this particular project. Let's say that there is no prior history at all. There is no prior history at all, right? In that particular case, based on the CRF, because the CRF, what and all submitted, what and all not submitted based on this. Let's say that, so I have this one, action taking with this particular one, let's say AECAN1. Now you understand that AECAN1 is part of AE. So AECAN1, which is part of AE dataset, you are going to open the AE dataset and you are going to prepare what is the length of it, what is going to be the values of it. You are going to prepare this Excel on yourself based on you. You are the one who are going to produce it. In general, if it is prepared once, anyone can use it. So it is an incremental thing. It is not going to be created on one night or one day. No. Let's say there is a team. Let's say that you're working in a team. The team has a TL who is part of this particular project from past 2 years. So they already have created this particular one. You're simply gonna use this particular. Is that clear? You are not going to create this particular one from the scratch. No. If you want to do this, it is completely a manual process. This manual process can be done based on these 4 things. Am I clear? Now, this one. This can be done by creating based on your blank CRF. First, you are going to use a blank CRF. You have your protocol. You have your SDTM implementation guide, you have raw data sets. In addition to that, you also have CRF guidelines. So I told you right just now how to create this. There are 3 steps you are following. Based on the 3 steps, first you are going to create the ACRF. The ACRF will be validated by your TL. Once it is available, then you go to your SDTM mapping specification. To prepare SDTM mapping specification, you need these 4. Once you have it, then you will go ahead and you create your programming plan. Guys, is that clear so far? Can I have a quick confirmation? Right? Still, SAS programmer role is not here. So up to this, there is no SAS programmer. The SAS programmer role is about to start from SDTM mapping specification. If you are the one, there is no history available. Just a second. Hey, sorry guys, I unexpectedly hit on the mute and I was talking. Sorry about it. So first of all, ACRF, all of you are clear. We are going to have the blank CRF. The blank CRF based on the protocol, SDTM guidelines, raw data sets, and CRF guidelines, we are going to create it. Once it is done, since either you are going to get the leverage on the previous guidelines that I have already created, if not, you are going to create from the base. That is based on the ACRF protocol, SDTM raw datasets, you are going to create the guidelines like this. That is mapping, SDTM, mapping specifications. Once you have this particular specification, the next thing is going to be, you are going to start a programming plan. It is not the programming yet, it is programming plan. So what do you mean by programming plan? A programming plan is that, let's say you are involved, let's say your protocol or your clinical research involved 5 different ones: TA, TE, TI, TS, TV. Let's say that. all the 5 domains cannot be given to you, and you cannot work on all the 5 domains at a time. So what needs to be done? Let's say we are 3 programmers. I can say that, hey, let's give TS and TV to Sanchi. TA I can work on myself, and let's say that give TA and TE information to, let's say, Manida. So we are going to prepare a plan such that who and all the programmers are going to be involved in this, and what domains we are going to give to which programmer. That particular plan we are going to come up with. That is called your programming plan. Once the programming plan is there, then we are going to actually start with program. That is, here is what we are going to write the SAS program. Remember, whatever the program we have created, the output of this particular program is nothing but, that is SDTM output is nothing but, can I say SAS datasets? All of you? Because we are not going to ADaM yet, we are not going to your TLF yet. So at this particular level, the SDTM output is going to be SAS datasets. Am I correct? Every one of you accept that? The results are nothing but SDTM, the SAS datasets. Yes or no? Right. So, but remember, they cannot utilize, that is, the report cannot be in your SAS datasets. We need to convert this particular SAS datasets into one of the specific format. You can call it as XTT files. There are going to be bunch of softwares available to you, and by using those bunch of software, I'll tell you, I also help you all this information like how these XTT files are going to be. So we are going to convert your SAS datasets into XTT files. Then we are going to use different companies use different softwares, but one of the most commonly used software is going to be Pinnacle 21. By using the Pinnacle 21, we are going to use all these XPT files. We are going to say that these are going to be the SDTM implementation guide 3.2 as per the 3.2 standards. We are going to create XML file. This is our final output, right? So this is called an XML file. It is always going to be define.xml, this particular file, and we also going to give validation notes. That is, we are going to validate this and we are going to produce this XML file. These 2 files we are going to submit to the, that is called submit study. These are the ones we are going to submit. So the first initial step is going to be get your protocol, get your CRH, and also have your SDTM implementation guide. Create your annotated CRF, which have information about domain and variable name. Then prepare your SDTM mapping specification, which has information about what variable, what table, what is the length, nothing but the metadata information. Once you have it, have your programming plan. What are all the domains involved, which programmer is working on which domain, and then start your programming. Once you complete your programming, you're going to have your output files. That is nothing but SAS datasets. Convert those SAS datasets into XPT files. Input these XPT files to the Pinnacle 21 software, which is going to create XML file. We generally call it as define.xml file, and it also you need to create a validation notes. Both of them submit as your study results. Is that clear? What is called your SDTM is and what is going to be your workflow? Whatever you can see it on the screen, I'm going to put everything into Excel with a proper flow chart, and again I can submit to you or I can share it with you. Is that clear? First of all, on a very high level, on a day 1 of your SDTM, are you able to understand the workflow on a very high level? Yeah, Sanchi, we are not going that detail here. True, but we are not going that detail here. Any questions from anyone? Can I have a quick confirmation on this? Any questions from anyone? On a very high level, what we are doing, right? So what we are going to start then, let's say if this is going to be the workflow, what we are going to start. We are not going to start about how to create the CRF, what is the CRF and all. Instead, because our main thing is going to be identifying this, correct? I want to create SDTM mapping specification. For that, either you're going to get leverage on the previous programmer's data or something. If not, if you want to start from the beginning, you need to have a very good understanding about these domains, correct? What is each domain, what variables are part of it, you need to understand that, correct? So we are going to have different domains in SDTM. We have special purpose domain, we have interventions, we have events, and we have findings, right? Sometimes we can also say that trial design models also there. That is called TA, TST, TBTI. All these are these 1, 2, 3, 4, 5. The first 5 you can see, these are called trial design models. Okay, let's say if I say AE is part of your events. This one, this is events, right? And you can see there is something called MH. The MH is also part of the events. So you can see something called CM, right? CM is nothing but as part of your interventions, right? You can see something called other SDTM. There are so many other, I can give you the details as well. But on a nutshell, we are going to have the different domains. We start with the special purpose domains, which are very important, which are nothing but the DM, CO, SE, and SV. So those are the ones we are going to start with this. Yeah, core variable Raja, I can explain because it's already 9:15. We can discuss in detail because when you go with each domain, we are going to discuss about all this, not one, all this, right? In detail, what is each variable is, what label, and all the information. So no worries about it. I can explain that. Is that clear, guys? So tomorrow we are going to start with the SDTM guidelines first. There is going to be a guidelines. And then we are going to start discussing about each of this. That is nothing but what is code. Under the code, we have, maybe I can show you one. So you can see REQ. REQ is nothing but required. I mean to say that these variables must be included in the domain. These variables cannot be null. That is, let's say study ID. I should not find any data set or any domain where my study ID is null. No, I cannot have it. So REQ is nothing but the required. And you can see something called EXP. EXP stands for expected, right? Meaning to say these values supposed to be part of your data set, right? But they can be missing values as well. They can be missing values as well. That is, study ID cannot be a missing, right? There is no study ID which can be missing. There should be a value for it. But when it comes to the EXP, that is expected variable, if the code is going to be expected, this supposed to be part of my data set, but this value can be missing. Now there is something called PERM. PERM stands for permissible, right? It purely depends on the sponsor either to include or to drop, right? You can have it or you may not have it also, no issues at all, right? Even if you drop it, it's okay. The sponsor or the CRO are the one who is going to take this particular call, right? Raja, is that clear? On a very high level, but when you go with each domain, we can discuss that in detail. But you can see there are 3 values here. What are the 3 values? I'm trying to explain that. Correct? All right. So first we are going to discuss all the SDTM guidelines. So once the SDTM guidelines are there, then we can start with the special purpose domains. Once we start with the special purpose domain, our programming also going to start. Clear? So to start the programming, we need to wait for 3 more classes. So I can say that this week we are going to dedicate for SDTM guidelines. That is nothing but all the guidelines we need to understand. The guidelines include how to write your domain name, how to write your variable name, how I can say that what are going to be the various data types that are allowed, what are the formats that are going to be allowed, what is nothing but your code, what is nothing but the rule, what are called identifier variables, timing variables, topic variables, and then we go with each of the two. Clear? Right. So when is going to be the next class? So since it is going to be theoretical in nature, so today is going to be Wednesday, let's have class on tomorrow and day after tomorrow as well. That is nothing but you have classes tomorrow and day after tomorrow as well. Right? So the next class is by tomorrow. Clear, everyone? So my target is going to be by day after tomorrow we can complete all the guideline, and then we can start kick off with our special purpose domains from Monday. Okay, so tomorrow and day after tomorrow we can complete all the guidelines. All of you, can I have a quick confirmation? Is that understandable? Harini, just get in touch with Balu sir, the person who you contacted, so he can help you with the recordings. Clear? Thanks everyone. See you tomorrow. Tomorrow there is a class. Please join. Thank you. All right. Good evening, everyone. Hope all of you can hear me and hope all of you can see my screen. So I'm still waiting for the participants, but it's already 8:07 p.m. IST, so better to start. So can I have a quick confirmation, everyone? Are you able to hear me? All right. So perfect. Thanks for the confirmation. So let's have a quick recap about what we have done yesterday, right? So we started with SDTM. We all know SDTM stands for, what is SDTM? Study data tabulation, correct? So this SDTM currently we are referring with 3.2. That is, so SDTM providing a simple implementation guide. The implementation guide that we are referring is currently 3.2. Even 3.3 is there, but most of us are still at 3.2. Mean to say all the companies are still sticking to 3.2 version. That's the reason we are discussing about 3.2. For an example, yesterday we started this, but we haven't go in detail. So let's complete that today. So what is the first thing? So when you want to start your study, the first thing is going to be in your study, the entry point is going to be you required review documents. So what do you mean by review documents? So first we have study. So I'm going to start the study. So when we start the study, the first thing that we are looking at is we want to stick to the, what is that? We want to proceed with the review documents. Can someone tell me what are the review documents that are required as part of your study? Can someone? Okay. The first one is going to be protocol, correct? So the protocol is nothing but the entry point. It has the information about what is going to be the trial that you are going to conduct, what is the methodology that you are going to follow, what are going to be the statistical measures, every piece of information. Number two, once we have protocol ready, the same, whoever is going to be the sponsor or whoever is going to be the lead is going to provide you the raw data sets. That is nothing but the data that has been collected. Correct? Number three. So from the CDISC, you can get your SDTM implementation, right? Which is 3.2. Okay. Number four. You are going to get your CRF. CRF will not come here. Annotated CRF will not come here. You are going to get only case report form. Correct? The case report form is going to have the details about. So what is going to be the study? All those kind of details. This is your CRF. Your study title, your investigator details, your demographic details, inclusion, exclusion criteria. What is going to be the, the habits? That is nothing but these parameters are nothing but your smoking habits, alcohol consumption. Are you under any medication or not? What is going to be a previous medical history, right? All these things you are going to have it under your CRF. Along with the CRF, you're also going to have SOP, which is nothing but the standard operating procedures. You're also going to have SAP, which is nothing but your statistical analysis parameters, correct? So there are some specifications are there, those specifications also you have to consider. So all these are going to be here review documents. So once you have review documents and all, the next step is going to be we need complete details about the subject. The subject is nothing but patient, right? All the details we need to have. All those details we are going to keep it under the ACRF. ACRF is nothing but annotated case report form, which is going to be validated by your DL. So how you are going to prepare your ACRF? To prepare the ACRF, what is required here? To prepare ACRF, you required first a blank CRF, correct? First, I required a blank CRF. Again, you are going to have your protocol. Am I correct? The same protocol is there. You are also going to have your case report form. You are also going to have your SDTM technical implementation guide. Along with that, you are also going to have your raw data sets. By utilizing all these, by utilizing the SAS software, you are going to produce it. Yesterday, I have also given the details about how to produce it. Correct? So if I want to talk about that on a lighter note, what you're going to do, there are 3 steps involved in preparing this. What is the first step? You are going to prepare an Excel. The Excel is the one which is going to have the details about your CRF, your columns that you want to populate in your ACRF, and also you're also going to have your derivation, sorry, orientation, correct? So orientation have 4 different values. Those 4 different values ranging from 1 to 4, that will be decided based on what kind of study that you are actually doing with. Am I correct? So these are the Excel. So once you have this particular Excel, by using your SAS software, you are going to input source this particular Excel file, and what you are going to get, you are going to get a very nice format. Here is what that particular format, you can call that as a XFDF file. You are going to create your XFDF file format. After that, what you are going to do, this XFDF file format is nothing but this is the one which is having those annotated boxes, because your case report form is going to be like this, correct? So this is going to be a variable, means your variable what exactly it is, which domain it belongs to, what is the variable. Correct? Those are called annotated boxes. Those annotated boxes will be prepared or will be produced by using particular SAS program, by utilizing your Excel that is manually prepared. Am I correct? So once I have these 2 information, and what is the next step? Finally, we are going to create the ACRF. By exporting that, we are going to get your ACRF. That ACRF is going to be validated by your DL. So once DL has validated this particular one, the next step is going to be, we are going to create what we are going to create here, guys. Once we have this particular one, then we are going to create SDTM mapping specification. Correct? So this is a having specification. This is also called as this. We also can prepare based on the DDT. DDT will be called it as your data derived table. But if you want to prepare this particular specification, this what I can call the specification document, if I want to create, I need some mapping Excel. Correct? I need some specifications. I told you yesterday, this specification either you can prepare from the scratch, or you can also utilize this particular one, which is already prepared by some other programmers as part of your project. Any of this will be done. 90% of the cases, we are going to utilize the one which is already available, and your specification documents are going to look like this. So for each domain, what is going to be the, where exactly I can find this particular data set and other details. Am I correct? So this also we have discussed yesterday, but not on a very lighter note, we discussed this. Okay. What is the next step? Once you have this particular one, now you know your domains, right? What domains you have, what and all, from where and all you are going to pick the data. All these details will be available for you. The next step is going to be, once I have these particular details, the very next step is we go with the programming plan. Am I correct? That is actual SAS. You are going to be a SAS developer is involved here. So here is what we are going to have. So instead of SAS, I can simply say that programming plan. What do you mean by programming plan? A programming plan is the one where your TL along with your, it can be a TL, it can be a Scrum Master, it can be anything. So here we are going to decide who and all working on what domains, because I have multiple domains involved in this particular project. So who is working on what domain? That particular details will be mentioned in this particular domain. Sorry, in this particular phase. So once we decided that, so there are 5 domains, there are going to be 5 programmers. Each programmer is going to work on this specific domain. Then immediately your programming is going to start. So for my convenience, I'm just keeping here programming plan along with the programming, both of them. That is programming implementation. I can say that implementation. Both of them we are going to do it here. So what do you mean by the programming implementation? Meaning after I complete my program, I can expect that my output is nothing but the SAS tables, and all these SAS tables are in compliance with the implementation guide. That is, whatever we mentioned in the implementation guide, whatever CDISC standards are there, according to the particular standards we are going to prepare. We have already prepared this SAS tables, correct? So this is, we can call it as SDTM output. This SDTM output is nothing but your results. These results are in the form of SAS data sets. Am I correct? So the SAS is actually entered into the into your thing here. This is where we actually enter into the show, correct? So we are going to have our raw data sets, and the raw data sets we are using here. So we are going to prepare the data sets which is in compliance with the SDTM implementation guide. Now, the next step is going to be, we need to convert all these SAS data sets into a different file format, that is, so the different file format that I'm calling here is nothing but I'm converting SAS data sets. Just a second. My keyboard, just give me a moment. Now it is okay. So my converting SAS data sets into, we can call a file system which is nothing but XPT files. We can convert all our SAS data sets to XPT file system. So these XPT files, we will source it to, there are multiple softwares available. One of the common software that we all use is going to be, we will give this particular one to Pinnacle. So precisely speaking, we can call it as a Pinnacle 21. So this software and all I'll show you, no worries, like how it looks like and what we are going to do with this, we can discuss that. This is just the base to understand the entire workflow. So now I'm going to have my Pinnacle. So I'm going to supply this to the Pinnacle. The objective of using the Pinnacle is going to be, my final report is going to be in an XML format. So the output of the Pinnacle is going to be, because it is going to utilize your SDTM implementation, right? And it also do the validation. By end of this particular one, I'm going to have two different reports, or I'm going to have two different files as the output, or two different files I'm going to generate. So the first file that I'm going to generate here is nothing but my define.xml. This is the XML file, or this is going to be the final file that we are going to give it to the, who's that? Our people. That is for submission, we are going to use this particular. We are also going to prepare something called validation notes. That is, we are specifying that we have also validated this particular document. We are giving that particular confirmation to the entire team. Now, once I have all this, because I have define.xml, my final report and validation, then I can say that, hey, I'm done with everything. I think this is going to be the end. Let's submit this particular report and get the approvals. So my final step is going to be, I'm going to submit this particular report. That is nothing but my end of study. That is submit study. So if you go in detail in clinical, like there are going to be few more steps you can see, but you don't need to be that, I can say that you don't need to go that deeper on a very high level, because the reason why we are discussing this particular flow and all is, this is really important for your interviews purpose. That is, if your interviewer asks you about, hey, what is going to be your day-to-day work, right? Or can you explain me your workflow? In that particular scenario, you can explain this entire flow. Can I have a quick confirmation? All of you are comfortable with whatever I have discussed so far? Can I get a quick confirmation? Any questions here? Please feel free to ask if you have any questions or if you have any doubts. Please ask me. Guys, is my voice breaking? Please confirm, because Raja feeling that my voice is breaking. If so, I can check it. So everyone is clear with this? Okay, Raja, can you check it your end, please? So let me save this. So just go with SDTM workflow. All right. So now today, so today we will continue with the next thing. The next thing is going to be, so in SDTM, what we are going to discuss. So we are going to discuss about various domains. What we are going to do? We are going to discuss about various domains, right? So what are those domains that we are going to discuss? Let me give a list of domains that we are going to discuss, right? So maybe I can also put this in a Jamboard. So this is also done. So now I'm going with SDTM domains. So what are the various domains that we are going to discuss as part of our number one? So the very first thing that we are going to discuss here is, we will talk about special purpose domain. First thing is going to be special purpose domain. You can ask me, Murli, first of all, what is this domain is all about? You're talking about the domain, but what exactly this domain is all about? How do you understand this particular one? Any idea, guys? What is called domain? Anyone? All right. So we all know that there are going to be SAS datasets, correct? What these datasets are going to have? The raw datasets. Can I say that the raw datasets is going to have the info? Let's say that I want to know about the patient information, the demographics I would like to know. All the demographics I would like to know. Right? Mean to say, someone is saying common domain, basic. Okay. Okay. So on a nutshell, can someone tell me, if someone asks you, because we are going to use two different terms continuously from here after. Number one is domain. Number two is going to be a data set. Anyone? Okay. In a simple terms, if I want to talk about it, in a very simple term, if I want to talk about this particular one, I can say that a data set is nothing but a structure, nothing but a table associated with a grouping of observations. Meaning that, let's say there is a patient. There is a patient. The patient information means to say that what is going to be his age, gender, right? Followed by which continent he belongs to. That is nothing but a race, correct? So this information, whatever I talked about, this is nothing but a demographic information, right? About a patient, right? All these things I'm going to keep it in one table. So this table has the information about the person, his demographic information. Similarly, at the same time, when I'm talking about this particular one, I'm also going to talk about subject elements. Mean to say that what are going to be the various phases in which this particular patient is involved. Mean to say phase 1, phase 2, phase 3, phase 4. In phase 1, what are the various vitals or what are the various parameters we have recorded? Phase 2, what we recorded? Phase 3, what we recorded? Then we also have the comments. What do you mean by comments? Comments are nothing but comments given by the investigator. So I have provided this particular drug as a result of this particular drug. This patient has responded like this. So comments. Then when we want to go with the information, when we want to go with the visits, scheduled visits. So when there are scheduled visits, there are unscheduled visits as well. So visits information I'm storing in one table. So now we can see demographics is part of one table, comments is part of another table, subject elements are part of the one more table, visits are part of the another table. So these individual tables which are having a specific information, we can call it as a data set. Am I correct? A data set is more about the information, the observations about the subject or about the about any entity. Correct? Then what is domain? The related data sets which we keep together, you can call it as a domain. Meaning, there is a patient, mean to say subject. The subject information, what treatment here, what are the elements that he is undergoing, that is what are the phases, what are the visits, and what are the comments that are produced, that are given by your investigator. If I keep all these, because these are talking about the details about a patient. What is the subject? That is, what is the patient ID, gender, his age, what drug he has taken, when he has visited the hospital, followed by what are the various comments passed by the doctor. All these together, because these are related information, these related information together, I'm going to keep it at one place. At that place, you can call it as a domain. So if someone asks you technically, hey, this is going to be the normal way, can you explain me in a technical way? Right? I can say that the grouping of the observations, you can call it as a data set, whereas grouping of the observations that are related is nothing but a domain. Right? Simply grouping of your observations is a data set. Grouping of observations from each data set which are related is nothing but a domain. Okay, let me explain you with the example so that it will be easily understandable. For an example, just now we talked about the first thing is going to be a special purpose domain. The special purpose domains has 4 different data sets in that. The first one is going to be DM. The DM stands for demographics. So what demographics is going to have? The name itself is going to tell you. The demographics is going to have information about the age, gender, the basic information, I can say that, about the individual subject. my keyboard. Yeah, second one. So we have the second one, which is nothing but CO. We can call that CO as comments. All the comments which are given by your investigator, we are going to keep it at one place. Then we are going to have something called SE. SE stands for subject elements. What do you mean by element, guys? Can someone please tell me? We keep on talking about it. Before I'm going to type it properly with the help of my keyboard. Can someone please tell me what do you call element? Come on. Anyone? Guys, I would need answers from all of you. Please consider this. And then I have something called SV. SV stands for, just give me a second guys, I'm going to change my keyboard. But it won't take time. I continue as we discuss. So no issues. Exactly, the clinical trials phase. The clinical trials phase itself, you can call it as a element, right? What do you call epoch? A continuation of that. What is called epoch, guys? Can someone please tell me? Group of elements will be called it as the epoch. Wonderful, wonderful. Ratnasri and Sweta, thanks for sharing these details. Okay, now let me talk about this. So we have subject elements, then we also have this subject, call it as visits. What are the various visits? Right, both scheduled as well as unscheduled information, both of this will be available here. So now we have 4 different domains here, and all these 4 different domains together we can call it as special purpose domains. Okay, now the second thing is we have something called interventions. You can call it as interventions. So now the thing is, what is called this interventions is all about. Thanks, Sanchi. Thanks for the detailed one. I really like it. All right. So now let's talk about interventions. Under the interventions, we are going to have something called EX. EX stands for exposure. So don't worry, guys. We are going to talk about each and everything, what it meant for, what are the various variables we have, everything we are going to talk, right? All the variables which are part of it and how to understand that. That is what we are going to discuss about each domain. So no worries about it. So then the second thing is going to be exposure as collected. EC stands for exposure as collected. Then the next thing is going to be, you're going to have something called CM. CM stands for concomitant medication. So we are going to have something called concomitant medication. So what is going to be the medication and the respective details? This also we are going to discuss. Then we are going to have something called SU. The SU stands for substance use. What are the various substances we have used and are they in the compliance or not? So these are nothing but my, just a second. I just want to keep everything together. So this is your interventions. So these 4 will part of your interventions. Okay. So the next thing is going to be, you have something called events. What is that? You have something called events. Under the events, again we are going to have 5 different tables here. The first one is going to be, you can call it as AE. You might have seen in the list that I have shared yesterday, correct? So this is going to talk about adverse events. The second thing is going to be, you have something called MH. MH stands for medical history. You have something called medical history. So what is the patient medical history, right? Then we have disposition details, DS. DS stands for disposition. And then the next thing is going to be DV. The DV is nothing but, you remember I talked about something called deviation in the protocol. Though we are giving the protocol, sometimes we are going to have a deviation in the protocol, and we need to know that the deviation in the protocol must be within the limits, correct? So that details will be part of protocol deviation. All right. So now the next thing we have here is CE. The CE stands for clinical events. What are the various clinical events and what is the information about it? So this is going to be a third domain. So then you have your fourth domain, and the fourth domain you can call it as findings. So findings is all about, so what you have here. So findings primarily dealing with the laboratory, where you find something that is a laboratory only one, right? So under this particular one, you have first LB. LB stands for laboratory details. So laboratory details, all the things. Number 2, that is the laboratory test results. Second thing is going to be VS. VS are nothing but vital signs. What are the various vitals and the signs respect to that particular one? Then we are specific about each of this particular diagnosis now. So now we are going to talk about EG. EG stands for ECG test. That is ECG results. Then we are going to have PE. PE stands for physical examination. Then you have, after the physical examination, there's no order, guys. You can go in any way. Okay. Physical examination, you have something called IE. You might know about this particular one, which is nothing but what is my inclusion criteria and what is my exclusion criteria. Inclusion as well as the exclusion criteria. So the next thing is going to be DD. This is not mandatory, but it is also required, which is nothing but death details, right? So final one in this, which you can call it as a DA, this is nothing but drug accountability. All these domains, in each domain, what are going to be the data sets? In each data sets, what we are going to, what are the various columns are there, and what is the meaning of each and every column that we are going to discuss. So no worries about it. So the next thing. So these are main, these 4 I can say that main domains. In addition to that, we have something else which is very small one. I can call that particular one as trial design models. So in this we have 5 totally. 1, 2, 3. Okay, let me write all those. We have 5 things in the 5 in this. One is TA. The second one is going to be TS. The third one is going to be TE. The fourth one is going to be TV. And finally, the fifth one, which is nothing but TI. Can anyone tell me what each one of this is basically referring? What is TA? What is your TS followed by TE, TV, TI? Wonderful, Sanchi. Yep. So the first one is going to be trial arm details. Second one, TS. Someone saying trial summary, someone saying okay, just give me a second. Trial summary, trial visits, trial inclusion. Okay, can someone please give me the concrete details about this? Anyone? So okay, Raja, you're saying TI. TI is absolutely correct, which is nothing but your trial inclusion. Okay. TV stands for trial visits. TE stands for trial events. Now the question is going to be, what is TS? So can I say trial summary? You're already given these details. So all these trial design models we are going to discuss. This is a small one actually. It is not going to be like a time-taking one compared to all other domains. This particular domain will not take much time. It is much simpler that we can what to say, quickly complete this. In addition to this, we also have few other domains. You can call them as directly. So maybe if time permits, we go with that. If not, we will not. But 90% of the time, I'll go with this. So those are called supplementary domains. And we also have something called rel-rec domains. So what are these and why we are not talking about the data sets within that? No worries, we can talk about this later. So this, if time permits, I'll go with this. If not, I may give the details and take this. But 90% I can go ahead with the details here. So first of all of you understand what we are going to discuss as part of your SDTM domains. We are going to talk about these domains. We have totally 1, 2, 3, 4, 5. In each domain, we have different set of data sets. All these data sets in detail we are going to talk. Any questions? If anyone is expecting anything other than this, kindly let me know. All clear? All right. So the next topic, who is this? Sanchi PK domains. So what I can do is I can give you the guidance about them. All right, what is PK domain, what and all we have in that, how you are going to read that. I'm going to give you the details about it. All right, this itself will take a lot of time, but still we can go ahead with that. See, naturally, if you're good with 1 or 2 domains, which is more than sufficient. So as long as you are good with these special purpose domains, your events, your interventions, findings, and trial design models, this itself is going to suffice, or I can say that this itself is going to meet all your requirements. Beyond this, if you are looking also, I can guide you how to proceed with them. We have implementation guide. I'll help you with how to utilize your implementation guide. Clear? All right. The next thing is going to be here. Now we are going to talk about the next thing, which is nothing but, before we start with each of this particular domain, we need to understand few things. Those are nothing but SDTM guidelines. What is that? We need to talk about something very important, which is nothing but SDTM guidelines. Just give me a second, guys. This is going to be SDTM guidelines. So let me tell you why this is required, SDTM guidelines. You need to understand, if I say domain, within the domain, what and all you have. I mean to say that we open the SDTM guidelines. Anyways, I'm going to share this with you. If you see that, the fundamentals of SDTM, here you can see not even this. Just give me a second. I'll take you to a page so that you can understand. Can you see this? This is your demographic data. DM examples, demographic data is there. Just assumptions. So this is the first model, right? Because in the special purpose domains, the very first one is going to be demographics, correct? So if you see the demographics, you know what is called variable name. So what is variable name? Which is nothing but the column name. Within the data set, what is the column name is that is variable name. So since we already understand what is called SAS and all, we can easily talk about this particular one. Second, variable label. Label is nothing but we all know that our SAS dataset and SAS variables have specific rules, correct? I cannot have space there, and I can also I cannot go with more than 32 characters long as well. So to make sure everyone understand what is this particular study ID or USCDJ ID, what exactly it is a unique subject identifier. So to give a name for it, we are going to put a label. You can understand this just like here. In your kitchen, you can see there are a lot of boxes will be available, nothing but your containers, correct? So for each container, you're going to put a label just to understand quickly, correct? So if the label is going to guide you what exactly it is. Am I correct? So that label we are talking about. Allow me a moment, guys. Sorry, guys. Totally sorry. There was a background noise. That's the reason I just put you all on the mute. Sorry about it. Okay. So now let me continue. So what we are talking about. So this is how your demographics data looks like. Correct. So I know what is variable name, and I know what is called variable label, and I know type, because SAS has only character as well as the numeric. So totally acceptable. Now, can you see the next one? Controlled terms, code list, or format. So simply I can call it as a format. So this is format is also fine. But we need to understand what is called this role, what exactly it means, what is the score is all about, right? And also we need to check about what is the CDISC notes is all about, how to prepare it, how to understand this particular one. These are very, 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 very important. Correct? So along with that, if you also see this in your previous slide, you can see all your domain names, all your dataset names are going to be like, you just have only 2 letters. Correct? All the special purpose domains, everything you can see, it only have 2 names, 2 letters. Correct? So we have a standards even in defining these things. Those standards we need to understand. So the very first thing that we would like to understand here is going to be the first thing. We need to understand that as per SDTM guidelines, domain name, all the domain names should be contain 2 letters. All I can say that it should contain only 2 letters. Whatever the domain you take, you're going to have only 2 letters. That is the first thing. Number 2, every domain should have a proper label. What is that? Every domain should have a proper label. What do you mean by label? Only when you have a label you can understand it. Otherwise, how do you know that particular one? Correct? And here there is another guidelines, that is maximum is going to be 40 characters. That is, whatever the label that you are going to put, it should not exceed 40 characters. The first one. Correct? The second one, whatever the data, whatever the label that you are giving, the label should be in a prop case. You know what is prop case like? We have uppercase, lowercase, as well as the prop case. So it should be in a prop case. These two are the mandatory things. All right. Number 3. If you want, you can check here. Just a quick comparison. Can you see this? DM. What do you mean by DM? Description or overview for the demographics domain model. Correct? That is nothing but your label. This entire thing is going to be a label. Correct? Next. Number 3. You also need to understand when I talk about the variables, all the variable names, it should be, or I can say that the variable names should not exceed, or variable name should not exceed 8 characters long. What do you mean by that? For an example, I can go with some of the variable names. Okay. So what are the variable names? I can simply say that I have something called here, this itself you can refer. I can say that unique subject identifier. This is the label, but what is going to be the variable name here? I simply have use subject ID. Correct? Simply I can say USUBJ ID. I can have something called age. The age is going to be 3 characters. Fine, because which is within the permissible limits. I have a variable called sex, which is also within the 8 characters long. And you also have some of the variables which are 8 characters. Can you see this particular one? That is date and time of last studies treatment. You can call it as a RFX and TC. TC stands for your timestamp. Both time we are going to have your time as well as the date. That's the reason you can call it as a timestamp. So this is end, this is going to be start. So there are 2 things. Right? So we are going to, let's take this particular example. In this particular example, I can see that the maximum length is not exceeding 8. Correct? So the first thing is going to be, we need to understand that whatever the data is there, whatever you are preparing there, all of them should be under the compliance. That can be a domain name, that can be your data set name, or that can be your label, or that can be of your anything that for the matter of the fact, even the format, anything for the matter of the fact, it should be of as per the guidelines. So I'm just keeping that here. That is final outputs, because we are creating the standard SAS datasets, right? So the final output domain also, the final output domain also should be in compliance with, or I can say that final output should be in compliance with SDTM implementation guide 3.2. Correct? I don't know whether you have identified or not. Look at this particular one, especially look at this particular, all the variables you can see here. Did you find anything odd? Did you find anything odd? Mean to say any variable or its name or it is going to be type, anything do you feel odd here? Anyone? Okay, let me point it out. Take this particular row. Now on this particular row, did you find anything which is odd? Variable names are in caps. Okay, that's fine. A good identification, but still that is fine. Anything other than this? I have highlighted the row as well. Maybe ISO 8601. Okay, ISO 8601 is going to be your date format. Wonderful, wonderful. Now it's a good catch. See this? Either you can consider this one, or you can consider this one, or you can consider this one. All these are date and time, right? In general, the date and time format or date and time must be in a date is in a numeric format, correct? But can you see this here? Everything is in a character format. So you need to understand this particular one, and you need to remember this. That is nothing but all SDTM date variables are of character data type. This is very important, right? For an example, I have a date, right? Let's say I'm just giving a date, today's date, 2022 04 followed by 07. This is our today's date, correct? What is the format here? Can I say that this format is nothing but what is the format of this, guys? Anyone can tell this? YYMMDD 10. This is the format. Either you can use this particular format, or in SDTM we use a different format, and that different format is nothing but the one that you have identified, which is nothing but ISO 8601 followed by DA. So which is nothing but it is going to be of the same format that you can see it at the top. That is nothing but 2022 0407. Both the formats are going to be the same. If you want to represent the both date as well as the time, that is nothing but the timestamp. In that case, I'll go with ISO 8601 DT. This stands for DT stands for timestamp. For an example, 2022 0407 followed by time, the timestamp. So what is the current time now? 9 o'clock exactly. So which is going to be 21:00:00. So this is nothing but you are representing the both date as well as the time. Is that clear, guys? All of you are able to follow this? Can I have a quick confirmation? Domain name should be only of 2 characters, 2 letters maximum. Every domain should have a proper label. It must be a prop case and should not exceed 40 characters long. Variable name should not exceed 8 characters long. Whatever the final output is there, it should be in compliance with the SDTM implementation guide 3.2. All SDTM date variables are of the character data type. Questions? Any questions from anyone? It's not ended here. Still, there is some more guidelines. We need to discuss that. So I just need a confirmation from you. All of you are comfortable with this or not? All right. So let's go with the next one. So now if you go to the implementation guide, now you know the type, you know the format, why you are having 8601 and all. The next thing that I would like to talk is, I want to talk about this particular code, what exactly this code is about, right? And you can see that under the core, you have something called EXP, you have REQ, and you have PERL. So a core is the one which is having three different variables, three different values. So what are those three different values? Number one. So let me talk about core. So the core is the one. Sometimes it's difficult to write here. So we have something called core. So what exactly this core is? There I can say that it defines how you want to utilize this particular variable, or what is the significance of this particular variable. So do you mean to say that this variable is mandatorily required in my data set? Number two, if it is mandatorily, it is part of my data set. So what kind of values that are allowed? That is nothing but constraints. You can call it as a constraints. Number three, if it is not that important variable, can I skip it? So if I skip it, what is going to be the impact? Right? All these three things are defined by your code. For an example, the first one, let's go with the important one. That is nothing but the first type of code is required REQ. Right? So we need to understand what is this, why it is important. So required means I need to say that these variables, if I say that it is a required variable, then I can say that variable must be included in domain. Whatever the case it is, it should be part of your domain. And also, this is the first case, variable must be included in the domain. Number two, the second important thing is going to be value cannot be null. Two things are important. What is that? The first thing, it is required, most important, no doubt about it. So I can obey that particular one. Second thing, I also need to understand that this particular variable, right, it must have it. It must have it. What is the best example? What is the best example? Let's say I'm just going with the demographics. This is my demographics information. Wonderful, Sanji. Use subject ID. Yes. Let's say a unique subject identifier. What is nothing but how you are identifying your subject based on your, what is that? USUBJ ID, unique subject identifier. What is the next thing? Let's say that I'm just having another variable here called age 6, and I also have age. For an example, under the unique subject identifier, I'm going with 111. So sex here is going to be male, and we also going to have my, let's say, age is going to be, let's say, around 30. So there is another guy. Let's say that this guy name is going to be, this subject ID is going to be 222. Let's say again M, and here it is going to be, yes, 35. And then let's say I have one more. This here I have 333. So female, and let's say that 28. Now, my first question is going to be, among these three, let's say, if I don't include subject ID, if simply I just have sex and your age, will that make any sense? Will that make any sense? Whose age it is, right? Who is this, male or female, right? So does it have any significance at all? Not at all, right? There is no significance at all, right? So what you need to understand from here, this is one of the mandatory variable. Without this particular subject ID, unique subject identifier, this table doesn't have any sense. So it is a mandatory variable for me. So it is a required variable for me. Even you can see here. Unique subject identifier, you can see it is a required variable. Without this, I cannot, because which subject it is, I cannot understand. Correct? Second, okay, can I have this particular value as a missing value? I have the variable name, but I don't have a value for it. Will that be any good? No, right? It should have a proper value as well. It should be a proper, it should have a proper value. At the same time, it should have a, it must be part of my domain. If I have such kind of variables, I can call them as a required variables. And if you look into this particular thing, your DM, the demographics one, around 30 variables you can see there. Among the 30 variables, around 10 are going to be your required variables. These are mandatory. These are supposed to be part of your thing. Okay. Now, the second thing is going to be expected. EXP stands for expected, right? So let me tell you what is this expected is going to be. Number 2, I have something called expected, which is nothing but EXP. So it is good to have. The value can be good to have. The value can be good to have, but when I talk about the variable must be included in the domain. Still, I cannot exclude the variable. It should be part of, right? Value can be missing. Value is good to have, meaning missing values are acceptable, right? Meaning I need to have this particular variable. Variable must be part of this particular domain, but I can, even if I don't have any if I don't have any value for it, that is fine. Maybe I can give you a different example here. Let's say that we have something called death date, right? So we have something called death date. So it doesn't require to have death date for each and every patient, correct? But we need to capture if someone is getting affected and if this particular person is about to die or he already dead, right? We need to record that as well. So death date is a mandatory field, but it may or may not have the value, but it is totally acceptable. Correct or not? Number 3, we have perm, which is nothing but permissible. Which is nothing but permissible. This is nothing but it totally depends on your sponsor. What is that? Totally depends on sponsor. If sponsor want to have it, we can keep it. If totally depends on sponsor to include or exclude this particular variable. Totally, it is depends on the what to say sponsor. One of the example again we are talking about this, right? This also we need to see the values. If the values are good to have, if the values are missing, that is fine. We don't have any issues with that. For an example, what are going to be the examples for this? If someone asks me the example for it, I can simply say that, let's say we have investigator, correct? I really don't need to put investigator name as part of my domain. I don't need. If you keep it, okay, fine. Who is investigating this? Who is going to be the doctor? If you don't put it, no problem at all. Correct? So I can simply say that investigator name, maybe I can use it, may not be use it. That's absolutely fine for me. No issues at all. I can use it or I may not be use it. Correct? So can I say that this is going to be every one of you? Are you able to follow this? Can I have a quick confirmation? Simple. A very simple one that we are talking about here. All we are talking is that is whether Core is nothing but how you are dealing with your variables. Do you want this variable to be part of your output or not? Sorry, in the table or not? If yes, what is going to be that? What is going to be the significance of that? Clear, everyone? Can I have a quick confirmation on this? All of you are able to follow this? Yes or no? Okay. All right. The next thing is going to be still this SDTM guidelines is going to be continued, and what we are going to discuss here is, so now if you go to this, we know what is called code. We still need to understand two things. One is going to be role, another one is going to be CDISC modes. So under the SDTM guidelines, so the next topic that I would like to discuss is what is called role. So every variable should have a rule. It is at a variable level. Every variable should have a rule. Now the question is going to be what exactly this rule is all about. Okay. So first thing. So there are 4 roles available there. Again, number 1, you can call it as identifier variables. Number 2, the role can be identifier variable. It can be a timing variable. All right. Number 3, it can be a topic variable, or it can be rule variables. In addition to that, we also have some qualifiers, right? We also need to talk about the qualifiers. Under the qualifiers, again, we have 5 qualifiers. Maybe we can talk about these qualifiers little later, because you can see here the record qualifier, synonym qualifier, you can see the timing variable, right? All of them are there. So it is also integral part of this particular one. So it's better to understand what is this qualifier as well. So let me talk about it. So under the qualifiers, the first thing is going to be record qualifier, or number 2, it can be a result qualifier. Number 3, you can also segregate this as a grouping qualifier. Number 4, it can be synonym, right? Or it can be a variable qualifier. So these are the various qualifiers, and these are going to be the various roles. So the role can be identifier, it can be a identifier variable, or it can be a timing variable, or it can be of your topic variable or a rule variable. Again, it can be a qualifier as well, right? It can be a qualifier as well. Under the qualifier, you have record, result, grouping, synonym, as well as the normal variables. So this part we are going to discuss tomorrow. In addition to that, we are also going to discuss about the CDISC modes. Correct? That is nothing but this. So we are going to talk about the role and CDISC modes. Once we have completed this, we can say that SDTM guidelines is completed. From Monday onwards, we can start with the very first domain, which is nothing but the demographics domain. So as I told you, by tomorrow, all the theoretical topics, the basic guidelines which are required is going to complete. From Monday onwards, it is purely about the domains. Clear? Any questions, guys?
Murli, I have just one question regarding the permissible thing which we spoke. As you mentioned that the sponsor, it depends on the sponsor if we want to keep that variable or no. So
Correct.
when we start with the study, does the sponsor give us the list of variables which would be permissible, or we as programmer have to decide, or we have to have a meeting with them and decide on those variables? How does it work?
No. See, look at from the demographic side. Let's say
Okay.
your sponsor, you can see this. This is my demographic, correct?
Mm-hmm.
Whatever you can see it on the screen, these are my demographic variables, right? So now among this, during the validation, all you need to understand is, let's say first thing is you are going to cleanse your data, you're going to do the data validation. Only after that you're going to work on your data, correct? So your sponsor has given these data sets, raw data sets. Sorry. Now, your sponsor has given these data sets. The first thing that you are going to see is, first you are going to see your data set has all the required variables are there or not. That is required as well as your what is the second one? Your
Expected.
expected variables, EXP variables. Both of them are part of your data set or not. That you can quickly validate by using your proc contents. Correct? It is going to give you the detail. Second, once you have this, the second check is going to be for all the required variables, you are going to check, do I have any missing value there or not? If I have the missing value, I can say that my required variable is part of my data set, but it has the null values. So fix it. So first itself, you are going to send these details back to your sponsor because you don't have the required data. After this, you really don't bother about your permissible variables. If it is there, good. If it is not there, you really don't bother about it because your sponsor is not interested to keep that particular one. Let's say you have your date and time of birth, right, of a subject. The sponsor can keep this variable as part of the data set. If you can't keep also, if you don't have this particular variable in your data set also, it is not a complainable thing. That's fine. It doesn't have impact. Is that clear what I'm trying to say? Right? It is only for the validation purpose. Right? You are not going to have a specific meeting or something. When you are going to have a meeting is going to be your required variable is missing or your permissible variable is missing. You are going to ask him. Your required variable is there, but value is missing. Still, you can send it back. In all other cases, you continue with what you have there.
Yeah.
Clear?
Okay. Yes. Okay. Actually,
Any
I
other
think this question was with respect to when we are creating the mapping specification. In case there is no reference to the study and the study is very new, in that case, when we prepare the mapping specification, that time I think even the permissible variables would be coming into picture. So in that case, how should we take a decision whether those variables need to be there or we have to remove it? So I think if it is there in CRF, we take it. If it is not, then we won't take it.
Exactly.
Is it correct? Yeah. Okay.
Yes. Because it is part of the CRF mean to say that from the subject it is recorded. Correct?
Yeah.
You collected the data from the subject.
Mm-hmm.
So it is available. Right?
Yeah.
Let's say you collected the birth date information of a subject. It is part of the CRF, loaded.
Yeah.
Right? If it is not there in your CRF, mean to say that your annotated CRF have all the information, but this data value is missing there. So then what do you understand? Mean to say that the sponsor is not giving this particular details to you. So you don't need to go with that.
Yeah. Understood. Yeah.
Perfect.
Yeah. Thank you.
Welcome. Any other questions from anyone? Guys, clinical domains are a bit harder. Mean to say that not to learn, but to listen, because these are very raw in nature. Because of that, you may feel difficult in the initial days. Mean to say, for the first 1 week of the classes, it will be little difficult, right? Once we started with the individual domains, it will be very quick. We can we can increase the pace and we can quickly cover the topics, and at the same time you also keep on looking at your assignments and all. I'll keep giving the assignments once you started with your first domain, so that it will be easy and you will also get the interest. Right? So till that, these are very important stuff. That's the reason, especially in the interviews, these are important. That is the reason we are knowing this particular one. Clear? Any other questions from anyone? All right, guys. Thank you, everyone. See you in tomorrow's class then. Santosh, is there any tips? It is too early to discuss that, but definitely we are going to discuss. No worries. It is a bit hard. Even I'm not from the clinical domain, so but I have learned about it. So no worries. Don't worry. Only initial days it is a bit difficult, but after that it will be a smooth ride. So don't worry about it. Okay, guys. See you tomorrow. Thank you. Hey, hello everyone. Good evening. Welcome. All right. So let's continue with where we left yesterday. If I'm not wrong, the last topic that we have discussed about was about the code, correct? We were talking about the SDTM guidelines, and as part of that, we have completed the course and we are about to discuss about the rule. Am I correct? Is this what, sorry, roles? What is the variable roles? That is what we need to discuss. So any questions on the previous classes, guys? Anyone? We're talking about the SDTM guidelines. So when we talk about the SDTM guidelines, we have started with the very basic terms, like how the things are going to be, how we are going to define them. Those stuff we have started, correct? So let me give a quick recap of what we discussed yesterday, and we can proceed with the today's session, right? So yesterday we started with the SDTM domains. That is nothing but what is going to be the overview of the domains that we are going to discuss. So we have primarily special purpose domain, events, interventions, findings, trial design models, supplementary domains, and also rel-rec domains. These are the various domains that we are going to discuss as part of our sessions, correct? This is what we discussed yesterday, right? So then, but in detail, we are not going with that right now. We are going to take each of this particular domain and we discuss in detail. All right. So, but before we go with that, we need to know few standards. Those standards we are calling as SDTM guidelines, right? So we all know that observation, a simple observation is nothing but a row in a data set. A domain is nothing but collection of observations in a particular topic. Nothing but a data set. You can consider that as a data set only, more or less, right? A variable is nothing but a column in a data set. Role, because the role is nothing but what exactly this particular variable is trying to convey, right? What information this variable is actually providing to us, that is nothing but the role. So the role is the one that we are going to discuss today. No worries, we haven't discussed this for this class. So first, a domain name should contain only 2 letters. As per the SDTM standard guidelines, any domain should contain only 2 letters. That's the reason you can see here, demographics DM, comments CO, subject elements is going to be SE, SV, AE, MH. If you see all of this, the domain name is only of 2 characters. Irrespective of whatever is going to be the domain it should be of 2 characters. Number 2, every domain should have a proper label. So that I discussed, that I have shown you here. Meaning of the label is nothing but DM means what exactly the DM is. How do I know that DM is, what is the meaning of it? Correct? So that information will be given as the label. Can you see this? SC stands for description overview for the subject elements domain. Correct? So again, we also discussed about the first one, demographics, DM. So what do you mean by DM? DM stands for specifications for the demographics domain model. It is not all about specification for the demographics domain model. Or you can also see at the topic, these are the specification. Sorry, this is the label. Description or overview for the demographics domain model. So everything we have a proper label, correct? Only based on the label we are going with that. And the label should also have maximum of 40 characters. It should not exceed more than 40 characters. Then variable names, that is the columns, right? The columns in the data set should not exceed 8 characters long, right? Let's say you have unique subject identifier. You simply call it as uSubject ID, right? So we can also have your age, sex variable, and you can have start date and timestamp. All these variables, whatever the name it is, you need to restrict that to maximum of 8 characters. Then the final output should be in compliance with the SDTM guidelines. You need to say that whatever the output that you have, whatever the final output that you have, the final output should be in compliance with the SDTM implementation guide 3.2. If you are following 3.2, it should be 3.2. If you are following 3.3, it is going to be 3.2. Throughout the design or throughout the course, it's supposed to be the same version. We have to follow the same version standards. Most important thing, yesterday I have also shown that here, if you see all your date variables, even though the date variable is a numeric in SAS, as per SDTM guidelines, the date variable's data type is character. That is, you are going to represent them as a character. The general format that you are going to follow is going to be Y by MM BD10, but you can also use ISO 8601. DA stands for the regular date, and if you go for the DT, it is going to be the timestamp. Then, under the SDTM guidelines, we look at this particular one. We know what is variable now, what is going to be the variable label. Label is nothing but a representation of the general name of this particular one, because you cannot have more than 8 characters. So you are going to use it as an acronym. What exactly that variable is going to talk about, what is the meaning of that particular variable, can be mentioned with the help of the label. It's a very generic one. Type is nothing but, is it a character or a numeric? Control terms or code list or format, simply you can call it as a format. Now we need to understand what is called role, what is called CDISC nodes, as well as the code. Yesterday we also covered code. Code is nothing but, so there are 3 types of code. We can call it as a required, expected, and permissible. Required are the ones, if I mention a variable as a required one, it must be part of my domain. It needs to be included in the domain, irrespective of whatever the variable name is. It's supposed to be part of it. And most important thing, along with your variable representation, the variable value should not be null. It should not be null. Means that there should not be any missing values under the required variables. What is expected? This variable is supposed to be part of your data set, just like your required variable, but the values, the missing values are allowed. Correct? The missing values are going to be allowed. What is the third one? The permissible. Purely your sponsor is the one who is going to, your sponsor is the one who is going to decide whether this variable is to be include or exclude from your domain. Correct? I can say that it is good to have. It is good to have. Even the missing values are going to be acceptable, right? But who is going to take the decision about this particular one? Your sponsor or CRO are the ones who are going to take this particular decision. So these are the ones that we have discussed in the yesterday session. So today, as I mentioned, we are going to talk about the rule. So here you can see code is done. Now we have rule. A role, if I want to talk simply, I can say that the role is nothing but, okay, every variable on the CDISC have a role. You can see study ID, identifier, subject ID is a topic, your timestamp, which is going to be a reference start date, and time date is going to be record qualifier. And you can also see here timing, synonym qualifier, right? So there are going to be a lot of things, not lot, I can say that there are going to be 4 different variables. Number 1, let me talk about that. So there are going to be 4 different variables starting with. You can type it in any order. There is no restriction like I should go in this way or the other way. You can go in any order of your choice, right? Just allow me a moment. Just a second. All right. So the first one is going to be, you can call this as a identifier variable, or it can be of a timing variable, right? Or it can be of your topic variable, or it can be also a rule variable. When I say rule, in addition to that, we also have something called qualifier. Along with these 4, we can also represent that with the help of the qualifiers. Again, when you go with the qualifiers, there are different qualifiers. One you can call it as a record qualifier, result qualifier, grouping qualifier, synonym qualifier, and finally we have a variable qualifier. So on a whole, your code, it is nothing but it simply represent what exactly your variable is all about. Meaning what this variable is trying to convey to you. All it is going to tell you is that what it is trying to convey. All right. For an example, let's understand one by one, because each of this is very, very important. First thing, the name itself is telling you identifier. Identifier in the sense, it is going to identify some things, or these are the variables which gives identification information. Mean to say, if I go to my implementation guide, if you look at the very first one, study ID. What do you mean by study ID? Here itself you can see study identifier. What study that you are actually doing here? Domain. It is also an identifier, because is it a events domain, or is it going to be a demographics, or it is going to be a what to say, subject visits, or what exactly it is, right? So by looking at it, you can identify something, correct? Unique subject identifier, nothing but patient ID, correct? It is something like a patient ID, right? So it is all about identifying something, right? It's simply going to give the information about some information that you are going, based on this you are going to identify. So what are the general examples for this particular one? I can simply say that the examples are going to be your domain. Based on the domain, you can say what data set it is actually, what study you are doing, right? What type of study you are actually doing, study ID, right? So what subject it is, what is going to be the unique subject identifier, USUBJ ID, right? Simply I can say that which gives information, which gives some identification, that we can call it as a identifier variables. Number 2, timing variables. Here also the name itself is telling you naming variables are nothing but timing variables are nothing but these are the variables which is representing some date. Exactly. That can be anything. Sanji, you're saying describes the timing of an observation. Wonderful. What that observation can be anything. That can be a trial start date, that can be trial end date, right? Or it can be the date when the collection has happened, that is nothing but the lab collection that we have performed, or it can be the measurements, when we have actually taken the measurements, correct? It can be anything when you have taken the measurements. Simply it identifies, or it gives the information about date, right? For an example, I have a variable called, for an example, I'm just taking the events domain here. In the events domain, I have something called AESDTTC. Exactly. It is going to be date and time, correct? So what is going to be, it simply represents the adverse event start date. What is this? It is going to be your adverse event start date. Or I can also call it as the same thing. I can also take adverse event end date. Meaning to say, let's say I have given one drug. The drug, it started giving some side effects. From when this side effect has started, right? That is your adverse event start date, correct? I also have adverse event end date, AEENDTC, right? Or when I look at the lab, so I have LBDTC. What is LBDTC? When the lab started collecting these particular samples. I have scheduled visits, correct? When I say scheduled visits, I can say, or I can go with vital signs start time. All these are going to be the examples. Simply, I can say that these are the variables which represents the date in a very simple way. Now, topic variables. Topic variables are nothing but, first thing you need to understand is, one domain has only one topic variable. Can have only one topic variable. So what exactly is the meaning of this? If you see this particular one, let's say demographic itself, I can have more than one identifier variable. I can have more than one record qualifier. I can have more than one synonym qualifier, right? You can see here it is, here is the other one, right? I can have more than one timing variable. But when you look into this, the entire demographics domain, I can have only one topic variable. The topic variable is the one which will focus the entire meaning of the domain. It focuses on the entire meaning of the domain, right? It specifies the focus. Okay, exactly. So Sanchi, right? It is going to give me the information about entire meaning of this particular domain. So the demographics is all nothing but, I can say that it is all about the subject, nothing but patient, correct? The subject ID is going to be my topic identifier. Suppose if I go with the other one, comments, correct? I have coval. Coval is going to be nothing but a comment that are provided by your investigator. That is going to be my topic variable, right? If I say that I'll go with another one, for an example, subject elements, SC domain. There, the element code is the one which I can call it as a topic variable. So let me write it. And in timing variable, EXENDTC is also there. Yes, it is there. So what do you mean by, what are you saying? Maybe are you giving information, Raja, or you're asking something? Are you asking me this EXENDTC is part of your? Okay, it's there. It's there. I couldn't understand your question. Sorry about it. All right. Yeah. Are you asking whether it is there or not? Means it is there. All right. So remember, guys, you need to understand, and you also need to, let's say that all the topic variables for all the domains should be on your fingertips, right? And I'll give you a simple way or a simple trick to remember those. All right. So let's go with them. So for an example, I have special purpose domains. Try to remember these. These are very, very important. Correct? So what are the various special purpose domains we have? One is demographics. So when I go with the demographics, only for the special purpose domain, try to remember them. So here we already know that it is nothing but my subject ID. That is my topic variable. I have comments. For the comments domain, it is going to be coval. This alone you need to remember, guys. Only for the special purpose domains you remember. For the remaining ones, I'll give you how you can identify them. Then I have SC, which is nothing but your subject elements. When I go with the subject elements, here I have ETCD. The ETCD stands for element code, which is nothing but the element code, right? Because I'm talking about the element, so the element code is going to be the topic variable. Then I have something called here subject visits. When I go to subject visits, the visit number. So the visit number you are going to call it as VISIT-NUM. So for this special purpose domains alone, remember. For the remaining, I'll give you the trick about how to remember them. So I am going to have the second thing, which is nothing but interventions, right? Interventions domain. So under the interventions domain, can I say that intervention is all about all treatment related stuff is going to be part of this? It have information about the treatments, all treatment related stuff, correct? So maybe if you are not aware of it, just remember that interventions is all about the treatments. So remember this code XXTRT. TRT stands for treatment, and you have two X's. Just remember those two X's. So for now, here I have EX. EX is nothing but your exposure domain, right? You already have your code, right? XXTRT, that is nothing but exposure treatment. Just replace the first two letters with your domain name. That's it. My domain name is exposure. Maybe I'll write it here. Exposure is the domain name, right? So for this, the topic variable is going to be TRT is going to be the common one. The first two letters I simply replace with the domain name. Right from here after, you don't need to remember. Let's say I have EC. So what is EC? EC stands for exposure as collected. Exposure as collected. Now you know the topic code for it. What is the topic code for it? Can someone please tell me? Exactly. The XTRT is going to be the same. The first two letters I'm going to replace with the domain name. Right. So what's the next one? I have CM. The CM stands for concomitant, right? Concomitant medication. When I go with the concomitant medication, here also you understand how to represent this. Only the first two letters are going to be CM followed by TRT. Exactly. The last one, SU. SU stands for substance use. When I go with the substance use, here also you can simply replace this. How you're going to replace? I'll replace this one with SU followed by TRT. Easy. Only for the special purpose domains you need to remember. From there onwards, you don't need to remember, because if you know what this domain is all about, you can easily form that particular topic variable. All right. So now we have one more domain. What is the other domain? Can I say events? What is events is all about? Something has already happened, something that already occurred, correct? Something already happened. The event already happened. That you are going to see, correct? So can I simply say that it is already a term? Exactly. It is already a term. I can simply say that XX followed by term, TER. Since it is a treatment here, I use TRT. Since it is going to be event is something which is already happened. Ankita, what do you mean by concomitant? We can discuss about that in detail when you go with the individual domains, right? Each domain we can discuss completely. Like, let's say that if I consider DM, what are all the variables which are part of this particular DM? What is the meaning of each variable? How to derive it if it is a derived variable? All of them I'm going to explain. So don't worry. All right, for now, even if I go with this particular one, you may forget. It is going to be a deviation. So let's wait for this. We can discuss each of this individually. All right, now something already happened. I can call it as a term. So it is going to be XX term. So what are all the various things I have here? One is adverse events. AE stands for your adverse events. The reason why I am writing again and again is you need to hear this again and again. You need to remember this. This is very important, right? So all your domain names and all your topic variables should be on your fingertips. So what I have done here, I have turned the two X's, the first two characters, I have replaced them with the domain name. What is the next thing? I have MH. MH stands for your medical history. So when I go with the medical history, I can simply say MH term. And then I have something called your DS. DS stands for your disposition. When I go with the disposition, it is going to be DS term. And then I have a DV. DV is going to be protocol deviation. Correct? Protocol deviation. So when I go with the protocol deviation, I can simply say that it is going to be DV term. And finally, the last one here is nothing but CE. The CE stands for clinical event. So when I go with the clinical event, it is going to be CE term. All right. So all the events also done. So the next thing is going to be the thing is going to be findings. Findings, the name itself is telling. It is all related to the test category. Correct? You test something and you are going to find something. Correct? So all these are related to test category. So what is going to be your topic variable here? How we are going to form? Can I simply say that XX test code, TESTCD test code. Each test have a specific code. So the first one is going to be LB, laboratory. So when I go with the laboratory, I can simply say that it is going to be LB term. Right. So the next thing, VS. VS stands for vital signs. So when I go with the vital signs, it is going to be VS term. Test code. It is going to be VS test code. Correct? So the next thing is going to be EG. EG stands for ECG test. So when I go with the ECG test, the name itself again is telling EG test code. So what's the next one? PE. PE stands for physical examination. So the physical examination for domain, it is going to be PE test code. So the next thing is going to be IE. IE stands for inclusion and exclusion. So both of them we are going to talk about. That is inclusion as well as the exclusion criteria not met. So for this particular one, it is going to be IE test code. So from next class, healthcare encounters is part of events. Yes. Raja and who and all having idea about your clinical terminology. Whatever we have here, we're strictly going as per CDISC, that is SDTM standard guidelines, and also we are not going with each and every domain. The domains which are very important to us, the domains which we generally work on, only those domains we are actually discussing, right? And generally, it won't go beyond these boundaries. There are a lot available, but we are discussing about the ones which are most important and which will be part of your day-to-day work. Then DD. DD stands for death details. So when it comes to the death details, it is going to be DD test code. And the last one, DA. DA stands for drug accountability. Try to remember all these domains, guys, please. Okay. So now it is going to be DA test code. That's it. Now you see here, only for the special purpose domain you need to remember. For all other, you actually don't need to remember. If you know what that particular domain is all about, what they deal with, right? Automatically you can form your topic variables for that. Oh, sorry, still we have one more, which is nothing but your trial design, right? Trial design domains are there now. Only even for that we are going to discuss. For trial design domains, there is no, I can say that code or something, but we try to understand them and we try to remember them. Let's say TA. What is TA? TA is nothing but trial arm, correct? So since it is going to be ARM is nothing but your ARM code, so I can simply say that ARM code, ARMCD. So we have TS. TS stands for trial summary, right? So here the parameter code is the one which is important. So we are going to end up with saying that TA param code, PARM CD, trial parameter code. All right. Then I have TE. TE is nothing but your elements, correct? Trial elements. Now you can see you have elements here as well, where subject elements. So I have subject elements here and I have trial elements here. Both of them are dealing with the elements, right? So whatever is going to be the topic variable here, the same topic variable will come here as well. All right. The next one, TS, sorry, TV. TV is nothing but trial visits, right? Same applicable. What is that? I have subject visits here. The subject visit has visit number. The same thing will be the topic variable here as well. All right. What is the next one? TI. The TI stands for trial, inclusive, as well as exclusive. All right. Here also, again, you have this one here, right? Somewhere we have already mentioned this particular one. Here it is. Inclusive, exclusive criteria not met. What is the topic variable for this? I have IE test code, right? The same IE test code will be part of this. So now you guys tell me, is it really difficult to remember this? Only for the special purpose domains you need to remember. For the rest of them, if you know what the domain is all about, you can easily one. So the reason why we are discussing this in detail is the topic variable is the one which will simply give the whole information about the particular domain. So that's the reason it is important. That is the reason you need to understand the domain as well as the topic variable. This is supposed to be on your fingertips. Questions? Any
Murali,
questions
for
from anyone?
TI, for
Yes.
TI and the other one where we wrote inclusion and exclusion. Yeah. So there, here we have IE test CD. So we can also have EX test CD, right? Where we have inclusion. Okay.
No.
No.
See, for this particular one, for this IE domain, maybe I can show you here. So in detail we can have, but it is not like, since it is inclusive, I want to go with I. If it is exclusive, I want to go with E. No, it is not like that. So here I stands for inclusive, E stands for exclusive. So it is for both of them together, it is IE only.
Understood. Yeah.
It is IE only. And we are not doing this. This is, we are defining as per the CDISC.
Yeah.
It is part of the CDISC. We are using that as is. As is. Clear?
Yes. Understood.
Perfect.
Thank you.
Any questions? Yeah. Any other questions from anyone? All right. Now, so now you know what is called your identifier variable. We know what is called timing variable. We also know what is called your topic variable. Three variables, three types of rules, three types of roles are already done. The fourth one is rule variable. Here I have rule, right? So the name itself is telling, which gives information about start rule and end rule. When the rule is actually starting and that specific information. Start rule and end rule information. That is part of your. Okay. So let's talk about this particular one. For an example, I have trial elements. Let's understand this easily. Trial elements. So what this trial elements is going to tell you, Maybe we don't have complete idea on it, but simply I'll give you two variable names there. Let's say trial TESTRL. I have TENRL. Now you can see TE stands for trial elements, RL stands for rule, ST stands for start, EN stands for end. That is nothing but when this element rule start, when this particular element, okay, or I can say that element start rule, when it actually started, when it actually ended. So for everything we are going to have this kind of information. For an example, you have TV. TV stands for your trial visits. So when I go with the trial visits, here also I have two things. I can simply call it as TVSTRL. What do you mean by TVSTRL? The name itself is telling to you trial visit start rule. When this particular rule started, when this particular rules ended. TVEN. Simply, so we have a bunch of rules. So when this rule started, when this rule ended, it gives the information about the rule. Understandable? It only gives information about when the rule is starting and when it is ending. All right. Now, we also have something called qualifier, correct? So let's go with the record qualifier. First one. Record is nothing but collection. You're recording something. Okay. From whom you will record? Okay. Go here. Let's look into your demographics. Yeah. So here it is. You can see something called record qualifier. It is nothing but actual arm code. Right? Okay. Maybe even we can take some simple one. Yeah. This one, see this. Sex, race, ethnic, birth date and time, age. Who is going to provide this information to you? From whom you can get this information? Can I say from the subject itself, right? Right? Even if you say that, let's say your sponsor is there or whoever it is, whoever is bringing all these subjects to the picture, right? In the screening, first we are going to do the screening, right? We have ICF, that is consent form. Once the consent form is signed, you are going to do the screening. While doing the screening, you are going to take the information about what is the birth date and time, right? So when this particular person, what is the age of this particular person, sex value, that is either female or male, what is the race, that is nothing but where exactly this particular person is part of, right? He's Asian or he's from the, what to say, US continent, exactly what is the race, correct? Or sometimes you can also go with black, all this kind of stuff. Ethnic, all this information, the record qualifier means those variable information we are actually collecting from the subject itself. So simply I can say that we ask subject, that is nothing but patient, we actually ask subject and record this information. Am I correct? In a very simple terms. Right, we don't need to do anything. We simply ask the subject, either we can record or collect this particular information from the, right, from the subject itself. Exactly, Sanji. From the patient will get this information and the investigator will be the one who will document it. During the screening itself, we are going to document this. So what I can simply say that this is the information which we are getting from the subject itself. So what are the examples? The examples are going to be in a very simple way. I have age, sex, birth date and time, right? Your race, all these are going to be the examples for this particular one. Second one, result. It is also easy to understand. The name itself is telling to you this variable contains, right, information about any result. Information about any result. That can be a lab result, that can be your ECG test result, whatever it is, vital signs results, all this, right? Any test if you perform, the outcome, the result you are going to record it. For an example, let's say LBORRES. So you're going to remember all this, don't worry. So what is this LBORRES? I can simply say that laboratory original results. Exactly. We have something called LBSTRESC. Oh no, sorry. It is more than 8 characters. Let's say LBSTRESC. 4 plus 4, 8. Correct only. So it is going to be laboratory standard results. Here you have one more. Let me put that, both of them, let me explain together. Instead of C, I'm going to have N. Both of them are standard results only, but in a character format, in a numeric format. Easy to understand? When we discuss about the individual domains, you remember all this. No worries about it. Till that, just go with this. All right. Suppose this is for the laboratory. Only can you explain for any other domain? I can simply say that instead of laboratory, I'll replace that with VS, vital signs. So what do you mean by that? Vital signs original result, vital signs standard results in character, vital signs standard results in the. Clear, everybody? It is all about information about your results, mostly related to the test, the outcome of your test. Clear? Any questions? All right. Let's go with the next one, which is nothing but your grouping variables. The name itself is telling you the grouping variable is all about it will group the information, which will give you the grouping information. Correct? Maybe we can go with one simple example, then we can easily understand. The example is more easy to explain this. Let's say that there is a lab category. Let's say that there is a category in the lab, that is LBCAT stands for your lab category. The lab category, the test that you want to do is, let's say that it is going to be urine analysis. So when you go for the urine analysis, there are a lot of sub-tests that you are going to do there. There are a lot of tests that they are going to do, correct? Let's say that LB test 1. So they conduct a test 1, test 2, test 3, test 4. But all these tests, all these 4 tests are part of one simple category, and that simple category is nothing but your urine analysis. Correct? I'm grouping all these test information on one category, that is nothing but your lab category. Sometimes for few tests, we also have the subcategories. Let's say LBSCAT. LBSCAT stands for the laboratory subcategories. Let's say there is something called blood analysis, right? My category is going to be the blood analysis. What and all I can do under that? I can go for CBC, complete blood picture, CBP, correct? That is my lab subcategory. I can also go ahead with, this diabetes test, correct? I can go ahead with HB1AC, or I can go with your fasting blood sugar, or I can also go with the PBS. All these 3 are going to be the subcategories which are part of my, these 3 are the tests. Let's say subcategory is going to be, let's say that diabetes. Under this, what are the various tests that we are going to do? I can say the test number 1 is going to be fasting blood sugar. I have PBS, and I also have HB1AC. Correct? All these 3 are the tests, but these 3 tests are groups under a subcategory called diabetes, and all these subcategories are part of the main category, which is nothing but the blood analysis. Are you able to understand now how we are going to do the grouping variables here? Under the lab, if I go with the lab test, there are so many tests, but they will be fall under either a subcategory or the main category. Is that clear? Can I have a quick confirmation from all of you? Is this understandable? All right. Synonym variables. These variables or these qualifiers generally comes to the picture. These are nothing but similar meaning variables. Simply means synonym is all the same, right? Anywhere it is going to be the same. Similar meaning variables. What do you mean by similar meaning variables? When I go with a similar, you are comparing more than one. Correct? What you are comparing then? In that particular case, I can see that, let's say, we have something called AE-term, adverse events terms, followed by, I have something called AE-DECOD. This is nothing but dictionary derived codes. That is nothing but medical dictionary. So we have medical dictionary, right? Just like how we have the different dictionaries for English, we have the medical dictionary. So the medical dictionary codes are different. For an example, let's say we have fever. Fever is the general terminology. In the AE, we are going to just simply mention that, hey, it is going to be fever. But if you go with your medical dictionary and if you search for this particular, disease, you don't have meaning. The meaning is different there. You do not the meaning. You are going to have a different name there. The name is not going to be the fever anymore there. You can call it as pyroxia, right? The fever is the term which you are entering into the AE term, but the same one has a different meaning in your medical dictionary. You can call it as pyroxia, right? Let's say headache. Headache is also a, but in the medical dictionary also we are going to have the same name for it. It is also going to be the headache. Sometimes you are going to have the same name in the medical dictionary as well as in your AE term, but sometimes the names are going to be different. These information you are going to get it from your synonyms. Clear, guys, all of you? Simple. What is the decoded value as per the medical dictionary? That's it. Finally, variable. So the variable qualifier is nothing but these are the variables which contains units. What is that? Simply which contains units. So what exactly meaning? What is the meaning of it? Let's say here age. Can you see? Sorry, AGU. AGU is nothing but a variable qualifier. What it has what this information? Units associated with the age. What are the units associated with this particular age? Correct? Or so what are the units associated with this particular one? Can I say that number of years? Right? The different, you can see the age is different, age U is different. Age means, let's say 34, directly you can put there. But age U is years. Age U is years. Because what is the 34 is? The 34 is number of months, or it is going to be years, or it is going to be days. Correct? So that unit is part of your AGU. Can you understand the difference between age and AGU? One is a record qualifier which you are going to get it from the subject. The other one is a variable qualifier which is representing your units. Clear? Maybe I can go with another one. Let's say that we already have this one here, right? What is this? Lab original results. Correct? So simply I can put lab original results units. Again, I have vital signs, right? Vital signs original results units. It is all about what are the units, on with what units you are actually representing it. Correct? That information you are going to put there. Any questions from anyone? All of you are able to follow this. What is this role is all about? Now you know variable name, variable label, type, format, a role. Also you understand what is called code. The only thing which is missing here is going to be CDISC nodes. I just need to tell you what is the CDISC nodes is. Before I go there, can I have a quick confirmation? All of you are able to understand so far? Any doubts? Record qualifier we get from the subject? No. So record qualifier you don't get from the subject. It is the standard. Let's say that let's say I'm a subject, right? If you ask me how about your age, I can tell you in number of days, I can tell you in number of months, or I can also tell you in years, number of years. That's how many number of units, correct? Subject will not give you the precise information. Let's say I simply say 34. Or you need to ask me, if you are an investigator, you need to ask me, hey, as per the standards, you need to tell your age only in the years. So as per the CDISC guidelines, the CDISC guidelines is the one which is going to define it. Whatever the definition, whatever the units which is mentioned by the CDISC, as per that particular one, you need to record this. So the answer for your question is going to be CDISC is the one which is going to decide the units. As per the units, you are going to ask the details while you are collecting or recording the data from the subject. Ankita, is that clear? When you ask the age, you ask, hey, tell your age in number of years. Like this, you are going to ask. All right. Even if you look at the laboratory, for every laboratory, if you see the standard, if you see any laboratory report, for an example, let's say diabetes itself, fasting blood sugar, let's say if it is going to be 120. The 120 is the number that you have got. What is that 120? It is going to be mg, or what exactly it is, what exactly the unit it is, and what is the permissible range. These two will be as per the FDA, as per the CDISC. Clear? That's the reason you also have the lab results information. You also have your vital signs, original results information. CDISC. It is quite easy and straightforward. I can say that it is all about the guidelines for variables. I can say this and I can tell that. See, study ID. What is the CDISC notes is telling? Unique identifier for the study. So it is giving the information about this particular variable. Right? Can you see this? Unique subject ID. It is telling that identifier used to uniquely identify a subject across all the studies for all applications or submissions involving the product. But the main important thing is this must be a unique number and could be a compound identifier formed by concatenating study ID, site ID, and subject ID. You have subject ID here, you have study ID here, you also have a site ID, that is hospital ID, correct? By concatenating all these three, I'm going to derive this particular variable. So what it is telling, not only it is giving the information about your variables, it is also helping you how to populate that particular variable. Am I correct? It is not just giving the information. For few variables, if it is a derived variable, it is also helping us how to derive that particular variable. Correct? So how to derive or how to populate. Am I correct? And finally, it also gives you what is the exact meaning of that particular variable. Am I correct? It will give the guidance. It also gives the information about how to derive or how to populate. And it also talk about what is the exact meaning of this particular variable. All these three information here there. Correct? So now you know how to go with the domains, and in domain, what is the information is there. All of you are able to follow, right? That's the end of your SDTM standard guidelines. You also have given this particular information here. What is this identified information is, what exactly, how to read it, each and everything I have mentioned here. And in addition to that, this notes also I will add there. So it is quickly you can refer it. Okay, so that's the end of guidelines. So from Monday onwards, we are going to start with our domains. We will start with our very first domain, which is nothing but under the special purpose domains. We will start with demographics. How each variable we are going to define there, what is each variable is telling, all the information, the standard guidelines, everything we are going to discuss under the demographics. All right. Is that clear, guys? Any questions? Can I have a quick confirmation from everyone? From here onwards, we are going with all the things. Trial events and trial elements are same. Oh, you're talking about the domains. Okay, let me show you that. So here it is. I only have trial events. Elements is different, events is different. Event is something which has happened. We are talking about the trial events. The subject elements are different. So we don't have any trial elements here. Questions? All right. Let me share this SDTM guidelines, the notes, and also the implementation guide to you. So please follow that. By Monday, we are going to start our very first domain, which is nothing but the demo domain. All right. So from Monday, you can also expect tasks as well. Questions? All right. Have a good weekend, guys. See you on Monday. Take care. Hey, hello everyone. Good evening. Welcome. Hope all of you can see my screen and hope all of you can hear me. Can I have a quick confirmation, guys? Everyone? Perfect. All right. Hey, I have some laptop issues, so I'm using some new laptop. Backup laptop. That's the reason you can see some disturbances. Like not like every day. I'm not going to use Notepad++ or anything, but try to adjust for today. Okay. It is not going to be a stopping factor for you in any means. No matter it is going to distract you, but just I would like to highlight before we start the class. All right. Now, first things first. Just give me a moment. Yeah. Now, let's start with where we left in the previous session. If I'm not wrong, we have completed all our SDTM guidelines. That is all the guidelines that we would need to proceed with the domains, correct? And we also discussed about on overall what and all the various domains we have. Even that particular discussion we have completed. And I believe the last topic was, just a second, I just want to increase the font size. Yeah. So the last topic was we about to start discussing about special purpose domains, correct? So this is where I have stopped, and I said that we are going to start with our special purpose domains. The first special purpose domain is going to be a demographics, DE, correct? Sorry, DL, demographics domain. So this is the first domain that we are going to discuss, because this is the most important domain, because it is going to talk about the subject, correct? We are all interested with the subject. This is the domain which will talk about the subject. So we will kickstart with our discussion on demographics. Remember, guys, for this strictly I'm going to follow SDTM implementation guide. Remember, for all of us, the SDTM implementation guide is like a Bible. We are going to use that particular one, and strictly based on that, we are going to discuss everything. All right? Because whatever the raw data that we are going to receive, or whatever the Excel files are, the text files that we are going to receive from the sponsor or from your biostatistician, we are going to create the data set, SAS data sets, as per the compliance with SDTM implementation guide. So the current version most of the organizations are following is 3.2. Even 3.3 is available, but 3.2 is the version that people are following currently. All right. So first thing first, demographics. So what is demographics? In a simple way, if I want to talk about the demographics is the domain which will be primarily talk about the subject. The subject is nothing but it can be a patient, right? It is going to be the volunteer who is going to participate in your clinical trials, or simply instead of talking about the patient or volunteer or any other name, we strictly go with the subject. It will primarily talk about all the subject related details. So that is the reason the special purpose domain demographics is most important to us. All right. So last week we discussed about some of the roles. Each column, that is each variable present in your domain, have a specific role. Correct? What are they? It can be an identifier variable, it can be a grouping variable, it can be a record variable, or we also have some qualifiers. Correct? There is a most important variable, we can call it as a topic variable. What do you mean by topic variable? Topic variable is the one which will indicate, or with a single column, it is going to represent what this domain is all about. So since we are talking about the subject here, for the special purpose domain, for the demographics domain, the topic variable is subject ID. SUBJID. This is going to be my subject ID, is going to be my topic variable. So last week in the class I have mentioned to you, a topic variable is the one which will identify your domain. Correct? And also I discussed that it is going to focus the entire meaning of the domain. For every domain there is only one topic variable. Correct? I also given some of the example. For demographic domains, it is going to subject ID. For comments domain, it is going to be coval. Correct? For subject elements, it is going to be element code. For subject visits, it is going to be visit number. For all other domains, let's say interventions, since all the interventions are related to the treatment, it will start something like XXTRT. Correct? Under that, if it is going to be exposure, it is going to be EXTRT. If it is exposure as collected, means it is going to be ECTRT. Correct? And also I told about the events. Events are nothing but which already happened. Correct? Something already happened. So there we are going to have something called XX term. Correct? If it is going to be adverse events, it is going to be AE term. If it is going to be like disposition, DS term. Correct? When it comes to the findings are nothing but the test related. Something related to your test. So XX test code. If it is going to be lab, LB. If it is going to be ECG, EG. If it is going to be death details, DD. Correct? Even for the trial design domains, we have the topic variables. So in a single way, in a single line, if I want to talk about it, the topic variable is the one which will focus on the entire meaning of your domain. So for subject, for my demographics domain, the topic variable is going to be subject ID. And here itself, we also talked about something that is nothing but your identifier variable. Correct? What do you mean by identifier variable? Which can be the name itself is telling, which is going to help us to identify the domain. Correct? These are identifiers. So what are the different identifiers again here? Can I say STUD? Again, I'm going to discuss all this, but don't worry about it. I'm just telling on a whole now. STUYID, study identifier. It also can be a domain. Correct? Domain is also a identifier. What's the next thing? It can be U subject ID, unique subject identifier. USUBJ ID, unique subject identifier. Correct? In addition to that, because I'm just giving you these details because these are the ones which we discussed in the previous class. So for each domain, you should aware of what are going to be my topic variables, what are going to be my identifier variables, and at least what are going to be my recording variables. So what do you mean by recording variables? These are the ones, just the information which we are going to collect from the record qualifiers. These are the ones which you can collect from the subject directly. So what are the various examples for that? Gender, age, date of birth, birth date and birth time, which country, what is the ethnicity, what is going to be the race. All these are going to be the information that we are going to get it from the subject, correct? Right? On a whole, before we talk about each and every variable that is present in your demographics domain, I just try to bring all the variables that we are already aware of, because we discussed this in the previous class. In addition to that, what are all the other variables we can, or other information we can have it here? I told you on the beginning of your clinical trials, when you volunteer, let's say that there is going to be a clinical trial, right? Your sponsor is looking for the patients as well as the volunteers, correct? So there are three people who are involved in the clinical trials. Who are them? The healthy volunteers, the patients who are suffering with the same disease, the patients which are suffering with the related diseases, other diseases, correct? All three groups will be part of my clinical trials. So before we pick these volunteers, we are going to do the screening, correct? So before even going with the screening, I said that there is going to be one form the patient is going to sign, or the volunteer is going to sign, or simply the subject is going to sign. That form will be called it as ICF. Can someone tell me what is called ICF? Anyone?
Informed consent.
Exactly. Informed consent form. What is the main details that we are going to capture there in the informed consent form? Can I say when the ICF is signed? That is ICF signed date. This is the first date because I'm trying to write the date variables here, right? So here I talked about the topic variable. Here I talked about the identifier variables. Here I'm talking about the variables that can be collected from the subject, which are nothing but the record qualifiers. And finally here I'm talking about the dates. When I talk about the dates, some of the dates that are part of my domain is, the first one is ICF date. That is when the subject has actually signed the consent. Okay. What is the next thing? Once subject is signed it, he will go to the screening. Once the screening is completed, we are going to create the arms. Arms are nothing but the different groups, and we have two different groups. Remember, what are the two different groups, guys? I said that we are going to group all the volunteers, subject, patients, and everyone all together subjects. We are going to do the randomization. During the randomization, randomly we are going to take the people and we are going to keep them into two different groups. Yes, wonderful. One is intervention group. What is the other one? Control group.
Control.
Correct. One is intervention group, or you can also call it as a study group. The other one is going to be control group. Correct. So these two groups we are going to create. After that, we started giving the treatments. That is nothing but arms. Right. So the second date that we are interested is when treatment started. What is my treatment start date? That is my next important thing. Correct or not? When the treatment is started. Correct? Simply I can call it as a treatment start date. We try to identify in general what might be the dates. That's the intention here. The other one is, when is the treatment end date? Correct? So treatment you're going to start, then you're going to end the treatment at some point. That can be phase 1, end of phase 1, end of phase 2, or phase 3 or phase 4, or between that you also have your washout period. Correct? So before we give if I'm going with a crossover study design, in the crossover study design, each subject will undergo different drugs. Correct? When he or she going with the different drugs, first of all, before we give the new drug, whatever the initial drug we have given, the drug completely need to be taken out from the body. That is, the effect of that particular drug need to be completely off from the body. So how much time the body is going to take it, that time you can call it as a washout period.
After the washout period, we are going to start the other treatment. Finally, after all the treatments, when the final treatment was done, that is nothing but treatment date. For an example, something went wrong. Patient is no more. So you need to also capture the death date. Correct? Finally, let's say all these trials are completed. Everything is done. All of them are completed. There is going to be a team. The team is going to collect all this information, right? They're going to record all your clinical trials information, and then your medical writer is the one who is going to create the report. Correct? We also talked about it under the various roles. So we also need to talk about DM, demographic information collection date. So before we start with all the variables and all other details, can I say that up to this we are all aware of? These and all somehow here or there we have discussed, correct? Because when we start talking about the subject, when you talking about the various variables as part of the examples, so far we have already discussed these, correct? These are the variables we pretty much aware of. Apart from this, there are a lot of other variables that we are going to discuss. So let's start with them. So first we start with the variables that we are aware, and then we are going to start discussing about all other variables. So in general, the variables which are part of my domain are study ID. Remember, the reason again and again why we are writing this is going to be, we need to make sure that this one is in our brain, right? At any point of time, if someone asks you what are the various variables which are part of your domain, if they give the domain name, you should be in a position to see them. So the first thing, the variable here is going to be study identifier. Then domain, that is abbreviation for your domain. Unique subject identifier. I'll go with each of this separately now. I will explain how to, whether it is going to be a derived variable or what. If it is a derived variable, how to derive it. I'm going to talk about each and everything about it. Then I'm going to have something called subject ID, that is subject identifier for the given study. Then I have INV ID. INV ID stands for investigator identifier. Then I have INV name. So INV name stands for investigator name. And from here onwards, I'm going to talk about the date variables. So the date variables I'm going to talk here are, remember, most of my date variables here starts with R. So RF, IC, DTC. TC stands for your timestamp, correct? That is timestamp in character, date and time in character. End of your variable, if you see C there, it is in character. If you have N, it is in numeric format. So this is nothing but reference informed consent date and time in character format. That is, we talked about informed consent date, right, when we signed it, correct? This is informed consent date and time. We have something called RF, RFP, ENDTC. END stands for your end. This is nothing but reference end participation date and time in character format. No worries, I'll explain each and everything separately again. For now, I'm just listing out what we are going to discuss. And then we have subject reference start date. Subject reference, ST stands for your start, DTC. That is subject reference start date and time. Same thing, I have RF, ENDTC, subject reference end date. That is end date and time. Then I have something called RFX STDTC. Again, ST stands for your start, STD stands for your start date, TC stands for your, T stands for your date and time, C stands for your character format. The same thing will be continue as is. This is something like the first study treatment start date. Will we have multiple study treatments, right? The first study treatment start date. The same thing, instead of STD, if I go with END, that is going to become my, this is going to be the last study treatment. That is the date time of last study treatment. So these are the dates, these are going to be the variables, the basic variables. Now, we have something called ARM. So what is the ARM, guys? Can someone tell me? Meanwhile, I'll write what and all comes under ARM.
Each treatment group will call it as harm.
Yes. So each treatment group we can consider, or even the treatment that we would like to give it to that particular group also will be called it as ARM, right? Either the treatment we can call it as, or the treatment group also will be referred as the ARM. In general, ARM stands for treatment. Mean to say, if I'm giving a drug A to a group of people, then my ARM is going to be group A. If I'm giving a placebo to the intervention group, right, then my ARM is going to be placebo. So the ARM is nothing but the description of a planned ARM. What is the ARM that we have planned, right? ARM code. So ARM code, the name itself is telling you whatever your planned ARM is there, that ARM code. So every treatment has a code. So I can simply call it as a treatment code. You plan something, but when you come to the treatment, you have given a different treatment. I mean to say, you planned something, let's say you want to give one drug to a particular patient, but before giving that particular drug, you thought like the patient is already having some headache or other things. So instead of giving that particular one, you started with a headache tablet first. So your planned ARM is different, the actual ARM is different now. So what is the ARM, or what is the drug that you have given, what is the treatment that you have given to this given patient or the subject is nothing but your actual ARM. Then the actual ARM, so ARM also has a code, that code is nothing but the actual ARM code. Now, DTHDTC, nothing but DTH is nothing but death, DT stands for date and time, C stands for your character, nothing but date or time of death. There is something related to this, the death flag. So if the patient is no more, or the subject is no more, the death flag is going to be yes, L it is going to be yeah. So the flag, death flag. Then we have something related to birth, BRTH followed by DTC. So which is nothing but date or time of the birth in the character format. So we all know that this is death and time related, birth related. Then we all know that all these clinical trials are going to be carried out in a site. So every site is going to have the ID. So site ID. So this is nothing but your hospital ID or clinic ID, where particular this clinical trials is actually happening. That subject, the site identifier. Then I'm going to have the generic terms. The generic terms are going to be age. We are going to have age U. Can someone tell me what is the difference between age and age U? Last class we discussed this.
Age in units.
Exactly. What is the unit, right? Age units and age. U stands for units. Wonderful. Then we have something called race. Race is pretty much explainable. Country. And we have ethnic. So these and all, I can simply say that except the site ID, the remaining 5 are nothing but your recorded variables, correct? This information you can actually get it from the subject itself. Then finally you have DMDTC, which is nothing but a date and time of collection when you collect the information. Finally, DMDY, study day of collection. These two we discuss little elaborately in the subsequent classes. So these are all the variables, correct? Only where can I get these variables? Is there a reference place or something? Absolutely. All you need to do is simply go to Your implementation guide that I have already sent it to you. In the implementation guide, all these details are available. Correct? Pretty much same variables are there. All these variables, you can see all of them I have covered, right? But here the order is different. Whatever I have defined, the order is different because based on our availability, based on our understanding levels, I have defined them. Okay, but we are going to discuss all of them. We are going to understand the significance of each one of this, and we also need to understand how to define them, right? Okay, first start with the most important one. So now we will go in the same order. So what is going to be the first thing that I would like to do, understand here? These three. What are them? Study ID, domain, and use subject ID. Can I say these three are nothing but your identifier variables? So first let's start with these three identifier variables. So before even start this particular one, remember the structure of your demographic domain is, for each subject we are going to have only one record per subject. You don't have multiple records for each subject. Whatever the information that I would like to collect or that I would like to record for the given subject, I'm going to keep it in a single record only. I mean to say that one row for each subject in a straightforward way, one row for each subject. That is the structure. So let's start with your identification variables. Under the identification variables, we start with the first one, that is nothing but your study ID. So when I talk about the study ID, the most important thing that we need to talk is what exactly the study ID is. Study ID stands for the study identifier. What exactly is going to be the study? Okay. Who will define this? Who will define the study identifier, or who will give this particular one? Can I say, or where it will be present? If I go to this, the first thing, study ID, unique identifier for the study. Well and good. Who will give that? Any idea? Wonderful, Sanji, sponsor. So who is going to give the information of this particular one? Your sponsor. So can I say that this will be provided by sponsor, right? The person who is actually funding our entire clinical trials, either the sponsor or the contract research organization, CRO, are the ones who are going to provide this. What exactly it contains? Can I say this is nothing but the name of the study as mentioned in the protocol? Correct? This is nothing but the name of the subject which is actually mentioned in your protocol. As simple as that. Right? Okay. How it looks like? I have already shared one protocol with you last week. Correct? Maybe I need to, I don't know whether I can access it or not because it is a new laptop. Or okay, you can refer in your that document that I have shared with you. If I look for the example, wonderful, Sanji. If I go for the example, it can be of any format or any name. Each sponsor will go in a different way. Let's say I'm just going with one of this, CT1PM CM _0810. Pretty much it is going to be my one of the example. Or I can simply say that there is going to be a new drug that we would like to go ahead with. Let's say Covaxin. All right, Covaxin followed by some dosage name. Let's say 111, 112211. This can be of any name, this can be of any format. There is no specific format for this particular one, but all you need to understand is going to be the study ID is nothing but it is the name of the study as prescribed by your sponsor. The sponsor is the one who is going to provide this. It's simply going to identify your study. Then we have something called domain. Domain is all about, it is simply an abbreviation of domain name. because we all know that as per this your implementation guide, it's supposed to be only 2 characters long. Correct? Simply it is going to be a 2 character long. Always it can be defined in your uppercase. What it is going to mention? Simply it is going to mention it is going to be a abbreviation for your domain name. For an example, if I go with the example for this particular one, for demographics it is going to be DM. For subject elements it is going to be SE. So subject visit is going to be SV. For adverse event it is going to be AE, like this. Correct? A very simple and straightforward one. All we are trying to discuss here is going to be, it is simple an abbreviation for your domain. That is going to be a 2 characters long. It is going to be always, we mention that in the uppercase. All right. Then what is your use subject ID? It is a derived variable. Correct? It is also an identifier. This is nothing but unique subject identifier. Right? Now, the main important thing here you need to understand here is it is a combination of, or it is derived based on 3 variables named. What are the 3 variables? Study ID. The second one is going to be site ID. That is nothing but your hospital ID or where this trial is happening. And finally, the third one is going to be subject ID. Now you can ask me, how do I divide this? The formula for it. The formula is going to be, it is going to be concatenation of, again, the same 3 variables. What are them? Study ID, site ID. Try to write as much as possible so that you remember them. And finally, your subject ID. So simply I can concatenate all this to get this particular one. So you already know what is called site ID is. It's a study ID identifier. On the top itself, I just have mentioned that. And we have something called the other one is going to be your subject identifier, right? All the 3 you are pretty much aware of, right? So enough is theory, right? So let's start with our first task. Let me save this. I can call this as, so DE domain 1. Okay. So far we have discussed a lot of theory. Though it is going to be a clinical SAS class, we are trying to cover this as a CDISC class. Correct? So far no code, nothing have done. So it's an opportunity to write our first code. All right, let's start. I'm giving your assignment number 1. So under the assignment, or you can class task or assignment, whatever you can consider it. It is going to be okay for me, right? So first thing is, let's say I'm going to give you 3 different details. I'm going to give you 3 different details. I'm going to ask you, I'm going to tell you what is my input. I'm going to tell you what is the expected output, right? I need you to do that particular one. All I have done here is, I have a study name because we just talked about a study name, correct? So I have something called my study name here. So input details as per protocol. So what and all I have here is, number 1, I have something called my study name. I have my study name. My study name is nothing but NFP 222 G1. This is my study name. So what is study name? Your study ID simply, correct? This is nothing but your study ID. All of you accept that? Okay. Number 2, I have information about my site details. My site details are nothing but the site is happening somewhere in the, let's say it is happening in the Hong Kong. So I'm just calling as Hong Kong, followed by 111. HK111 is the place where this particular clinical trial is happening. Number 3, I have details about the gender, that is sex. It can be either of male or female. We don't have any other thing. You know, you have transgenders as well, but we are not considering the transgenders. Number 4, the domain that I'm talking here is, let's say the domain name. The domain name is going to be DM. These are the basic details. In addition, I have patient details here or subject details. I call it as a patient now because you need to write as per your SDTM patient details. My patient details are, these are, I can call it as so from protocol. These are the details from protocol. Now I'm giving the details, patient details until. So when it comes to the patient details are in a data set. This is called your data set. So you guys need to create a data set now, right? So the data set is like this. 111. This is your patient ID. This is your patient ID. Then I have gender. Maybe you may confuse, so just you can consider as a patient ID here, right? So when I have patient ID and gender, my patient ID is going to be 111. Gender is equal to M. 222. Gender is equal to male. 333. Female. 444. F. 555. F. 666. M. 777. 1. 888. 2. Remember, 1 stands for male, 2 stands for female. Okay, this is like note. From patient's data set. All right. So all I'm saying here is going to be, I'm giving the details one by one. So this is my input details as per the protocol. What are them? I have a patient information. Patient details are here. That is nothing but I have around 4 plus 4, 8 patients are there. I just have given the gender details of them. For each patient, I just have given the gender details. So I have the gender which is mentioned in a irregular format. I mean to say that as per the convenience the persons has provided them. So we need to convert them as per the SDTM. You need to look at your variable names. You need to look at all other details. You should not miss anything. This is my input. All these are going to be my input. Now all I'm expecting from you is I need the output like this. I'm calling as desired output, or you can also call this as your expected output. My expected output or my desired output is this. I need you to prepare SAS code and you need to share that particular code with me by tomorrow, all of you, without fail. All right, so it shouldn't take more than half an hour. I can say that 10 minutes is more than sufficient, but I'm going with half an hour. Right, so what is my output? You all know that I told you already, in demographic domain, 1 patient, 1 subject. Sorry, for 1 subject, 1 record. Correct? So how many patients are there? 1, 2, 3, 4, and 8. So obviously, I'm expecting my output to have 8 records. Correct or not? I have already mentioned this here, right? 1 record per subject. I have 8 subjects. Automatically, my output is going to be 8 records. Okay. Then, for all the 8 patients, can I say my study ID is going to be the same? What is my study ID? Study ID is nothing but study identifier, right? So you have your study name here. Can I say this will become your study identifier? Can I have a quick confirmation? So everyone saying yes. So for my study ID, this is going to be my first output. Correct? Right. Cool. That is fine. Second, can I say my domain name for all these will be called it as DM? The domain name is also DM, right? That I can call it as a domain, right? Then as per my clinical trials, it is going to be sorry, SDTM, it is going to be domain, so it is DM. All right. After that, what I'm going to have? Can I call it as? Can I? What else I can go for? Maybe I don't want to show my site ID and other details for now. So what I can do is I want my unique subject identifier, USUBJ ID. What is this unique subject identifier? If you go a little up, here I mentioned that it is a combination, the concatenation of study ID, site ID, subject ID. So your study ID is this, correct? Right. This is my study ID hyphen. My site ID, this is my site ID hyphen. What is the third thing? My subject ID. What is my subject ID? For the first record, it is 111. Can I say this is going to be my subject ID? U subject ID, unique subject identifier. And after this, for each subject I want to show, I need to go with the subject ID here, 111. And finally, I want to have sex details here, each gender details, which is going to be either M or F. Yeah. So now I'm going to copy this particular one. I'm going to populate this for 4, 5, 6, 7, and 8. For all of them, domain is going to be the same. For all of them, study ID is going to be the same. When it comes to the subject identifier, here alone I'm going to have the changes. What are the changes I'm going to have? This is 222. This is going to be 333. This is going to be 444, 555, 6, 7, 8. Correct? The same thing will happen here as well. It is not going to go. 111 will be there. It is going to be 222, 333, 4, 5, 6, 7, and finally 8. And most important stuff: female, male, male, female, female. That's it, right? The first one is male, male, followed by female, 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 male, male, female. Can I say this is going to be the expected output for me? I'm going to share this particular one with you. All of you must give me this output by tomorrow. First of all, you understand what is the requirement is? Can I have a quick confirmation from everyone? Yes or no? All right, let me go with the second one. So now let's proceed with the remaining details. Now I want to continue with the next one, which is nothing but the INV ID. What is INV ID? INV ID stands for, this is nothing but investigator identifier. Investigator identifier. Okay. What is on a lighter note? Can I say simply it is going to be doctor ID? It can be a doctor, it can be a clinician, right? Those ID, correct? If I want to give some examples for this can be anything. Generally, guys, again, if I go with the doctor IDs, generally doctor IDs will be generally this will be mentioned in the form of the TIN. TIN is nothing but 9 digits or 9 characters. Each doctor uniquely identified with TIN. Each hospital will be uniquely identified based on the NPI. NPI is 10 character, TIN is 9 characters. For an example, A, B, C, D, E, 1122. This is one of the example for the investigator ID. Number 2, let's say I have INVNM. What is INVNAM? Investigator name. This is nothing but my investigator name. For an example, this can be name of a doctor, clinician, or even also you can call it as a nurse. Correct? All these are nothing but your investigator names only, right? Then the same will be applicable even there. This is also a doctor ID. It can be of your clinician ID. It can be of your nurse ID as well. All right. Then purely it is going to be a character data type. Correct? And now the most important thing, oh sorry, here you don't need it. Anyways, it is going to be a character data type. So I specifically doesn't need to mention that. So if I go with the example, you can go with any name. So I just go with my favorite characters, which I generally follow in the series. I can say that Simon Baker, or it can be of your Megan Boone, or it can be of your James Spider, James Spader. It can be anyone. Correct? Simply investigator names. Now, the other thing that I would like to talk here, because these two are very generic in nature, that's the reason I'm not explaining anything, because investigator identifier, investigator name only. Now I am going with RF ICDTC. Simply, if you want to remember, this is going to be first instant your informed consent form signed date, right? Or sorry, it's not a signed date. Yeah, it is a signed date only. But while explaining it, what we can say is your RF stands for reference. Go with the same acronyms. RF stands for reference. IC stands for informed consent date and time. D for date, T for time. Most important thing in character format. This end C stands for, you see, RF stands for reference. IC stands for informed consent. D stands for date, T stands for time, C stands for character format. What exactly it is? If I want to talk about it, I can simply say that this is the time. This is actually the subject. Okay. When the subject is signed the ICF, informed consent form. When actually he have done this, he or she have done it. That format is the one that I need to remember. Sorry, that is the date that I need to remember. Most importantly, it should be character data type. We also discussed this when we are talking about the standard implementation guide. That is, all of your dates in your SDTM implementation guide are in the character format only. So let's go with the examples. The example is going to be, let's say, 2022. Today's date itself we can take 12/04/12. This is the format, correct? So what exactly the format it is? The format is going to be, what is the format, guys? Either you can call this as YYYY MMDD. The exact format for this IS 8601DA. This is the format, correct? Sometimes I can mention this in the date and time format also. 2022/04/12 followed by T, the timestamp. That what is the current timestamp now? 8:58 p.m. 20:58/12. Meaning, if I look at the format of this particular one, the format is going to be YYYY MM DD T HH MM. Correct? What is the original format name for this particular one? IS 8601DT. Correct? All right. Now, all of you are clear up to this? The very basic details, but we are going in detail. Now, it's a time for another assignment. With assignment number 2. The assignment number 2, the input details, again I'm going with the input details here. The input details are, I'm giving you the first data set. So I have details about the let me take the same one. So I have my patient details. Let me take the same one. It will be easy for you even to prepare the data set for you, right? And then I also have the details from ICF details. The ICF details are like this. Again, it is going to be a data set. The data set here I'm talking about I have my patient ID, term. No worries about the term. Later we can discuss about it. For now, simply follow your DT, that is the date, signed date you can call it as. And I have time, signed time. So the patient ID is, I'm just going with all of them one more time. All right. So when it comes to the term is going to be whether the patient is signed or not, when the patient was signed actually. So here I can say that ICF signed. When this particular patient actually signed the form. So all of them, the term I'm just going with signed only, because without ICF signed, we cannot have the patient to be within the clinical trials, correct? So all of them are signed. Okay, when? This guy, 9th April 2016, 12th September 2018. No, it should be in the almost same ranges, maybe different trials, but all of them I just try to maintain in the same year. Okay, maybe we can consider the COVID scenario. So when I consider the COVID scenario, I can say that all of them somewhere in 2020. Okay, and then I have something called 11 August 2020. Then I have 13th July 2020, 16th June 2020. Maybe I can go with only 5 of them. Yeah. So I'm just going with the time. So all of them somewhere that they signed it at 10 o'clock, 10, 1, 12, system capture time, 10, 10, 13, then 13, 30, 00, 14, 11, 46. Again, I have someone signed by evening 4 o'clock, 16, 15, 40. Okay, these are all your input details. So now all I'm expecting here is I have my inputs, both the inputs are there. So now I'm just going with the desired output. My desired output is like this. I'm expecting an output which should be like this. I need to have subject ID. All right. After the subject ID, I need to have the gender. You know from where you can get the gender details. The gender details you can get it from the previous data set on the top, right? I just need the reference, inform, consent, date, time in character format. All right. So how do I get this particular one? The subject ID details are here. Correct. Take this here. All of them should be as per the standard. The standard is going to be either it can be male or female. All right. The most important thing here, it should be of a timestamp, correct? Because I'm looking at the timestamp details. So how can I write the timestamp details now? Can I say, maybe I can copy this. It won't be helpful because I don't have, I can do like this. So this can be male. This also can be male. The remaining 5 are going to be female, right? This is how it is. All right. Yeah, male, male, female, 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 right? So now between these two, I have to build up the timestamp as per the SDTM protocol. What is going to be as per the SDTM protocol? I just need to replace this with T. And then I need to ensure that between this, there's supposed to be a, oh, again, I need to change it anyways. Since it's April 04 followed by 09, correct? 2020 0409. Then it is going to be September, right? 12th September. Since it's going to be a 12th September 0912. It is 11th August. That is 811. It is going to be 0811. It is going to be 13th July 730. 0730. This is going to be 6th June 616. 0660. Right? So this is my expected output. So the first one is going to be the input data that I have provided to you. The second one is going to be the expected output. So all of you has to try this. All of you has to provide me the details because You have already completed this particular one. Mean to say that these details are already aware to every one of you. Correct? So it shouldn't take time for you guys. All right. Now the next one. So we have already talked about some of the dates. Let's proceed with the remaining dates now. RF SDTC. What you can talk about it? RF stands for reference. STD stands for your subject. Correct? You can call it as start. Correct? So I can call it as subject reference subject, or first I'll go with this subject reference start date and time in character. So what exactly the meaning is? Simply treatment start date, when the treatment is started, treatment start date and time in character format. Clear? All I'm trying to say here is when the treatment is started for the given subject. Simple. So RF ENDTC. So the name itself is telling to you what is RF ENDTC to you. This is, maybe I can write it in the same way how I have written on the top. Subject reference end date and time in character format, or in a generic way, if I want to talk about it, I can simply say that it is going to be your study, the complete clinical study completed date, sometimes treatment end date. Remember both of them, treatment end date in date and time in character. Don't get confused about this. This is very important. Either it can be study completed date and time in character, or it can be treatment end date or time in character. There might be a question. Only if both of them are available, which should I consider? Right? So what is the meaning of study completed date and treatment completed date? There will be multiple treatments in a study, right? Correct? I can have multiple treatments in a study. So there are cases where there is only one treatment. Let's say parallel study. So what do you have in a parallel study? The same treatment will be continued to the subject till the end of the study. If you go to the last stage of your study, that is your treatment end date, that is also going to be your subject end date. That is just study end date. When both of them are available, very important thing, remember. When both treatment end date and study end date are available, okay, Sanji is saying that whichever is going to be the last date of treatment, that need to be considered, right? So in general, when I have a study end date and treatment end date, both of them are available, priority should be given to study end date. Remember that. This is generic also. Maybe Sanji, whatever you are saying is also the same, because when I have both treatment end date, study end date, always after the treatment only you are going to complete your study. Correct? So obviously study end date is going to be the last date that is available. If you want to talk it in a generic way, yes, you can consider that. Correct? If you are not considering, if you say that, maybe I can give you the example here that will give you. The flow is there, right? What is the flow? First, ICF will be signed. After that, you will go with the screening. After completion of the screening, you will go with the randomization. After you go with the randomization, you will enter into the treatment. Once all the treatments are done, then you also do the follow-up. Once the follow-up is also done, all the scheduled visits also done, then you say that it is going to be an end of the, yes, treatment could be ended if the subject drops out. Yes, true. So this is going to be the total screening, right? Total process, right? Correct. So what I can say now, if this is going to be the total process, in this process, treatment is ended here, but you also do some of the follow-ups. After your treatment, you do one follow-up just to see are there any side effects or not, right? After that, you can confirm that yes, the drug is working fine. So the treatment ended by last week. Let's say that somewhere in 4th of your April, you went for a follow-up. You schedule a follow-up of 1 week. That is yesterday. Yesterday you had a follow-up and mentioned that everything is going good. So your treatment, the study end date is going to be 11th April. 4th April was the date the treatment completed. 11th April was the date where your follow-up also completed. So the study is completed on 11th April. So both the dates are available. So you need to consider your 11th April is going to be your final date. That is going to be your RFND. Am I clear, everyone? Why we are going with that? All right. Maybe I can put this one here. Maybe I can just put this here for reference. Any questions here? All of you are able to follow? Right. Okay. This particular date, remember another thing, most important things that you need to remember here. Number one is this. There is another thing also you need to remember here. That is, these values can be null, right? Right? This is nothing but both your reference start date and reference end date can be null for whom? Can be null for unassigned subjects. So Mohanty, this is what you talked about. So what do you mean by unassigned subject? Unassigned subjects are the one, in a simple way, the volunteers are the subjects the total subjects dropped, or I can say that, okay, subjects who got failed in the screen. That is one case. The other case will be even after the screening, right? The people who are dropped, voluntarily or intentionally, both of them will be called it as unassigned subject. For unassigned subjects, the treatment will not start. The treatment will not end. Am I correct or not? So let's say that this is going to be the drug that we want to test it for the diabetes, right? Not for the diabetes. Let's say there's some retina therapy. We don't want the people who are already suffering with the diabetes. So in the screening, we have identified that 2 subjects are already suffering from the diabetes. So we will not consider them, but we have recorded the subject ID, we have recorded their sex and other details, but for them, my RFID is going to be null. And for my RFID, start date, end date, both of them are going to be null. Let's say there are no health issues for one subject, but once he cleared the screening, before the treatment start, he or she decided not to go with the treatment, right? In that case, what we need to do? Again, since they are not taking the treatment at all, the start date and date will not be available for them. There is nothing but they are dropped. Am I correct? All of you, any questions? Guys, I believe it must be interesting, not like the previous classes, because we are going with both the theory as well as the practicals together. So it must be interesting. If any of you are not able to follow, if any of you are feeling difficulty, let me know. Now time for assignment number 3. Wonderful. Because once you have the base ready, right, then we can go ahead with all this. That's the reason first I tried to complete all the base. Now I'm just going ahead with the complete theory plus practical together. All right. So let's go with one more task, and we can wind up the session for today. All right. So last one. Just give me a second. I'm trying to let me take the patient details from here. So here are my patient details, and then just a second. And the second thing is going to be, here are my treatment details. Right, here are my treatment details. Under the treatment details, what I have is, let's have them if we don't want gender, let's say we have treatment detail, and finally we have something called start date. Or I can say that simply date, treatment date. I can call it as a treatment date. All right. Now, the treatment is going to be something called placebo. Then, give me a second. So I'm too addicted to Notepad++, so feeling difficult. Okay, here it is going to be drug B. So when it comes to 10,333, there is only placebo given. When it comes to 444, we have given drug A, then drug B, then finally we go with placebo. All right, and 105, drug A, drug A. That's it. Now let's go with the treatment dates. So when I go with the treatment days for 111, so somewhere here we started, right? So 2020-04, that is 2020, April 2020 we have started, correct? Let's use the same just to have a consistency. 9th April 2020 we started with the placebo. That is the first stage. That is again on 11th May, again on 17th July. So we have started the placebo treatment for him somewhere in the month of April, and we have completed it by 17th July. Throughout we have given only the placebo to this particular person. Then for 222, we started by 12th April, then it ended by 10th October. And then for 333, it is 2020 August 2020, correct? Let's have it. And for 444, it is started somewhere in 13th July 2020. So 13th July 2020, we have started the treatment. It continued by till 31st July and then finally ended on 7th September. And again for 555, it is 16th June 2020 and it is 15th September 2020. These are the treatment dates, various treatments that we are given at the different stages of the, let's say that the patient 111, the subject 111 is in 3 stages, that is 3 elements in your clinical trial. So here are 2 elements in the clinical trial, only 1 element, 3 elements and 2 elements, right? This is the data. Now what I am expecting here is, I'm expecting the output like this. That is my desired output. All I'm expecting in the desired output is, let me take this completely, then I can modify as per my wish. The first one, this is going to be subject ID, correct? And the second one is going to be, I don't want this placebo and all, I just need the sex details of this particular person. I'll enter this later. Then I have something called my reference start date and time. I need a reference end date and time. All right. Now there are going to be the changes. So 111 M. So for this particular guy, this is the start date, correct? So the start date is going to be 2020 4th 0409, correct? For the same one, can I say this is going to be the end date? There's a question. Why 444 is getting different treatments? This is a crossover study. This is a crossover study, right? Crossover study doesn't need to be all the time. All the patients will be having the same different drugs. It can be few patients I continue to give the same drug also with the different dosages, right? So here for the first guy, I have given the placebo. Placebo is a fake pill, no issues at all. For 222, I have given 2 different drugs, but with 2 different dosages. No, that's what I'm saying. Same drug with different dosages. I haven't talked about the dosage details here. Correct, Mohanty? Are you getting what I'm trying to say? Right. So now when it comes to this one, it is going to be 2020 17th July. This is going to be the treatment start date and end date. So can I say that for 111, this is going to be my start date and end date. So 222 is also going to be a male. It is going to be 2020 September. That is 09 is the September and 12th. It started by 12th. Now, what is going to be the end date for this particular person? 2020. October is 10th, followed by 10th. Correct? 333. She's a female. The treatment started on 2020 08/11, right? 11th August. And the treatment end date, there is no treatment end date. I can say that. Can I say that there is no treatment end date for 103? So what can I understand here? The same is going to be considered as an end date also. Only one treatment, correct? So the same day is going to be the end day as well. So when it comes to this, that is your 444, again female, 7/13, it is going to be 07/13, It is here 2020, September is 09 and 07, correct? So guys, you need to understand what I'm trying to do here. It is not, I can straight away give you the output. Instead, the reason I'm explaining this will be, you can understand the logic. That's the reason I'm explaining it, nothing more than that. Otherwise, simply I can say, guys, this is going to be the output that I need. This is going to be here, September 0915. So can all of you understand what I'm expecting here? All I'm expecting here is going to be, I need to, when the subject is undergoing the different treatments on the different days, the moment the treatment start is going to be a treatment start date, and when the treatment is completed is going to be a treatment end date. And I need both treatment start date and end date in a proper format. What is that proper format? Yeah, IS 8601 DA, 8601 DA, not DT, DA, because we don't have the timestamp, we only have the date here. Can you guys do this? I'm going to share the notes right away. Right after this, I'm share it with the Bolisa, and Bolisa will share it with you. Otherwise, if all of you are in this WhatsApp group, I am part of one of the WhatsApp group. So if you are part of that group, I immediately share it in that particular group as well. All right. But on a lighter note, what I can do is immediately I'll share it with Bolisa. Bolisa will share it with you right away. Ask him to share it right after this class. So now the question is going to be, so there are 3 tasks that I have given to all of you. So now the question is, you want to have, can you complete this by tomorrow, and can we continue in the tomorrow's class, or you want to have the class by day after? You take your time, you complete this, and day after tomorrow we can connect again for the next topic, or we continue the class by tomorrow. It's up to you. So if you tell me, based on that I can get the confirmation. So can we continue tomorrow, or can we meet day after? Everyone, I need your inputs. So sure, Mohanty, I'll share it, but I just need the confirmation. When do you need it? Tomorrow or day after tomorrow, the next class? Day after, 2 people so far. How about others? All right.
Here I have one doubt.
Yes, please.
This subject ID 111, we have given only placebo. So here we are desired output. We are mentioning reference start date and reference end date. It is not applicable actually.
True. If you go with, to say, strictly as per the clinical trials, I can say that the same placebo I have given 3 times, because the subject doesn't know, because let's say that it is a double-blinded one. Patient doesn't know what exactly the treatment that he is providing with, we are giving with, correct? He don't know that the placebo we are giving to him. So when the patient is not aware of it, it is applicable. If it is a single blinded, or if it is a no blinded one, as you said, it is not correct. So there is a small clause here. The clause is going to be what blinded it is. If it is going to be a double-blinded one, it can be applicable. I can give the same placebo to the control group anytime I want. Nothing wrong in that. Am I correct? I don't know who asked this particular question, but will it be correct or not? But if it is an unblinded one, yes, you are correct. Questions?
Okay.
So don't go too much into the CDISC. So try to go with SAS now. So you understand the concept, right? So this is about creating your output. So suppose if you feel like, Murli, I'm not comfortable with placebo here, all I'm going to do is I just change it to drug A. So all these I'll change it to drug A, so that no issues at all. Or since it is going to be what to say, crossover study, I'll just say drug A, drug B, and drug B. Okay, with the different doses in the drug B, so that no issues at all. Because anyway, I'm not having that in the output. All I'm concentrating on, like, what is going to be my treatment start date and end date. So it is not going to be a big problem. Just to make sure that everything is going to be in line, I just go with like this, so that no issues now. All right, that's all for today, guys. So let's connect on day after tomorrow, except Raja. Everyone is asking for day after tomorrow, so let's connect day after tomorrow on it. All right, thanks everyone, take care, bye.
Can I have a quick confirmation, guys? Can someone please confirm? Are you able to hear me? Wonderful. Wonderful. Yeah. So in the last session, we started about with our special purpose domains. We started with demographics as the first domain, and we understand that the demographic domain is the one which purely talks about the subject, or I can say that it will give more details about the subject. And first, initially we started with what is the topic variable, because topic variable is the one which will give you the complete information about, or it is going to focus about the information that is provided by the domain, which is nothing but the subject ID. We have identifier variables which will identify what is this particular domain is all about. So we have three identifiers here. What are them? Study ID, that is study identifier, domain. Another one is unique subject identifier. In addition to that, we also have some other basic variables or basic columns that we are already aware of, which are nothing but the gender, age, DOB. These are nothing but, these are the variables which we generally collect from your subject itself. We can also call it as record qualifiers, correct? Record qualifiers or collecting variables are the ones we can call it as gender, age, and DOB. And generally we try to understand what are the various dates which are part of this one. We can say that the moment the subject signed the informed consent form, we can call it as a ICF signed date. When the treatment starts, when the treatment ends, if something goes wrong, what is the death date? When the information is collected, that is demographic information collected date. So these are the various dates we basically aware of, correct? So now what we did is we try to write all the variables which are part of this particular domain. So those are study identifiers, domain, unique subject identifier, subject ID, which is investigator ID, investigator name. The second one is going to be a reference informed consent form, date and time in character format. Then we have few more dates here. We will discuss one after the other. Something related to arm, something related to the death and birth, and site, hospital or the clinic. Basic demographic information, age in units, race, country, and ethnic. And when this particular details are collected. These are the various variables which are part of your domain. We haven't discussed all of them. We discussed one by one. We started with the initial ones and slowly progressing. So as part of it, first we try to discuss about the subject. The subject is nothing but the structure. The structure of your domain is only one record for each of the subject. Then under the identification variables, as we mentioned before, we started with study ID, domain, and unique subject ID. Out of all these three, your unique subject identifier is a derived variable, which is a combination of study identifier, site ID, and subject ID. I have given you one assignment here. I hope all of you might have done. I'm not sure of that. So far, I have received only one email, that too from the Mohanty, that too only for the first task. I have checked around 1 hour back. After maybe in the last 1 hour, if someone might have send it to me, I'm not sure, right? But as per my understanding, the last email that I have received from the from Mohanty, that too, that I have received on some time back today. So I'm not sure of others. Have you done it or you haven't completed? I'm not sure. Right? Guys, until and unless you work on them, you don't get a clarity. Why I'm telling this? These tasks together is nothing but our project. Right? When you go with the project, so you're going to do two different projects. There, okay, Ankita, you don't have my email ID to whom you can send. Sure. Let me share a common email ID, right? So you all can, get you all can share your details there. All right. So just let me do that to all of you. You can send it here. Murali at, or maybe I can do GMK 12816 at gmail. Just a second. So everyone, you can send your email IDs or you can send your details to this particular one so that, yeah, we can go ahead with that. Okay. Whoever is completed, please share it. Anyways, we are going to discuss these, whatever the task that I have given. I will explain all of them, but I would like to know, are you able to do this or not, or how you are progressing, what methods you are following, if anything, if any improvements are required, I can suggest. That is the reason I'm asking. All right. Investigator ID, identifier, which is nothing but either a doctor ID, clinician ID, or nurse ID can be called it as your thing. Investigator name. So we started with the date. The very first date is going to be when you have signed the consent form, or when the subject has signed the consent form. Remember, it is always RF stands for your reference, IC stands for your informed consent date, D stands for date, P stands for time, C stands for character format. Because the data type is character. We generally follow IS 8601. TA stands for only the date, DT stands for date and time. So here on these, I have given another assignment. I request you to complete it. Then RF SDTM, this is nothing but a subject reference start date. We say when the treatment actually started. So RF NTC is nothing but when the study is completed or when the treatment is ended. So there are two things. There are multiple treatments which are part of your study. When you have treatment ended, also you have your study ended. When both of them are available, you need to consider the treatment end date will be the date you need to consider for RF ENDTC. For few of the subjects, that is for unassigned subject, these values can be known. Who are these unassigned subjects? The people who are failed in the screening are maybe another group is the people who clear the screening but has not participated in any of the elements. Means they are not part of the evaluation. So those will be considered as unassigned subject. For unassigned subject, start date and end date, both of them will be null only. So I have given one more example here and I have asked all of you to work on this. So these are the various things we have discussed so far. Now the question is, any questions here? All of you are able to follow or any doubts? Can I have a quick confirmation from everyone? Understandable. Do you have any questions? If you have, what are those questions? Guys, if you are calm and if you are normal, I couldn't understand it. All right. So I will be in an assumption that all of you are clear after this. So with that assumption, I'm just proceeding with the next topic. The next, the next parameter. The next parameter is going to be under the dates itself. So we were talking about the dates, right? Just a second. So we are talking about the dates. So the next date that I would like to talk here is, maybe I can pick it up from here itself. So we have completed informed consent date, and we have completed your reference treatment start date, treatment end date. So now let's take this particular one. These three we can take. First thing. So RFX STDTC. What exactly it is, right? This is nothing but, this particular one is nothing but date and time, or date or time of first study treatment. So now there is a question, what is the difference? What do you mean by this? Because you have something else as well, correct? So what exactly the meaning of this particular one? How do I know this is going to be, how it is different from the previous STDTC? Because here also I said that it is a study treatment start date, normal treatment start date. But here I'm calling it as first, it is a date or time of first study treatment. What is the difference? Or if I want to even elaborate this, I can say that study treatment start date, date and time in character format. Now the question to all of you is, what exactly it is? Can someone please tell me, or someone, if you have an idea about it, can you elaborate on this? I'm asking about a reference study instead of treatment, study treatment start date. What exactly is study treatment start date? And how it is different from your regular treatment start date? Anyone? The patient come on time treatment. No, I couldn't get it. Patient come on time treatment. Anything else? Scheduled and actual date of treatment. Which is scheduled and which is actual? Are you saying that both of them are same, scheduled and RFX? Okay. So Sanjay is saying that this is for the entire studies start date. In this way also we can say. Okay. Can I call this as the start, whatever the study is there, entire study start date. Okay. So this is also another way of telling it. But what is the, how it is different from the other one? Any examples?
So Murli, the other one might be for that particular site. So the site, for that site, the first patient must have arrived. Sorry, the first patient treatment must have started. So that we can consider that date as RF STDTC. And whereas for RF X STDTC, it might differ. In the sense, site B might have the first treatment done for the patient instead of site A. So the date might change in that case. Is it?
Let me do that. Yeah.
Yeah.
True.
Yeah.
See that whatever you said is possible when it is a multi-center. Correct?
Yeah, correct. Yeah.
Can you tell me if it is not a multi-center?
Then it would be same. The RF STDTC and RF STDTC would be having the same dates, because if it would be the first patient's first treatment, which would be same for the study level also and for the site also.
Okay.
Is it correct? Yeah.
One way it is, the other way there is a exclusion for that as well.
Okay.
So let me.
Okay. Yeah.
For an example, it is not a multi-center. it is, let's say that it is a only one site is there. Within the same site, the treatment is started. For an example, my study, okay, my study is about, let's say, study drug. The study drug is about, let's say, Covaxin. So why we are testing Covaxin? The overall Covaxin is going to be I want to give it as a vaccination for the COVID patients, correct? Or maybe it is a vaccination. Let's not take vaccination. Maybe we can take another. Maybe let's say a simple example: fever. So I'm just developing a drug for fever. That is the overall objective. My study drug is fever. So can I say that in this particular case, I want to give my stage or my treatment should start with by providing fever drug, a fever pill to the patient, correct? That is what the actual drug is, right? I want to test this. So already there are patients who are suffering from fever. So my trial or my study starts with giving a fever related pill to the given subject. But the same subject mentioned that he or she suffering with, or she having vomiting sensation, not even vomiting sensation. What will do? Now, the first thing that we are going to do is, first we provide treatment or drug to vomiting, to control vomit. Only after that, right? Only after that we go with the study drug. Am I correct or not? Without providing the first treatment, without controlling the vomiting sensation, even if we give the study drug, if the subject vomited that particular one after having it, what is going to happen? Can I say that whether whatever the study that we would like to understand, the efficacy of the study drug that we would like to understand will not be efficient? We cannot have the proper measure of that particular one, correct? So what is the first treatment here? The first treatment we started is with the controlling this particular vomiting. Only after that we are going to provide the study drug. Am I correct? Let's say you control the vomit, right? You have given the drug on, let's say, today what's the date? 14th, correct? Let's say 10th April 2022, you've given this particular drug. And then the original study drug, the fever drug, that you have actually started on 11th April. So you have waited for 1 day. Once you got a confirmation that there is no vomiting sensation or there are no vomitings at all, only then you proceeded with the original study drug. Now you tell me which one is going to be your, can I say this is the support treatment, correct or not? The vomiting is going to be the support treatment. Whenever your study now started with a support treatment, not with a study treatment. Am I correct?
So study treatment word is important, yeah.
Correct. So your study treatment is here, but your support treatment before you go with the study treatment started on the 10th day. So this one I can call it as RF XSTDTC, whereas this one I can call it as RF STDT. Am I correct? Now is it clear between these two? All of you are able to understand? A very simple. Try to go on a lighter note. Yes or no? Everyone, any questions on this? For an example, let's say if the subject doesn't have any supporting treatment, doesn't go for any support treatment, in that case, can I say in that case, my RF-XSDTC is equal to RF-SDTC. Both of them. Everyone accept this? Can I have a confirmation on this? Right. Now, let's go for another example.
Murli, just
Yeah.
one question here. So, in this case, the supporting treatment is the concomitant medication, right?
Mhm.
So, in DM, we would be capturing the concomitant medication data also.
Yes. Yes.
Okay.
No. We don't consider the drug information and all that. That medication details we don't have there. We only have this information.
Okay.
Plus, if you look at the total number of variables which are part of your which are part of your domain, the domain details will not talk about the remaining things. It only talk about what is going to be the arm code, actual arm code. So in this only these two I'm capturing. Arm and actual arm, these two only I'm capturing. I'm not capturing any other details, correct?
Yeah, but just the date we would be capturing, right?
Correct. Date
Okay.
alone we will. That's
Okay.
what I'm
Yeah.
All right.
Understood. Yeah.
Now, perfect. Any questions from anyone else on this? Any follow-up questions on this? All right. Now let's go to the example 2. Let's say it is going to be a multi-center study. So what is multi-center study? The study is happening at the multiple places in the same nation, correct? Or it can be also a multinational study. It can be also a multinational study, correct? What is multinational study? Across the globe it is happening. In this case, in these two cases, we can go with a different example. In these two cases, we can go with a different example. That is, a subject have completed, let's say subject had a treatment in site 1, right? And the same subject going with a different treatment in site 2, considering a washout period. So what do you mean by this? I have a washout period, that is during that particular period, the drug is completely gone away from the body, but the same subject participated in the two different sites. So initially he got drug A in site 1. After the washout period, again we are providing drug B to him in the another site. So when I look at, let's say the site 1 is going to be phase 1, the site 2 is going to be phase 2. So two different phases. Can I say that when I go with the phase 2 alone, when I go with the phase 2, when he is actually taking the drug 2 at site 2 is going to be my RF SDTM, the treatment start date. But this guy, the same guy, during the phase 1 of the clinical study, he have actually taken the drug A. That happened at this site number 1. That will happen. That is going to be considered as my RFX SDTM. Am I correct? Are you able to understand this? That all of you guys can differentiate these two? Can I have a quick confirmation? All right. Perfect. Now let's go with another next parameter. The name itself is telling you here. What is that? RFXENDTC. Simple. This is study treatment start date and time. I can simply call it as study treatment end date and time. Correct? It is always going to be study treatment end date and time in the calendar format. Questions? Any questions on this? So can I give one common example for both of this? Right? Consider both of them and we'll give an example. Example for dates. I simply call it as dates. For an example, there is a patient, nothing but there is a subject. So this subject, maybe I can take, yeah, this one. So we have a subject ID. Let's say subject ID is 111. Sex is nothing but M. Let's say this particular guy. So before he started with, let's say that this is the guy who got this particular vomiting sensation. So for him, the sensation started on somewhere on 10th April. Maybe I need to follow the clinical standards. So let's go with this. 2022 March 1st. So by this time, for him, the vomiting sensation started. So we have given the vomiting to control the vomiting sensation. We have given the treatment on May 1st. But the same guy, the original study treatment started on, that is on March 2nd, the original study treatment. Let's say that the treatment is about fatigue fever. So it started on this particular day. Right. Then, what is this end date then for the same people? So we have done all your follow-ups and everything, and finally on one day, that is 2022-03, let's say we considered 14 days period to go with the complete details. After 14 days, that is 16. By 16th, we consider that that's the day we have given the final. That's the day we have given the final drug, or I can say that some final diagnosis we have done. That is the end of your treatment. Now, can I say that the same person, after this particular one, there is no other treatment. There is no other treatment pending. Can I call that this is going to be my same date as my study end date as well? Can I say this is the correct one? That is, we started the support treatment on March 1st. We have given the original study treatment on March 2nd. We waited for 14 days to see all the side effects and see whether all the things are controlled or not. On 16th, we confirmed that everything is okay. We just have given the final drug on that particular day. That is going to be the end of the treatment also. We said that there is no follow-up required after this particular one. Now, what is going to happen? Can I say that this is going to be my final treatment as well? This is going to be the end of the study treatment. This is also going to be the end of your treatment, final treatment as well. Correct or not? Right? Now, okay, all of you are clear with this example? Is it giving you the right answer? Any questions on this? All right. Let me go with the next one.
Murli, just one question here. So in case if we have that patient dropout, subject dropout, so in that case, the RF ENDTC for this subject would be blank, right? And RF X ENDTC might be for the for that site which closes last.
No. Let's say that. Okay, give me a second.
Yeah.
Raja, I think I'm clear enough. Guys, is my voice, all of you, is it comfortable still, or are you feeling that my voice is low? Okay, if any of you feeling that my voice is okay. Now, Mohanty, you need the different dates example. I'll give you that as well. All right. For an example, this is one for fever. Let's say there is another subject. 222. For this guy, maybe I can go with a different example. There is another subject 222. Let's say this, but for this particular person, let's say female. For this particular lady, we are going with the orthonet treatment. Mean to say that she is having some problem with the knees. That is, let's say that the knee problem is there. So the doctor diagnosed with that particular one and understand that. So we want to start with, let's say that initially the doctor mentioned that let's go with some of the tablets which will improve your whatever the gel in your kneecap. So with the increase in the gel, the friction at your bones will be less. Because of that, you don't feel much of the pain. That is the initial understanding and the initial diagnosis is provided by the doctor. So let's say on the same March 1st, the doctor actually given the, so I'm just talking about the orthonet. So problem is burning knee. So the first on the RF SDTM here, that is nothing but on 2022-0301, the doctor mentioned that why can't we go with injecting gel into the kneecap. So this gel is going to help you to go with the to reduce the friction, which will give you some relief. So the patient has waited for, let's say, 10 days or 1 week. On 10th, the patient revisited the doctor and mentioned that, hey, whatever it happened, it has reduced a bit, or it has given me bit of the relaxation, but still the problem exists. I'm still feeling the pain in the knee. So then the doctor decided that it won't happen. So why can't we go with the other option is, let's say we will go with the knee replacement. That's the only alternative because it is not going to happen. So knee replacement. So when the knee replacement happened, the knee replacement happened on the 10th. So doctor mentioned that after the knee replacement, there won't be any major things that you need to take care, but all you need to do is, let's say, before I discharge you, let's say that after a week we are discharging you. So at the time of discharge, so for the knee replacement, right, the time of discharge, I can say that whatever we are going to do, some basic medicines, antibiotics, or other some healing things we are going to do. Healing medicines. I'm giving you on a hypothetical notice, guys. It may not be a relevant example, but I'm trying to give you my best. All right. Now, after that, doctor mentioned that you need to have a follow-up. Right? I need to check your knee. Right? I need to check your knee, and I need to ensure that everything is proper. So he requested the patient to come after 15 days. That is after 10 days. So here, sorry, 14 days. So here what he's trying to do is he checked the knee again, and he understands that healing is good, but there are other side effects. Let's say that some issues the patient is having. For that, he has given some supporting medicine. So now the treatment, the original treatment starts here, right? The study treatment started here, and the study's treatment ended at the 16th itself. But the original, the complete treatment is actually ended on 30th of the March. Mohanty, is this understandable? There are cases where we are going to have the different dates as well, right? May not be the right example for this particular one, but I'm trying to help you with the more or less relatable things. Any questions on that?
Murali, you see the end, RFX and DTC, the variable is different.
What do you mean by that?
Okay, you read out the variable subject, sex. No, you're same data. Sex, RFX SDTC, RFSDTC, and then RFX It should be E and
END. Okay, got it. I got it. This is
so. So my question was RFX NDTC and RF and DTC when they will be different. You got it?
This is
maybe
every time
yeah. So that's okay. Yeah, I got it.
Perfect. Any questions from anyone else? All right.
So Murli, in this case, if we take the example 2, in this case, if the subject doesn't come on if we
After the
say
follow-up, let's say 16.
before,
After the subject haven't come, right? He haven't visited.
Yeah.
That is going to be your pending.
Acha.
That is
Okay.
participation date.
Okay. So for that particular subject, the data entry would not be there for these variables, right? The date would be populated for RFP, EN, DTC, correct?
Exactly. The ENDTC signal
would be blank, huh? Yeah, understood. Yeah. Thank you.
Now let me explain this. So we are talking about this. This is nothing but reference and participation date and time in character. There are multiple scenarios for this. Particular one. So what are they? Number one, what are these exactly? It can be a study completion date. Correct? If everything goes well, the end participation date is nothing but the study completion date. Correct? That is the time for the last time the subject is participated in the study. If not, it is going to be my final follow-up date. After follow-up, he or she haven't come. Or it can be a death date. So let's say that there are two more phases in that particular treatment. Let's say once we started the study treatment, before we go with the original treatment itself, the original treatment itself, the patient is no more. So that is going to become my end participation date. Or it can be the discontinued date. So this is what you asked me, Sanchi.
Yes.
Discontinued date. Correct?
Yeah.
In all these cases, I can say that this is going to be my discontinued date. So let's say that in the example that I have just talked about now, let's say take the first one. So in this example, what is going to happen? Subject is there, start date, end date, both of them. Let's say we started with the vomiting sensation, the treatment we provided, original treatment has started, then it is going to be treatment end date, subject treatment end date. Let's say everything is done. This is the last time. After that, the subject need not to visit the hospital anymore. Can I say that in this particular case, this is going to be my participation end date as well? Correct? My participation end date, my reference, that is my treatment end date, as well as my study treatment end date. All these three are going to be the same. Possible? Take this particular example now. So in this case, the study treatments supporting treatment start here, the original treatment started here, it ended here. After the follow-up, the patient is not visited. None. Correct? So there is no reference NDTC. So what is going to happen? Let's say this is going to be the time where we want to do the follow-up. Correct? For the follow-up, the patient haven't visited. Right? The follow-up is actually on the last thing happened on the 16th. After 16th, this patient hasn't visited. So can I say that this is going to be the day where the patient last participated in the study? So can I say that this is going to be my participation end date as well? Right? When was the last time the patient or the subject has done this particular study? Right? That is equivalent to my patient participation end date as well. All of you are clear up to this, the dates part, right? So all these are scenario based. We go as per the scenarios. Questions?
Murli, in this case, if the patient has not come after the RF STDTC, then in this case, RF X ENDTC, everything will be blank, right? And we would populate P.
Yes. STDTC will be blank, XE and DTC will be blank, and RF and DTC is also blank. But the last time he participated was on the STDTC. So it is going to be my STDTC is equivalent to your participation end date as well.
Yeah. Okay. Yes. Thank you.
All right. Let's see where we are now. So let me talk about this, and then we'll go with the ARM code. Okay. So let's say we are talking about death date. The first one. The name itself is telling it is going to be death, date, and time in character format. Correct? If any subject, this is going to be subject, death, date, and time in character format. Correct? That can be on any day, that can happen in any phase of the study. Correct? Now, where it will be? Meaning, we need to have little understanding about some note I need to tell you here. Let's say we are part of this disposition domain. Let's say domain is equal to disposition. Correct? In the disposition domain, right? If we have for disposition domain, we have DS term, right? What is the DS term? Nothing but the focus, which is nothing but the topic variable, right? So if we are going to have your DS term is equal to death, if it is equal to death, in that particular case, my DSSDTTC, that is nothing but your start, date, and time, your death, start, date, and time in character format. This will be considered as your death date. If you are talking about the DS domain. Suppose if you are talking about your adverse events, because the death is also an event, right? So in this particular case, if AE out, there is a variable called AE out, and if that AE out variable is equal to death, in that particular case, adverse event end timestamp, end DTC, this is supposed to be considered as your death. These two you need to remember. These two you need to remember. What are them? As long as the name itself is telling you death, date, and time in character format, the note you need to understand is if it is a disposition domain, we'll talk about that again anyway. If the DS term is equal to death, then DS STDTC is going to be a death date. If the domain information that we have provided is of the adverse events domain, then if AE out is equal to death, then only in that particular case, AENTC, that is going to be your death date variable. You need to remember these two, and these are going to have a lot of impact while if something happened to the subject. So you need to remember this particular one. All right. So then DTHFL, which is nothing but death flag. Simple, death flag. The death flag can be, it can have only two values. The two values can be either yes or no. Right? So what is the meaning of it? How can I go with this? If the patient is no more, if the patient has the death value, sorry, death date, I can say that it is going to be a death flag equal to yes. If not, death flag is equal to no. Okay. So let's have a simple task on this particular one, and then we can proceed with the next exam. Let's say here assignment number 4. So the kind of assignment that we would like to go here is a very simple one. A very simple one. All I'm going to have is going to be, I just have patient details as well as your name, because I told you there are 2 domains information, correct? The domain information that I have given here is going to be two domains. I told you one is going to be DS domain, another one is going to be demographic domain. Let's say from demographics, your input data, the input data, that too I'm getting from the demographics domain. So from DM domain I'm getting it. So from DM domain, what and all I have, I'm going to have the patient information and the gender information. Let's say take this. So I have these 2. All right. And then I have one more domain, which is nothing but my disposition domain. Let's say DS. So from this particular one, I have 3 information again. So I have, let's say patient ID is there. Here instead of the gender, I have term, because it's all about the term there. And again, I have the third one, which is nothing but STD. Okay, 3 information is there. So under this particular one, for the same patient, let's say it is going to be death, and the date here is going to be 2022-03-11. For the other one, the study completed. Just nothing happened, the study got completed. Again, for the next patient also study completed. Then the other patient, it is death, and for final patient it is study. So I need to have the different dates. The different dates are going to be like this. Let's say study completed on today. Let's say that 14th April. The same study completed today. Study completed today. But this guy, something went wrong after some phase. Let's say 31st. He's now what happened? I have, what are the details that I have here? I have patient details. At the same time, I have their term details, different term details. Correct? Now, all I need is my desired output is here. So my desired output is going to be, I just need information about subject ID. I need information about the sex, and I also need the information about the other things, which is nothing but death date, DTH, DTC, and also we need death flag, DTH. Yes. Can you tell me what are the details I'm going to get here? 111. So for this particular guy, it is M. Then the death date and time is going to be this. It is in the same format. And finally, I'm going to have that death flag. What is going to be the death flag? Yes. 222. Study completed. I don't have any death date. So what is going to be the flag for me? Either I can have Y or I can have it as N. Correct? So there is no death flag for this particular guy, N. Then 333. 333, it is going to be female. Study completed. No death happened. 444. Yes. Death happened on this day. Yes. 555. Female. No death date. The death flag is going to be yes or no? Or if you don't want, you can also remove this. outputs. Just make sure that the expected output is going to be, if it is yes, just keep it yes, otherwise just leave it. All of you are clear?
Yes. Yes, ma'am.
All right. Maybe here itself, let me complete. Okay, maybe I'll go with this. Guys, please complete your assignments. Don't neglect that particular one. That's my request. Okay. All right. Then just birth date and time, nothing but date of birth, subject date of birth, subject date of birth in character form. I don't need to explain anything here, correct? So I'm not going in detail. So the next thing is going to be, we are going to talk about age and age U. So age. So the name itself, it is pretty much straightforward. That is subject age. And remember, there is going to be a age calculation as well, right? We'll talk about this later, but I'm going to give you the calculation alone. So subject is generally calculated as, mean how much time you have spent in the study. That is the meaning. How much time you have spent in the study. That also will be considered, because okay, let me tell you how it will be calculated. For an example, this study is have a span of 2 years. Let's say somewhere we started in the 2020 and we expected to complete this particular study by 2022 June. That is 2 more months is there. Now, by the time the subject entered into the study, the subject age was 20 years. Now it must be 22, correct? The subject age is always, as the number of days progressing, the age also will increase. Now, can I consider my subject age as 20 or 22? By the time he entered into the study, it is 20. Now it is 22. The study is still happening. So what is the age of this particular subject?
22.
22. Any other? Anyone? (22). Mahipal also saying 22. Guys, give a shot. Nothing wrong, even if you say wrong or wrong answer, right? So don't hesitate. Please tell me. Okay. See, as the span increasing, the age also will change. So always when someone asks you, hey, at what time the study started, that is going to be my age. So generally, if I want to calculate the age of the person, generally how we are going to calculate, let's say age is equal to today minus my date of birth divided by number of years, 365 days. Am I correct? Of course, leap years and all that, but in general, it is going to be 365 years. Is this how you are going to calculate your age? All of us. Now, since the today is always changing, right? For tomorrow, if I do the calculation today and if I do the calculation after 1 month or 2 months, all the time, this factor will be keep on changing. So generally, what we do is the time when the consent was signed, the consent form was signed, that is RF ICSD, that is the time when the reference informed consent form signed by your subject, that is going to be my day minus the date of birth. Just now you calculated, the variable representing that is going to be date of birth divided by, instead of going with 365, we will go with 365.25 by considering the leap years and everything. This is going to be your age calculation. All of you remember this? Age calculation is simple and straightforward. It's supposed to be your whatever the day you want to calculate the age, the day minus your date of birth divided by 365. But as the span increasing, there is always your age will be changing. To make it as a constant, the time you sign your informed consent form, that is going to be your day for your age calculation. To avoid all your leap years and everything, we generally consider it to be 365.25. Any questions on this? Any questions? All right. This is self-explanatory. What is that? Age in units. The units. Yeah.
Murli, just one question here. So when we analyze the study at the end, that time the age will differ.
Correct. But
Okay.
when the treatment has started, the initial
Sorry?
treatment, when the
Mm-hmm.
initial treatment is started, let's say I want to have a study on the people, right? Let's say, for an example, maybe not the right, but maybe I can go with this. Let's say there is a analysis happening on AIDS, HIV. So we have considered a volunteer who entered into this particular study somewhere in 2014. So still the study is happening because we don't have a drug for HIV yet, correct? So from how many years he is participated in, have been participating in the study? Can I say for past 8 years? By the time he entered, right, his age was 30. Now he is into 40, the 38. So can I consider his age as 38 or can I consider his age as 30? Because we have started providing the drug to him at the age of 30. That is when it started, correct? Suppose if I try to give the same drug to the patient who is of 38 years of age, by considering the current day is 38, considering that if I give it, the drug may not be efficient because I have been giving this particular drug to this particular patient from past 8 years. So over the 8 years period, it is giving me the result. Suppose if I want to give the same drug to the person who is into the early 40s, the drug composition should be different. The dosage should be different. I cannot give the same dosage. If I give the same dosage, the efficiency will not be the same. Am I correct? So
Correct,
always
Murli.
consider. Yeah.
But then when we are starting the study, we have the inclusion and exclusion criteria. So
Correct.
in inclusion criteria, if we consider the same example that the subject is 30 years old, and that's why we have enrolled that subject in the trial. So as the trial progresses, he is reaching 40. So in that case, wouldn't the analysis or the final report be incorrect in the sense the include it won't be passing the inclusion criteria, right? Or there must be other parameters which we would be taking into consideration during that analysis?
There are a lot of parameters you are going to consider because if it's a treatment drug, if it is a treatment drug, the example that I have given will not be the correct one, correct?
Yeah.
Because how many diseases you have with such long impact or with such long span? Can I say very less?
Oncology, yeah.
Very less, to be honest, correct? So for those, we always have this inclusion and exclusion criteria. If the majority of the people are falling under this, all right, majority of the people are falling under this, we still consider it as a correct one. Let's say only 5 or 6 people who are taking this particular drug for all these years, there is no death happened and there is no improvement in their health condition also. Both of them are there. Can I consider them as outliers and I can remove from my study itself? I don't consider them as part of my study anymore. I'll consider them as outliers because there is no improvement in the health, or death happened, or in the worst case, there's nothing happened to him. So my drug is not efficient. Even the death happened, the drug is not efficient. There is no improvement in the health, there is no efficiency. In both the cases, my drug is not working on this particular fellow, even though he is there in the study for 8 years. So let's exclude him. Don't consider him. Consider him as an outlier. Right? It is not one example. With that one example, we cannot conclude, but all I'm trying to say here is going to be, it is a standard. The standard is going to be, when you want to consider the age of the person, the moment the subject signed your informed consent form, at that particular day, whatever is the age of the person, that is going to be the final age. We don't consider the span. Clear?
Yes, Madam.
Yeah. There is another reason for it. There is another reason for why we want to do that. 90% of the cases, except the ones where we don't have any medicine, clinical trials won't last for more than a year. Am I correct? Until unless it is critical. Most of the clinical trials will be, the span will be not go more than a year. So when it is not happening more than a year, even if you consider your informed consent date, or even if you consider the date on time by the time you are collecting all your measurements and all, like your final study and all, it won't make an impact. It won't make much difference. But for the standardization, we consider informed consent date. Okay, there is another reason for it. Questions? All right, next thing. So here we are again, then we consider the simple parameters. The simple parameter is going to be country. There is nothing we are going to talk about this particular one, but only thing that we are going to talk here is going to be the country code. Country we always represent with country code. That is represents with country code. Okay. Then what? So you are representing with country code, but what it is? It should be 3 characters long. Meaning, example, if you are representing India, IND. If you are representing USA, it is going to be USA, United States of America. Always 3 characters. Right? There is nothing much to explain. I'm simply going with the other one. Race. Simply speaking, color. Simply speaking, color. Or you can also say that continental information as well. Which continent he or she belongs to. That is also will be considered as your race. For an example, in general, black, white, Asian, all of us, correct? Then most of us, sorry, all of us, not all of us, most of us. Sometimes it can be other as well. What do you mean by other? Who and all falls under other. So this example for others is going to be, sometimes the person is not a black or white or is not belongs to the Asian continent, but other continents are there. European continent, but in that again we want to go with some. For an example, Italian. That don't consider it as a language, right? Brazilian, Canadian, like this. You can also represent the race as based on the country information. The another thing you need to understand here is going to be, sometimes whatever important note that I'm mentioning here, guys, the meaning is there is a high possibility that you can expect this as part of your interview, right? So if anything other related race, we will pass those values to the supplement domain, right? If race is other, we will be passing this information to the supplement domain as well. So what is the meaning of this? The meaning is very simple. That is, suppose maybe we can simply put like this. Under the others, right? If the race is other, we can simply call it as, this will be, this information we also can pass like this, SUPPTM. That is, get this information from the supplementary domain. All right. For an example, in the demographic, if it is going to be, again, if it is going to be a demographic domain, then we pass this as SUPPTM. If it is going to be adverse events, if it is going to be adverse events, in that particular case, you will call it as SUPPAE. That is, instead of others, we can pass this information as a supplementary domain. Clear? So you can understand this particular one once we go to the respective domain. Under the supplementary domain, again, I'm going to explain this particular information. All right. So we talked about country and race. Let me put this at the top so it will be easy. Age is over. Okay, let me take all this together. Age, age units, race, country. Now you need to go with ethnic. All right. So what is ethnic? Anyone? What do you represent under ethnic? Can I say it's simply going to be my ethnicity? Ethnicity of respective country. Whatever the country that we are talking about, that country's ethnicity we are going to talk about. For an example, a person, maybe I can give you a few examples here. Let's say if the person is coming from Latin America, can I say simply it is going to be call it as a Latin American? Can I do like that? So based on the country information, every country is going to have that particular one. For an example, simple. Yeah, thanks, Anji. We are in India. We all belongs to India, so we can simply call it as our ethnicity is going to be India. As simple as that. All right. So all the people who are coming from United States of America, Americans, right? A very simple and straightforward. Right. Now we'll enter into the most important thing, which is nothing but when we go with the collection. Maybe I can do this by tomorrow because there is too much of TD happening. So I don't want to go with that particular one. So let me stop here. But before I stop, let me help you with what we are going to discuss tomorrow. So DMDTC, which is going to be a date and time of collection. That is the first thing that we are going to discuss. Then we are going to discuss about DMDY, study day of collection, right? So it is going to be a derived variable. Let's discuss about this in detail by tomorrow. All right. And what else is pending? There are a few formulas will be there that we need to discuss as well. Then ARM is pending. So maybe once we complete it, there are a lot of tasks here. So then we go with ARM. Yes, perfect. So we talk about ARM, that is description of your planned ARM. We are going to talk about ARM code, actual ARM code. Correct? Anything other than this? I think if you complete these 6, 1, 2, and these 4, 6, then I can say that we are good with the 99.99% tomorrow I'll complete this. So that we will be having ample amount of the time for the next one. So once demographic domain is done, then we can go with the subject visits, then subject elements. So those are all very quick. We can simply quickly we can run. Demographics is the most important one. That's the reason we are spending our time here. So DMDTC, DMDY, there are a lot of calculations involved there. So we are going to do all those calculations. Then we go with the ARM code, actual ARM code. With that, we can say that we are good with the demographic domain. So whatever the notes, I think today's notes are also important because you have one more task to do. So I will send this particular notes right today itself. Okay, or I'll do one thing. This is the only thing that I'm expecting here, right? So assignment 4 I'm keeping here itself. All right, so you can do it. Otherwise I'll send it. Why unnecessary confusion? I'll send it. I'll send the notes, complete the assignment 4 as well. So by tomorrow at least can I expect everyone complete all the 4 assignments? 1 2 3 4. Yes or no, guys? Can I have a confirmation from everyone? All right, perfect. Okay, tomorrow we'll meet and we'll complete the demo. Okay, take care of you guys. Take care everyone. See you tomorrow. Bye. Tomorrow class is there. Tomorrow class is there. And I'll take the class tomorrow. Okay, take care. Bye. Thank you. Okay, hello everyone. Good evening. Hope all of you are able to hear me and hope all of you can see my screen. Can I have a quick confirmation, guys? Everyone? Team, can you hear me? Okay, perfect. All right. Now, let me start with the remaining portion of your demographics domain. So if you remember last class when we started with this particular one, we started with the basic structure. The structure is going to be a demographic domain is the one which is going to have only one record per subject. The identifier variables are nothing but study ID, domain, and subject ID. I'm just giving a brief overview of what we are done so far. Then I have given 3 assignments to you. The first assignment is on providing the patient ID and gender. I'm expecting everyone to create unique subject identifier along with the other details as per compliance with the CDISC. Then INV ID stands for your investigator ID. INV name stands for your investigator name. RF ICDCTC, which is nothing but your RF ICDTC is nothing but the time, the reference informed consent date and time in character format. I can simply say that it is the time when the subject has signed the informed consent form. So based on the informed consent form, I have given one assignment to you. And then RF SDTTC, which is nothing but this is subject reference start date and time. That is nothing but when was the first treatment, the treatment start and time in character format. Correct? So end date is nothing but it can be either you can call it as a subject reference end date, or it can be also call it as your study completed date, right? So there is a small class here. Let's say if both study date as well as study end date as treatment end date available, the priority should be given to the study end date. And also we have given one more clause here. The clause is going to be both study reference study start date, reference study end date will be null for unassigned subjects. Unassigned subjects are nothing but either they are drop out after the screening or they got failed during the screening. So based on this, I have given one more assignment here. Then RFX SDTC. This is nothing but date and time of first study treatment. The meaning here is, right, we are intended to go with the study drug, but patient has some issues. So to overcome those issues, first, before even giving the regular study drug, we are going to give some other treatment to him. I have given the example here. The example is going to be, let's say, the drug that we would like to give is the fever drug, but the patient is facing issues with the patient having some vomiting sensation. So the first drug that we are giving here is nothing but we are giving the drug to control the vomiting. Right? So this is nothing but your supporting drug, or it is going to be a supporting treatment. So here the RFX SDTC is nothing but here when the support treatment started. RF SDTC is nothing but this is the time where your regular treatment got started. When you go with the multi-center study or multinational study, in that particular case, we are going to have site 1 and site 2. So if the same subject is participating in multiple phases of the clinical trial and he is part of the site 1 and site 2, whatever the subject had, the treatment that he or she had in the site 1 is going to become your RFX SDTC. The other one is going to be RFSTD. Correct? For this also, let's say that we have given one simple example here, and then we went with RFPENDTC, which is nothing but reference end participation date. It can be considered as a study completion date, or it can be a follow-up date, or it can be a death date, or it can be a discontinued date. What is the meaning of this? Can I say that if for some reason the subject has discontinued, right? He or she is not interested to continue with the clinical trials, then it is going to be the reference end participation date is going to be a discontinued date. For some reason patient is expired, he is no more, then the death date is going to be the end participation date. Let's say that there is no follow-up date yet, follow-up is not done yet, only all the phases of your clinical trials is completed, so then it is going to be called as a study completion date. Let's say even after the study completion date, we made one follow-up to check all his vitals are okay or not. Then the follow-up date is going to be the end participation date, right? Then DTH DTC, which is nothing but the death date. For some reason, if the subject is expired, then it is going to be the death date and time in the character format. So here we need to understand, if it is a demographic domain, the DS term is equal to death means then DSS DTC, this is going to be a death date. If it is an adverse events domain, if AE out is equal to death, then AE and TC is going to be a date, right? Based on the domain, we are going to identify this. Then death flag. Death flag is going to say that if the patient is expired, if it is yes, else it is going to be no. For this, I have given one assignment to you. Then birth date and time. So what is going to be the subject date of birth? Age, subject age. Generally, if I want to calculate this particular one, it will be calculated informed consent date, that is a reference informed consent date minus birth date divided by 365.25. So why we want to do this particular one? A trial can continue over a span of time. That can be 1 year, 2 years, or it can be any time. Correct? So irrespective of that particular one, whenever the first time whenever he or she signed the informed consent form, that date minus his birth date, which is nothing but we have collected this information, right? These are going to be the record variable. So these two divided by 365.25. Age units always in the years. Country, which represents the country code, always it is going to be 3 characters long. Race, black, white, Asian, and other. If you go with the other, it is going to be this information we are also supplying to the supplementary domain. Right? Ethnicity of the respective country, like Latin American, Indian, American, like that. Right? So I told you in the last week, these are going to be the pending topics that we would like to cover in the demographics domain. What are them? We need to talk about what is called DMDTC, DMDY, arm code, actual arm code. So if we complete these 6 variables, then we can say that we are good with the demographics domain. Right? So before I start with the today's session, if any of you have any questions, kindly let me know. If not, we will proceed with the today's class. Any questions from anyone? I could see there is a good improvement in the assignments. I have been getting some of the mails from some of you so far, but still the number is less. The number is going to be very less. I can say that it is going to be only 20% or 25% so far. So I'm requesting every one of you, please complete it. I'll wait for one more day. If I haven't get any of the responses, or if I haven't get complete thing, then I will start explaining those assignments in the class itself. Questions? All right, so I will be under assumption that all of you are good with this particular one. So I'm continuing with my next topic, that is next variable, which is nothing but DMDTC. So the DMDTC is nothing but it is date and time of collection. So what do you mean by collection? So we are going to collect the information from the patient, right? From the subject, right? Or it is called it as a demographic date of collection. Now, when we will get this information, anyone? When we are going to get this demographic date of collection, or when we are going to get this collection details from the patient? Can I say this will happen, or the collection will happen? Anyone? During the screening. So enrollment, which is nothing but the screening, correct? So why enrollment happening? The enrollment is happening is for the screening purpose, correct? So at the time of screening, right, whatever the demographics that we are going to calculate, those are going to be that date is going to be my DMDTC, correct? Again, so what is the other thing that you need to understand here? The screening visit date, whatever the screening visit date, when the subject is actually visiting for the screening visit date can be considered as the collection date or as the DMDTC. Am I correct? Whatever the screening visit date, the same screening visit date can be considered as the DMDTC. Every one of you, is that correct? So if you want to go with one simple example here, the example is going to be, let's say I'm going to have the same patient details we can take here. Let's say take this particular guy. So there is a patient ID, the gender is equal to M. Yeah, there's a question. Anyone? Okay. So now let's say that this particular guy has, let's say the kind of visit. And here we are also going to have the start date. So now I can say that this particular patient has visited, let me check the date. So to see today is 18, let's say somewhere on 14th April, that is nothing but 2022 04/14. Can I say this is going to be a start date? But what is the purpose of visit? Screening. This is the time he came for the screening. So the start date is nothing but 2022 04/14. Now this can be considered as a DMDTC. So now in this example, DMDTC is equal to, I can simply consider as when he came for the visit. That visit date itself is nothing but the first time he came for the screening. So the screening date itself, I can call it as my DMDTC. And remember, here you need to understand one thing. It is not mandatory that DMDTC may or may not be same as, you have something here, right? What is that? RF ICDTC. What is this RF ICDTC? This is nothing but your informed consent date, reference informed consent date, correct? So he visited for the screening on this particular day, but let's say that he signed it on, let's say on the following day. Let's say consent signed date. So this can be the following day, 2022-04-15. In some cases, it can be the same day as well. The screening and both will be completed on the same day as well. For few, it may be on the different dates, correct? So there is no guarantee that it can be the same or it may be different. So don't get confused about this particular one. Can I have a quick confirmation? All of you are comfortable with this particular one? All I'm saying here is going to be, it may not be same that your consent signed date and your screening date cannot be the same, but at the same time there are scenarios it will be on the same day as well, right? If someone is trying to put an analogy between them, just make sure that there is no one can guarantee that it can be the same day or it can be the following day, correct? It may be same, it may not be the same, right? Few people will get confused here. That's the reason I'm stretching about this particular one. Can I get a quick confirmation? All of you are comfortable with this? Yes or no? Perfect. Any questions here? Right. So now let's proceed with DMDY. DMDY is very crucial. This will be called it as study day of collection. It is a study day of collection. Remember, most important thing, it is a derived variable. And it can be positive or negative, but never be a zero. Very important. I'll explain all of this. Okay. And also, most importantly, it is a numeric variable. Definitely, it is not a date, right? It is a study day of collection. Now, what exactly it is? I told you that it is a derived variable, right? So we need to understand the formula. So before we write the formula, we understand that derivation or input variables are there are two variables. Based on this we are going to calculate this particular one. One is your DMDTC. What is DMDTC? When the collection happened. This is the one, the date and time of collection. And the other one is going to be RF SDTC, that is your reference study treatment, reference subject start date and time in character format. Based on these two, we are going to calculate this one, correct? So we already know these two variables, correct? If you look into this one, we can clearly say here that it is going to be subject reference start date and time in character. This is nothing but when treatment has started. DMDTC is nothing but this is the time when the collection was happened, correct? So based on these two, we are going to calculate this particular one. Okay, now, so there are two calculations. Number one, let's say that my DMDY is equal to DMDTC, that is by the time your collection happened, minus your RF SDTC, the treatment start date, these two plus 1. That is DMDTC minus RF SDTC plus 1. When this is going to happen? If this is going to be the case, the meaning is This is the case when your DMDTC is greater than your RFSDTTC. That is, before treatment itself, we have collected all the information. Then this formula is hold good. Okay, the DMDTC is on. The DMDTC is on. That is, the collection happened. The collection is happened before your treatment started. Then it is correct. Second case, there is a scenario 2. The scenario 2 is going to be DMDY is equal to DMDTC minus, I'll simply say that RFSTDTC. When it is going to happen is, this will happen when your DMDTC is lesser than your RFSTDTC. That is, if your collection date is prior to the treatment date, then go with the first formula. When your collection date is lesser than, that is after your treatment start date, then you are going with the second one. Is that clear? Every one of you? Maybe I can also write like this. So here I can say that DMDTC on or before RFSTDTC. In that particular case, we go with the first one, on or before. Okay, if this is going to be like your DMDTC is after treatment start date. If the collection happened after the treatment start date, then you go with the second formula. If your collection happened before your first treatment, then you can go ahead with the first formula. Clear, everyone? Can I have a quick confirmation? Any questions on this? Let me give you an example so that you can get a clarity on it. It is just the calculation that we are going to do. Just a second. It is just the calculation that we are doing. So to make sure to calculate this particular value. So let me write a simple example so that we can all understand this particular one. Let me take this. Right. So we have subject, sex, and we have, I don't want this particular one. I need only the reference start date. I don't want this particular one. So I'm just removing this. And here reference end date also are not required to me. There is a question from Sanchi. Sanchi, let me explain that. Let me give the example first, then I can go with that. Okay. So then I also have DMDTC, and the DMDTC is 2022-03-20. And let's say that it is going to be 04, today's date is 18. And it is 041. Now what you can understand from here? Let's say that this is going to be my calculation. Let's say just go with the second patient as well. The second patient, the second subject is going to be 222 M 2022 410. It is going to be 418. So there are two people. We started with this particular one. Okay. Now can I say that DMDTC? Now your DMDTC, can I say it is greater than your RFDTC? Correct? Every one of you? Yeah, okay, let me take one more example where both the values are same. 333 F 80. Let's say both of them are same. Your DMDTC as well as your reference start date, both of them are going to be the same. Now, in this particular case, what is your DMDY value? Let's start with the last one. What is your DMDY value? It is on or before this. Your DMDTC is on or before. So what is going to happen? DMDTC minus your RFST DTTC, since both of them are same, this value becomes 0. 0 plus 1 is equal to 1. So can I say that this value is equal to 1? Consider this particular one, 10 minus 18. Am I correct? April 10th minus April 18th plus 1. Is that correct or not? Hey, I'm writing a slightly wrong one here. Apologies for it. There is a small confusion. I made it here. Sorry about it. It's supposed to be like this. If it is before, it is on or after. So when it is on or after, the first one still holds good. Now my DMDTC is before my RSDTC, correct? That is first the collection happened. After the collection, it happened. So can I need to say that my DMDTC minus start date, I have to consider 10 minus 18, which is going to be minus 8, correct? And here it is 1 minus 18. So what is 1 minus 18? I can say that minus 17, correct? So most of the time, so what we can understand from here, I can say that most of the times it will be negative. Most of the times the value is negative. Now let me take your question. So in the second derivation, we might get a negative study day value. Okay, actually it's a first definition in the first itself. Yeah, sorry, in the second itself. Yes, most of the times it is negative, Sanchi. Guys, all of you are able to follow this. A very simple one. All we need to do is you need to remember DMDY value is 90% of the times it is going to be negative only, because before the treatment itself we are going to take the collection, because screening is the first one. Either it will happen on the same day or it will happen on the follow-up day. Correct? Just give me a second. How about others? All of you are able to follow this particular one? Can I have a quick confirmation first? It is all about a simple calculation. All we are doing is we are calculating the study day of collection. It is an integer. It can be a positive or a negative value, but it is not going to be 0. Most of the times the value is going to be negative. All we are doing is we are considering two things: collection date and your treatment start date. 99.99% of the cases, your collection will happen before you go with the treatment. So this value will be negative. Is that clear, guys? Any questions? Right? Okay. Let's go with a simple one. Let's go with a very simple task here. The task, I don't call it as a task, but I'm just going to give you a very simple example to calculate this particular, but doesn't require, because anyways I have shown the demonstration here. The same demonstration you can use it. When based on your RFSDTC, based on your DMDTC, you are going to calculate this particular one. Okay. Now You need to understand one small thing here. That one small thing is going to be how to calculate these formulas. There are a bunch of formulas other than this, because this is not going to be end in the demographics itself. You are also going to use this in your adverse events. You are also going to use this in your tests will be there, right? Test domains. There also you are going to use it. For that reason, I want to give you a very simple example here. Let's say since it is a demographics, you are calling it as a dm dy. Okay, suppose if the same is going to be you're visiting your scheduled visit. Okay, in that particular time you are going to call this as, okay, let's say vital signs, vs dy. Suppose if it is going to be the lab test, whenever you're going with the lab test, then in that particular case, you're going to call this as lab dy, laboratory dy, correct? So it is not going to end here. It is going to be there in the other domains as well. So in all the domains, when you want to calculate this particular one, we can represent your study day. Let's consider this as xx dy. You can simply replace the first two x's with your domain name in a simple way, right? So then what is the other thing we have? We are going to have study start date, correct? Study start date. It can be your, in demographics domain, it is going to be a treatment start date. In other domains, it is slightly different, but you can simply call this as a XX SDTY. Okay, it is going to be day, just consider as a day, right? Similarly, there is going to be a study end date. So the study end day is going to be simply XX END Y, right? We're trying to generalize your formula, right? For an example, if I want to go with the examples now, let's say a few examples we can take. If it is going to be our adverse events, I can simply say that it is going to be AESTDY. All I changed here is I replaced my XX with the AE. Suppose if it is going to be like at the same end also, if you want to consider, I can simply say that instead of STD, I'm going to use the END. Correct? So this is going to be the simple example based on your whatever is going to be your domain. Based on the domain, you can simply change this particular one. Okay. Now, so what is going to be the formula that you can derive now to do this particular one? So what is the formula that you can try here? The formula that I can try here is going to be to calculate xx dy. So here you have it already, right? That is when xx dtc is greater than or equal to your this one, let's say RF STDTC. In that particular one, what you can say? You can simply say that I'll just call this as xx itself. Then your formula is going to be xx dy is equal to this. We have already done this, correct? When it is happening, it is xx dtc is greater than or equal to your RF STDTC. If it is the other way, if it is lesser than this, that is nothing but before, I can simply say that take this one, right? This is what we done, right? When xx dtc is nothing but is lesser than your treatment start date, we have done this. Correct? Now The same thing we are going to apply for remaining things as well. So what is meant by remaining things? What do you mean by remaining things? So what is your study end day? Because we need the study end day, right? So the study end day is equal to the study end day calculation. This also be required, right? Because I mentioned that there is going to be a study end day. So xx endy is equal to xx endtc. So here it is a start date, but here it is going to be end date. xx endtc minus rf sddtc plus 1. When it is, if my formula going to say that if xx dtc is greater than this, I'm going to call this particular one. That is study end day. When I want to calculate the study end day, all I want to do is going to be my, if it is going to be demographics domain, I can simply call this as a RF ENDTC minus RF start DTC plus 1. Okay. Suppose if it is the other way, I can simply call that as this. So the first one is going to be is that making sense, guys? All we try to do here is I want to calculate how to calculate my study end day. Suppose if it is going to be, for an example, I'm going with the adverse events. In the adverse events, we are going to have two things. That is AE and DCC, AE SDTC. Adverse event start date and time, adverse event end date and time. When these two are there, now my calculations are going to be AEDY, that is adverse event days, is equal to how to calculate your adverse event dates. Can someone please help me with this? Can someone please help me to calculate with this particular one? Anyone? Or at least can you help me with how to calculate your adverse end date? Okay, let me calculate this. AE STDY. Similarly, calculate AE ENDY. I want to calculate these two, right? So how to calculate? Can I simply say this particular one? Copy this here, copy this one here. This is end. This is going to be start. Correct? So how to calculate this particular one now? Anyone? Can someone please help me with this? Can I simply replace this with AE? Can I simply replace this with AE? Here also, can I simply replace this with AE? This is going to be my adverse event date and time of collection minus adverse event your start and treatment start date plus 1. Here also adverse event study end date, treatment end date minus your treatment start date. Can I do like this? If at all it is confusing you, please ignore it. All I want to do is I want to generalize this. When you're feeling difficulty to understand this particular one, you can simply leave this. It is not going to be that important. Clear, everyone? See, we have the adverse study end day. How to calculate the study end day is the question mark here. To calculate the study end day, I'm using this particular method. Everyone, is this understandable? Guys, some confirmation? All right, let me go with the next one, ARM. So ARM stands for, what is nothing but ARM here? Can I call this as a, ARM is nothing but description of a planned ARM, correct? So how I'm going to get this particular one? ARM is nothing but a treatment, description of your planned ARM. This is nothing but a planned treatment name, correct? So when it comes to this particular one, I can say that it is nothing but planned treatment code, right? So how we are going to get this particular value? We will populate this value based on randomization sheet, correct? So we are going to do the randomization sheet, right? Based on the randomization, we are going to populate this particular variable, correct? So I'm going to have an ARM here. I'm going to have the planned ARM code here, right? This planned treatment code is nothing but it is a short name for planned ARM. Maybe I can give you a simple example for these two together so we can easily understand this. So we have a subject, SUBJ ID, and we have ARM. Let's say we have ARM code. So I can simply say that the subject is going to be 111, the ARM is going to be drug A, and the ARM code is going to be DA. Let's say I have patient B, the planned is drug B, and the code is going to be D. It is a short name. Clear, everyone? All right. So what is your actual ARM? You planned something, but you are providing a different treatment, the actual treatment, right? Based on the randomization, you have two different groups, right? So you want to give a specific treatment to a specific set of people. You planned something, but when we go actually started our treatment, we are giving the different treatment as per the protocol only, but we are giving the different treatment. So now I can say that it is not a description of a planned ARM, it is going to be the actual ARM. It is the actual treatment that we have given to the patient. The same thing will go to the actual ARM code as well. That is, instead of planned, we simply replace this with the actual. Correct? So the same example goes good for this one as well. So the example here is going to be, so for this particular subject, let's say ACT ARM, and it is going to be ACT ARM code. So initially for the subject 1, we planned drug A, right? But the treatment that we have given is drug B. We start with the drug A, but we have given the drug B. So now we planned drug A, but we provided drug B to him based on the different parameters we have considered there. And finally, we have given the drug B itself. So now I can consider that this is the planned drug, but this is the actual drug that we have given to the patient. Is that understandable, guys? When it comes to this actual ARM and all, right, there is something called what happened for the unassigned subjects. First of all, what is called unassigned subjects? This is also important, so we need to understand this. So after screening, we completed the screening. After screening, in randomization stage, just a second. Right. After screening, in the randomization stage, some people will get dropped. Right? Those we can call it as unassigned subjects. The people who got dropped after the screening and during the randomization stage, whoever is getting dropped, we call them as the unassigned subjects. When unassigned subject comes to the picture, right? For unassigned subjects, arm value is not assigned. Then what is the arm code for them? The arm code for them is going to be not assigned. Similarly, for the same subjects, actual arm is also going to be not assigned, and actual arm code is also going to be the same. What is that? ACT, ARM, CD, which is also equal to not assigned. Correct? So if at all is there any unassigned subject, for unassigned subjects, we need to ensure that we are going to populate arm code, actual and actual arm code as not assigned. So what is called your unassigned subject? The people who got dropped after the screening and especially during the randomization stage, we call them as the unassigned subjects. Clear, everyone? This is all about your DM domain. Whatever we have done so far, this is all about your DM domain, right? So let's do one or two examples on this, and in the next class we are going to start with subject elements. We start with the next domain, subject elements. I don't take it today, but tomorrow I'm going to take this. Maybe not the subject elements, we go with the subject visits. We go with the subject visits tomorrow, right? So from here onwards it won't take time, it will be very quick. Okay, and parallelly I will start with the phone. So in tomorrow class we are going to start with our next domain, which is nothing but the subject visits, and parallelly let's start with our project as well, right? So let's take one project, parallelly we can start work on this particular one. Okay, and one more thing, guys. Do you want me to go with this task 1 to task 4 in the class? You want me to explain this? How do you want to proceed? Because you know the input, you know the output as well. What is the desired output? You write a code, and if you get your output, that is more than sufficient. So if all of you get your code, that is fine. Still, if you are expecting me to do that in the class, yes, I will do, but all I need is a confirmation. You want me to explain that in the class? Perfect. Sure, guys. Yeah, maybe day after tomorrow or sometime in this week we'll do that. Okay, sometime in this week we do that. Yeah, any questions from anyone? So today I'm stopping here because anyways we have completed the domain. So instead of starting a new domain today, let's start by tomorrow. Okay, any of you have any questions, please let me know. I can spend time for explaining your doubts. If not, we can wind up the session.
Murli, I just have one question. The thing which you said that we would be doing a project, so we would be taking DM domain as one of the project, wherein we would be creating the specification or the programming. That is how?
Yes. Yes.
Okay.
I'm going to provide you the raw data sets and everything, and I'll help you with how to proceed, how to continue with the project.
Okay. Yeah, understood. Yeah, thank you.
Perfect. Perfect. All right, guys. See you tomorrow then. Take care. Thank you.